Everybody. I think people are still trickling through, but it has just turned 9 o'clock, and so we better get started if possible. So welcome to the UK Pharmacogenetic Stratified Medicine Network annual meeting. I'm going to just give you a few slides on what's been happening over the last year or so, and then we'll head on into the main program, if that's okay. So my name is Munir Pirmohamed. I am chair of the UK Pharmacogenetic Stratified Medicine Network. I set this up in about 2010, and this is our 12th meeting. There were some years we missed because of various things, such as a pandemic and so on, but it has been going very strong since then. So what is the UK Pharmacogenetic Stratified Medicine Network? Well, it is basically there to serve as a vehicle to showcase advances in all aspects of personalized precision medicine, foster the uptake and diffusion of precision medicine into clinical practice, but also it serves as a vehicle to enable people to network with each other, develop new collaborations, and that interaction is really important. We have an interdisciplinary membership, from industry to patient representatives as well as part of the membership of the network. It is free to join. We're very nice. We'll give you a cup of tea if you come and join us and so on. So what's been happening over the last few years? Last year in March 2024, we held a meeting in collaboration with the Pharmacogenomic Network of Excellence, which is NHS funded, and the paper from that meeting has just been published in the British Journal of Clinical Pharmacology. It is open access, so you can download it. It tells you where we are with pharmacogenomics at the moment and what the sort of issues are, challenges are, and where we need to be able to head to. There have been other activities going on with regard to the network. We've had a webinar in collaboration with the Association of Physicians, which was attended by over 100 people. We also had a symposium on adverse drug reactions and pharmacogenomics. As you know, pharmacogenomics can be there for efficacy as well as safety. Most of the advances so far have been in the safety area, and for example, that has also led to the yellow card biobank, which is being developed, and June Rain is in the audience. So thanks, Jen, for really supporting the development of that yellow card biobank, and obviously Rich as well for working in collaboration with Genomics England to develop that. We had a workshop on precision prevention on the 5th of March 2025. Prevention, everybody talks about prevention, but I think in many areas, complex disease areas and so on, that will need to be much more precision prevention. And the paper has been prepared from that and so on, will be available very soon. Last year, I told you about the fact that we had been awarded a UK Regulatory Science Innovation Network in Pharmacogenomics. That was a 6-month project, which we wanted to really identify the challenge areas. And we identified 4 different challenge areas. And then we applied for a CERSI, a Center for Excellence in Regulatory Science and Innovation. So glad to report that we were awarded this CERSI in pharmacogenomics, which has now been working very hard since the beginning of February to be able to deliver many of the particular issues that we highlighted needed to be delivered. It's divided into 5 different work packages. The first work package is very much about developing UK-based guidelines. We're working very closely with MHRA here, particularly in terms of how we can look at the summary of product characteristics and where there may be changes needed in terms of summary product characteristics and so on. Clearly, pharmacogenomics is important to the industry, and the work package 2 focuses on the industry and pharmacogenomics. That team has already developed an industry pharmacogenetics network, which is going strong. Work package 3 is about upskilling the workforce. It's about education training, and we're developing modules with the British Pharmacological Society, which will be available online in order to be able to improve the skills and training in pharmacogenomics. Health economics is always important. Work package 4 rests on that, and again, work with NICE there as part of the work of the CERSI. And most importantly, what we're doing is very important for patients, public representation, and we have a specific work package which is dedicated to that. And for example, one of the work streams in that is looking at patient information leaflets. If you look at all the patient information leaflets that are out there for medicines, the amount of genetics data in there is very disparate, and the way it's formatted is, again, quite different from one PIL to another. And we're hoping to work with MHRA as part of this to be able to standardize that kind of patient information leaflets and the pharmacogenomics information in there. So what else are we doing? We've got a sort of mid-year workshop for the CERSI on the 4th of July 2024. It'll be online. Again, if you go on the website, you'll be able to get the sort of, registration details. Anybody can join. There's no fee for joining that particular workshop. And next year, at the end of the first year of the CERSI, we'll be holding a 2-day workshop in London on those days. So please save the dates if you want to come there. Again, that will be free to attend. We also have developed a workshop at the LRIG Drug Discovery Conference, which is being held on 22nd of October in Liverpool. This is going to focus on use of novel technologies to enable drug discovery and development. And we're covering many different areas: genomics, proteomics, AI, etc., as part of the drug discovery process, which is really important. And you'll hear today the need to be able to develop drugs, particularly for rare diseases and so on. So, you know, those many of the people who are speaking there work on different aspects and different diseases, from rare to complex diseases. We've also got some other upcoming events for the network. We're scoping a workshop on polygenic risk scores on the 14th of October. We're just trying to see where we can hold it, whether it's online or in person, and we'll let you know about that. And next year's meeting for this network is already scheduled for 17th of June 2026. So if there are particular areas you feel that we should be covering, then please do contact me. We're very happy to listen, and we want to be collaborative and co-design particular aspects in terms of moving forward. So thank you to all our sponsors, Agena Bioscience, First Data Bank, Gene Driver, Lakar, who are all upstairs. So please do visit their stands at coffee break and at lunchtime and so on. So thank you very much for your attention. So we will move straight in to the first session. And what I decided to do this time was to really get the 3 major bioresources that we have in the UK, which are fantastic bioresources, and get them to come and give a talk on what they have been doing, where they see their future opportunities lie, and then get a panel discussion going. And really, this gives you a chance to be able to ask them questions that you may really want to know in terms of whether you're using the resources or not, or whether you're wanting to use the resource for the future and so on. So it's a great pleasure, first of all, to introduce Professor Martin Rutter, who is with the UK Biobank. I can't exactly remember what your title is at UK Biobank, but I know Martin is a professor at Manchester University in cardiometabolic medicine, and Martin's work involves a lot of different areas and so on. So Martin's going to talk about what UK Biobank is doing at the moment. Thanks very much, Martin.
Thank you very much, Munir. Can you hear me okay? Just wondering whether I need to stand close to the mic. So what a fabulous lot of events being organised by the network. It's really impressive, and I'm very grateful to Munir and the team for inviting me to talk today. So I'm going to talk about UK Biobank's relevance to pharmacogenetics and stratified medicine. I'll talk a bit about what makes biobanks so special, the range of data that's available in UK Biobank, and some examples of studies in pharmacogenetics and stratified medicine, just to give you a feel for the opportunities available through UK Biobank. And I'll say a few words about future developments in Biobank that are relevant to these fields, which again are quite exciting. So why is Biobank so special? Several reasons. It's a huge number of people in this cohort. We studied 500,000 individuals recruited about 15 years ago. They were between the ages of 40 and 70 at the time of recruitment, and they've been followed in the NHS for health outcomes. We collected a lot of information on these individuals at the baseline assessment, and we've continued to collect questionnaire data and other data. I'll talk about some of the detailed stuff that we do later, but we've got a huge amount of information on these individuals that's relevant to these fields. And we've got 15 years of follow-up. So we've got hospital records and other data on their what's happened to them since the baseline assessment. Probably the most important aspect of UK Biobank that makes it so popular for research is the easy access that we have for our data. And any bona fide researcher around the world can have access to the data, and we give everything away for £9,000 for a 3-year data access, so it's relatively cheap. So these are the sorts of data that we've got. I don't have enough space on the slide to cover everything, but we've got demographic data, clinical data, biomarkers, hematology, biochemistry, environmental data. We've got lots of omic data. We started with array data. We have whole genome sequencing data on the whole cohort. That's 500,000 people. Proteomics at the moment in 50,000. We have NMR metabolic data in 270,000, and we're planning to increase that to the whole cohort over the next couple of years. We had physical activity monitoring in 100,000 individuals. And as I mentioned, we've got health outcome data from hospital records in everybody, cancer registry data, ONS data for mortality, and we're trying very hard to get access to primary care data. I'll talk a bit more about that later. But this is the sort of impact that Biobank is having in the research arena. We have, at the moment, about 20,000 active researchers using Biobank. There are more than 3,000 active projects. The resource has generated nearly 17,000 publications since its inception and 500,000 citations. So it's staggering. And as you can see, the exponential rise in the outputs, I think we're entering the golden age of UK Biobank. And it's partly because the cohort's getting older, they're developing disease, and the resource is becoming more informative about those diseases. So what about pharmacogenetics? What sort of studies have been done with Biobank? And I can't cover everything that's been done, obviously, but I just want to give you a flavor of what's possible. This is really a bird's eye view of the cohort. So it's looking at the prevalence of genetic variants that are relevant in the pharmacogenetic space. So we looked at the whole cohort. The group looked at 14 variants that were defined by the Clinical Pharmacogenetics Implementation Consortium as actionable, that is, that they have a clinically relevant impact on drug effects. The researchers used the array data in 500,000. We had exome sequencing data at that time in 50,000. We now have it on the whole cohort. We had prescribing data on 200,000 individuals, and that was used in this study as well. We'd expect to have more when we get access to the full primary care data. And what I'd like to show here is that this is the number of pharmacogenetics alleles that were present in these individuals by ethnic group, and you can see that the average number of variants in the population of UK Biobank was around about 4. Pretty much everybody had at least one of these variants that would have predicted some sort of variation in the response to a particular drug. There was slightly higher prevalence of these alleles in some of the ethnic minority groups. So just showing the relevance of pharmacogenetics to the population. The researchers then went on to look at the drugs that had actually been prescribed to these individuals in Biobank to see whether they'd been prescribed drugs that could potentially be affected by these genotypes. And in fact, about a quarter of them had been prescribed medication that may have been influenced by these genetic variants. So the most common drug was Brufen, simvastatin, and amitriptyline were there as well. So just showing the relevance of pharmacogenetics at scale in the Biobank population. This is another study that looked at using the electronic healthcare record data in Biobank to look at this time the impact of drugs on outcomes like changes in cholesterol, changes in HbA1c, changes in blood pressure in relation to the genotype. So it was electronic healthcare records, prescribing data again from primary care, and clinical data. They used genetic data on 14,000 people in the UK Biobank. There was some replication done in all of us. They focused on the drugs that are relevant to cardiometabolic conditions, so statins, metformin, antihypertensive medication, for example. So they looked at genotypes that influenced the impact of these drugs on these outcomes. And they replicated many findings that had been found in previous studies, and they did identify a novel variant that influenced the HbA1c response to metformin. So I think it was just showing in this study that the prescribing data and the biomarker data in UK Biobank, the lipids that are available on HbA1c, for example, can be used to study pharmacogenetics in Biobank. And the resource is going to get richer, as I'll mention later in some further examples. And there's many, many more. So this study was looking at the influence of genetic variants on proton pump inhibitor impact on migraine. We've got statins and LDL cholesterol responses. This was a nice study looking at clopidogrel and vascular outcomes, showing how genotype influenced the risk of developing major cardiac events. So a 9% higher rate of these events in people who had a loss of function allele, but a 15% lower risk of hemorrhage in the same individuals. This study was looking at drug dose and how genetic variants influence the drug dose, and they actually looked at 200 separate medications and so on. So I'm not going to go through all of these, but they go on and on. But it just shows the potential of the biobank resource for doing this sort of work. I'd like to move on now to talk about stratified medicine and the impact of biobank in this field. I'm going to talk first of all about disease prediction, and I'm using an example just looking at clinical data, so we don't have any genetic data involved in this study, and I'm focusing on an outcome that's particularly important in our country and worldwide at the moment, which is the risk of developing dementia. So this risk score was used to identify risk for developing dementia in follow-up. They used clinical and sociodemographic data only, along with ApoE status, and as I said, looked at the risk of developing dementia. And the discriminatory ability of this model was reasonable, within an AUC of about 0.8, and they showed that there was some replication in the Whitehall study with similar results, and this particular risk score outperformed others that were available that had used clinical data to predict incident dementia. So just an example of using clinical data to predict risk, which is obviously very relevant in a precision medicine setting. But I'd like to just show how that compares with another study that looked at adding proteomic data to the clinical variables. So again, we're looking at a risk score. This time we were looking at a smaller proportion of the biobank population. At this moment in time, we have data on about 50,000 individuals with proteomics, O-link proteomics, and it was measuring about 3,000 proteins at that time. Using the hospital records, we'd identified about 14,000 cases of incident dementia over 14 years, and there were 4 proteins that particularly captured the risk of developing dementia. And the model performed very well indeed, with an AUC of around 0.9. As you remember, using clinical variables alone, we get an AUC of about 0.8, so a significant step up in the ability for these variables to predict incident dementia. And an interesting finding was that these changes in proteomics occurred 10 years ahead of developing dementia in many cases, so suggesting the power of these proteomic measures to predict incident disease in individuals. So, very nice study. Again, another study that's looked at proteomics and its ability to predict disease comes here, and this time we're looking at multiple diseases. Again, it's a relatively small proportion of the total biobank population because of the limited proteomic data. The authors of this study were looking at 218 common and rare diseases and the 10-year incidence and the ability of proteomics to improve the prediction. They found in many examples that just between 5 and 20 proteins had the ability to perform very well in predicting individual diseases, and those markers outperformed clinical variables significantly, and even when we added, the authors added assay data to those prediction models, the proteomics still outperformed those. As you can see, well, this is for 67 diseases, the median improvement in the C statistic. This is another measure of the discriminatory ability of the model to identify or exclude the development of disease. Overall, the C statistic improvement was about 0.07, but you can see in some of these examples there was quite a large increase in the C statistic for individual diseases. These are endocrine diseases, seem to do quite well, cardiovascular diseases, and cancers. There seems to be a larger increase in the C statistic for some particular diseases, but overall, a significant improvement in the C statistic. There was some replication of these findings in UK Biobank and in EPIC Norfolk. So I think just, again, showing the potential of UK Biobank to demonstrate the value of some of these omics data for disease prediction. Many of the audience here will be very familiar with polygenic risk scores, and UK Biobank has contributed significantly to understanding of these. Of course, I think we all know probably that the genetic risk for many common diseases is linked to the genetic risk associated with lots and lots of alleles that have small effects. And the idea of a genetic risk score was developed from the first idea back in 25 years ago to 2009 when the first study was published. This was before the genetic data was available in UK Biobank. That was a schizophrenia genetic risk score. But one of the first ones I could find in Biobank that was important was this one from 2018, where Kira showed that the risk for developing disease in the people who were in the upper, say, 3% or 5% of the distribution of the genetic risk score it was really high. So they were at the same sort of risk as people with monogenic diseases like familial hypercholesterolemia. So this particular study got a lot of press because it just highlighted the potential value of genetic risk scores for some individuals, not for everybody, but for those at the highest risk. These risk scores really do make a difference. So we're looking at the distribution here of the polygenic risk for atrial fibrillation. So these guys up here in the upper 5% of the distribution have a very high risk of developing these diseases, diabetes, breast cancer, and so on. They looked at cardiovascular disease particularly as well in this study. They showed that the people who were in the at the upper 8 percentile of this distribution for the coronary risk, they had about a threefold risk of developing coronary artery disease compared to the background population. So again, the same sort of risk that someone with familial hypercholesterolemia might have. So really dramatic. Overall, the area under the curve for coronary artery disease prediction was about 0.8. So that was one of the first genetic risk score studies in UK Biobank, and of course there's been many more since then. Bupa now uses genetic risk scores in their clinical assessment of some individuals, and this is just a couple of examples. This is predicting breast cancer. They use genetic risk score for breast cancer in Bupa. These are the people with who are in the top 3% of the distribution of the genetic risk score for breast cancer. This is the similar data for prostate cancer, just showing how these risk scores can discriminate very nicely people who are at risk compared to those who are at low risk. There are clinical trials going on now to look at the value of adding genetic risk scores to other screening methods, particularly in the breast cancer arena and prostate cancer arena, and they're very exciting as well. And the clinical impact of these genetic risk scores is starting to just nudge its way into clinical medicine. So there's been a pilot in the northeast of England looking at the value of polygenic risk score for coronary artery disease when added to the Q-risk2, and it seems to be feasible, acceptable in primary care, and potentially actionable results. So it's certainly promising that these interventions may be available in the NHS routinely at some point in the future. So I just wanted to finish with just a couple of words about machine learning, because obviously I think the options that are going to be available to us through these technologies, artificial intelligence, machine learning, are just immense in the future. This was a study using biobank data again. A machine learning model was developed that used all the phenotypic data that's available in UK Biobank to predict 3,000 diseases in UK Biobank. And this machine learning model, which is nicely called Milton, sounds very friendly, doesn't it? I definitely want to use that one. But it outperformed the genetic risk scores by and large in terms of its ability to predict incident disease. I was interested in the proteomics aspects of this paper because it showed that even when the machine learning model was running at full tilt, adding proteomics data did actually improve the overall prediction of the models by, and the AUC average improvement was 0.03, which isn't massive, but for some diseases it would have been, you know, a lot more than that. But that was the average improvement in AUC by adding proteomics data to the machine learning model. It has the ability to identify people who are at some points in the follow-up were classified as controls who then went on to develop disease. I think it just means that it can predict incident disease early. There was some validation of the data in the FinGen biobank as well. So there's no doubt that machine learning models are going to be very relevant in this space, as we'll all probably know already. What about the future? Biobank has several things planned that I think are going to be relevant to the field. We're eagerly waiting the primary care data. Wes Streeting gave an announcement that consented cohorts like UK Biobank would have access to primary care data. All our biobank participants are consented to provide their primary care data, but we find it very difficult to get our access to it. But we expect it'll probably come online next year. And I think that's important because we'll have prescribing data on pretty much the whole cohort. And many diseases are managed in primary care, dementia, diabetes, for example. So the number of incident cases is going to probably double in many instances. So we're going to have a lot more statistical power to identify relationships with those important disease outcomes. As I mentioned before, we're going to extend the proteomics data and metabolite data to include the whole cohort, which again will increase the power of the study. We're interested in getting digital histopathology images into UK Biobank. Many of our participants are developing cancer now, and we may be able to understand the biology of those cancers through sophisticated approaches of analyzing those digital images. So that's a very exciting area that we're just getting into at the moment. We're going to bring in initially 1,500 images over the next year and start that piece of work. Another area that's of interest is the brain health study. We're interested in doing some detailed phenotyping of people with dementia or cognitive impairment. We've just started a pilot to assess our methods, but we're going to be using brain MRI imaging, cognitive assessments, blood sampling, which will include proteomics, of course, and wearable devices to better phenotype these individuals to identify the underlying biology that drives Alzheimer's and these diseases. At the moment, the phenotyping is very poor for many people with dementia, so that's an area of great excitement for us. And we're also planning to repeat an assessment of the whole cohort at some point, which again will give us a lot of information about the natural history of many diseases that we don't have at the moment. So in conclusion, Biobank is a leading biomedical research resource. I think the combination of the genetic data, the proteomic data, and other assay data with the health outcomes at this scale is really unprecedented. I think our ease of access to the data makes us a very popular resource for researchers and explains largely, I think, its extensive use. And as I mentioned, I think we're entering the golden age of UK Biobank. We're going to have more incident cases. Our cohort's getting older. The average age is about 73 now. We're going to be doing more assays on biological samples like the proteomics and metabolite data. We've got primary care data coming in. So I think the resource is going to be more and more valuable for research in the proteomics, in the pharmacogenetics and personalised medicine space. So, I'd encourage people to use the resource and make the most of it. So thank you very much for your attention.
So we will take questions at the end when we have all three talks done. So next speaker is Dr. Richard Scott, and Rich is the Chief Executive for Genomics England. And obviously Genomics England ran the 100,000 Genomes Project, which was sort of born at the time of the Olympics, wasn't it, in 2012. And has done some fantastic work, and also working on pharmacogenomics, but mostly on rare diseases and tumors and so on. So, Rich, thanks very much for attending today. So good to hear from you on that.
It's a pleasure. Thank you very much for having me. And I think a lot of what Martin has spoken about is relevant to us, and how it helps us project forward to think about where we might be going, where we can play a useful role. So, as Munir says, I'm the CEO at Genomics England. I'm a rare disease doctor by background, so I'm a clinical geneticist. And I joined Genomics England about 10 years ago, and have been the CEO the last couple of years. And we started, and we always start with where genomics is ready to jump into clinic or is playing a role already. And if you think about the first 5 or so years of our existence as an organisation, it was very much about running this single program, the 100,000 Genomes Project, which was asking a question: could whole genome sequencing play a role for diagnostics in 2 key settings, in rare conditions and in cancer? And that program helped us sort of understand how that could play out, but also how we could play a role ourselves on an ongoing basis, how that same blueprint, if you like, for asking questions, developing resources, linking clinical care with research could help us explore the use of genomics more broadly. And I'm going to give you a flavour over the next 20 or so minutes of how we started off, what that's taught us, and some of those questions, some of the areas that we see as really important to explore in the coming years. And as I say, we started in these 2 areas. We work always in partnership with the NHS. Our research programmes are run at scale with the NHS, so that we understand as we go a lot more than we might about how this plays out in day-to-day care, because this is about understanding and developing evidence that can lead to changes in commissioning, changes in what can happen on the ground. And following on from the 100,000 Genomes Project in 2020, so just actually sort of in the middle of the big year of the pandemic, the NHS commissioned and launched the use of whole genome sequencing in diagnostics in those 2 settings, in rare conditions and in cancer. And here are 2 stories that come from that launch. On the left, little boy, baby Oliver, he's called, who was a boy who developed a lump. It was unknown what it was. His family were given great care at a specialist hospital. The concern was that it was a malignant tumour. But it was an unusual tumour, as is often the case in childhood tumours. Whole genome sequencing was performed on a diagnostic basis in the tumour and helped to understand that actually this was a benign lesion, and he could avoid the need for chemotherapy in a way that actually looking in conventional ways just couldn't. On the right is a boy called Charlie. There's a nice, you can see this is a Sunday Times story that came out in the last few months, about a boy who really demonstrates the benefit of linking clinical care to research. So in rare disease, as many of you will know, making a diagnosis is a real challenge. Even when you apply whole genome sequencing, our knowledge today is way better than it was 10 or 15 years ago, and we're really proud of those advances. But still, particularly with children with rare complex conditions, even when you know, looking at the constellation of features, this must have a genetic cause. Still, perhaps half of the time in those settings, you can't find an answer. But what we know is that the rate of knowledge is rapidly accumulating, and each year still 100+ genes are identified that can link to rare conditions. This boy, Charlie, is one of the children who had genomic testing to find a diagnosis. When we first ran the test, nothing was found. He joined the National Genomic Research Library, which is now what we call our cohort, that was, if you like, the founders of it, were the people who joined the 100,000 Genomes program. But now people who join our other research programs also join it. And researchers in it and teams at Genomics England keep looking for an answer. Last year, a new cause was found, probably one of the most common causes of developmental disorders. It was hiding in between the genes, so an area that techniques other than whole genome sequencing didn't look for. It's actually quite common. It's probably 100,000 children worldwide, we think, might have this condition. Still rare, but one of the most common causes that had just been hiding in plain sight. And this story is about the impact that that condition had for him and for the family. Still, though, it's rare for there to be a targeted treatment in these children. We'll come back to that. And this is why it's so important to keep on learning, keep on pushing the boundaries. In the last year since this discovery, a bunch of other genes, quite like the gene that, that is the cause in him, it's called RNU42, have now been found because the researchers are beginning to get their eye in for the parts of the genome that we should focus in on in particular. We're very bad at looking between the genes because we don't understand the language, if you like, that is being spoken from a biological perspective. The other really touching thing about this story is, genes are often, they're given these sort of very mechanical names, so RNU42 and the other genes that are like it are all called RNU 22, etc. The families have called this syndrome Renew syndrome to sort of riff on the very sort of alphabetical and numerical name for the condition, but to express the renewed hope that these diagnoses have given them and from meeting each other. So that's why we see that critical role of linking clinical care on the left of this infinity loop that we like to draw, which represents the role we play linking and working with many others across the ecosystem. As I've said, always with the NHS, but working with many of you, researchers in academia, in industry, and other partners, because joining this all together and making this work is very much a team effort. Perhaps to tell you a little bit about how the sorts of lessons we've learned and how we work, I'll tell you a bit more about the 100,000 Genomes project. So as I said, it started off with that big question, can whole genome sequencing be used for diagnostics in cancer? So this is a summary of the participants who joined the 100,000 Genomes project with cancer, 17,000+ of them with a range of different cancer diagnoses. Similarly, in rare disease, a larger number of participants, often family members and those affected, again with a big range of conditions. And during the course of the program, we didn't just sequence their genomes, we also, and supported research, we also fed back relevant findings to them to guide their care. And they were signing up to that as part of the consent that they joined the program with. They also consented to broad healthcare research in, as I say, what we now call the National Genomic Research Library. And following in, learning many lessons from biobank, and following that template, thinking about building up the data that we could alongside that genomic and the data that came in at the point of testing, if you like. So while they were joining a research program, they joined very similar to how you might undergo testing in the cancer or rare disease setting. Because we were doing testing in the context of NHS, and when we engaged early as an organisation about the right consent model, we took quite a conservative approach to how we hold the data, and it's one that definitely comes with downsides, holding the data in what people might now call a secure data environment or a trusted research environment that researchers can access once they've gone through an approval process through an access review committee. But it's one that gives us, we find, has very high take-up by people. So therefore, you get a really broad range of people joining the programs. And in that research environment, we can bring in other types of data, so other clinical data, very similar to what Martin set out for UK Biobank, and similar to the approach that Our Future Health and Raghib will I'm sure, describe. Like Biobank and Our Future Health, we're really excited at the prospect of primary care data coming in, and with that, a greater window on what's going on from a prescription point of view. And we've also, as I'll describe, brought in other types of data alongside, responding to the sorts of opportunities that we see for the participants who form our cohort. So currently in the research environment, we have 160,000 genomes, which is now a mixture of people from the 100,000 Genomes Project, people who've now undergone diagnostic testing in the NHS routine service and have signed up to say, yes, I want to take up this research offer in the National Genomic Research Library, which is a routine offer if people are having that whole genome testing. And also some of our other programs, for example, a diverse data program that we've run to increase the diversity of the participants in our program. And we've begun to bring in other sorts of modalities of data. So, for example, digital pathology images on those 17,000 cancer participants in our program, and also radiology imaging. In our rare disease program, we're building out the omics data set that we can display on those participants to enrich the sorts of research that our nearly 2,000 active researchers can access. One of the key metrics for us is impact back in clinic, and you can measure that in different ways. You can measure it, for example, in the adoption of genomics at scale in the NHS in new settings, so the adoption of whole genome sequencing, the adoption of new uses of genomics, so for example, individual clinical indications. So we've seen over the years the range of cancer and rare disease setting in which the NHS is commissioning evolving. It's also about individual benefits for patients. So Charlie, the boy that I showed on my first slide, is one example of now more than 4,000 families who benefited from specific answers to them that have come from the research. And that is growing, and that rate of knowledge is not abating. There will come a time in the future, I'm sure, where we've understood more and that begins to flatten off, but there's no sign of the rate of learning in rare disease or cancer flattening off. As we've heard from Martin, and the sort of work that has gone on in the UK Biobank cohort and elsewhere, rare disease and cancer are two really important areas, and they'll continue to be critical areas in terms of the impact of genomics. But really, it's just scratching the surface, and I'm sure many of you come here today because of exactly that sense. We see potential at multiple different areas of clinical medicine, at multiple different time points in life for genomics to play a role, whether that's in pharmacogenomics, whether that's through risk prediction of common disease, whether it's using genomics more for early diagnosis in cancer, for example, where at the moment genomics predominantly plays a role in diagnosis once a diagnosis is well established. And we see that potential as a challenge that we need to prepare for and think what it is that needs to be in place as an ecosystem to be ready, so that genomics, as the evidence evolves, could play a role in perhaps up to half of all healthcare encounters. And genomics really becomes something which we can all benefit from. And there's a lot in that word everyone to think about, in terms of different settings, in terms of different communities. It's a really challenging goal, and one that I think one will always have to sort of fight to achieve and do better. And that's something that, as we've reflected on that big potential that genomics can play, and on what we've learned already, on what we see as important for the role we play sitting at that intersection between routine care, working with the NHS, supporting their work, and supporting research, and helping the UK retain that leadership position it has in terms of being able to harness this power of genomics. So to support the provision of evidence and digital systems to be ready for that potential that in maybe 10 years we could be using genomics absolutely as part of the fabric of routine care. And so that whatever sort of innovator you are in this space, whether you're discovering a new gene, whether you've got a great new sequencing technology or a great AI algorithm or risk prediction tool, or a new therapy, the UK, with us being one part of it, is able to say, yes, we've got the tools where you can help prove that, but it's not just about sort of discovery and proving. You can also know that this could be something that patients can benefit from it, and that sort of last mile piece is often the hardest. So our role in that is to build digital infrastructure and tooling that's specialist for genomics for national scale use in clinical care. So our analytical algorithms, bioinformatics pipelines are being used for those national whole genome sequencing systems in the NHS, and will continue to be so. The national scale research use, so our national scale research library, and then in different ways to use that blueprint that we first learned about through the 100,000 Genomes Project to help develop the evidence and the learning that isn't just the scientific and the clinical discovery, the health economics, but it's also the broad understanding of how this could be used in clinical practice. And that includes evidence on public opinion and expectations and red lines in terms of how data might be used. Because that vision, a vision of genomics being part of routine care, is much more about genomic data being a routine part of your health record that can be called upon and used to inform how you're best looked after, how you make healthcare decisions for yourself and with clinical colleagues. So we've heard about Charlie's story, we've heard about where we are in rare disease, but I touched on, that we're delighted by the number of people that we can now benefit with genomics in terms of diagnosis for rare disease. But we're just beginning, really. We know that, for example, we think of rare diseases often in very specific settings, but we know rare variation is important in a lot of other settings. We have a partnership with the MHRA and the Electrical Biobank to understand what's underlying some of these adverse drug reactions that we don't understand. Is there more to find there? We also are working hard to understand how we can support the testing of rare therapies, which, as I said, are too rare at the moment. It's about 5% to 10% of rare conditions have a specific therapy. In cancer, the same story is true. Increasingly in cancer particularly, the use of AI and ML is really beginning to take off in our data set and understand and help us identify some new patterns that we couldn't see before. And increasingly, a world where genomics is playing a role in trial stratification in cancer is plain to see, and doubling down on that is critical. The advantage of having those feeds of ongoing health data, just as the other consented cohorts do, mean that you can track outcomes. This is one use of a particular algorithm, identifying a particular sort of DNA repair deficiency in ovarian cancer, helping us to, understand how that algorithm works, but also see that can predict outcomes, because it predicts how the tumor responds to particular therapies. So what are those lessons from those settings that that can help us say, what are the questions we need to ask in the future. One of the big areas that we've seen opportunity to the last few years is in expanding from from rare disease diagnosis to getting upstream. And so we've been running a research program called the Generation Study, which is asking the question, can genomics be useful alongside the routine heel prick to expand the number of conditions that we can look for in newborns, to screen for them, treatable, severe conditions. We've now, the Generation Study will recruit 100,000 babies. Ultimately, we've recruited 12,000 so far, and beginning to return findings of the sort that you can imagine here. We can look, for example, with genomics, you can look for spinal muscular atrophy, a condition where there's an NHS-approved treatment to really substantially change the outcomes of children born with a severe progressive neurological condition. We're looking for more than 200 conditions in the program. That compares to 9, shortly to be 10, that you can currently look for on the heel prick. Evidence in this area, as I've said, it's not just about health economics, clinical utility, how it plays out in the health system. It's also about people's views. There are big questions to be asked, and I'm sure we can explore that when we come to the panel section. And that's why the ethics and the engagement is a really key part of all of our programs, including the Generation Study. And that study has helped us start to think about that conversation about the lifetime utility of genomic data and what people's attitudes and expectations and comfort are with genomic data becoming part of the health record, and in what settings. One of the areas that we've explored over the last year through a public standing group that we've set up to help understand attitudes to that in the context of our newborns program is pharmacogenomics. And we can see, complementing evidence that others have produced, that there's real appetite in the public for the use of pharmacogenomic data in their routine health record, and we see that's a real area of opportunity. More than 90% of people in a recent study led by QMUL that we supported said that they would want to sign up to genetic testing to inform prescription decisions, and for that data to be something that could be reused. That's the sort of message that helps us look to the future and say we think, just like in the newborn genomes program, we think there are similar questions, quite a lot detailed implementation questions. I think in the adult and routine use of genomics, a lot of this isn't just about clinical utility, it's also about the digital wiring and the clinical pathways you need to establish to get them to work routinely. We can't expect everyone to become an expert in variant interpretation. That's never going to happen. Just like prescription decisions on the basis of allergy, what are the ways in which we can build on the great work that many of you have done to say, how do we simplify this system? How can you turn it to something that's really accessible? How do we develop the systems that can support that sort of decision making? So we're really excited about that opportunity and how we might bring that same sort of blueprint mindset that we've developed in the 100,000 Genomes Project, using our newborns program, the Generation Study, to ask those sorts of questions. And as I say, this is an ecosystem thing. It's not a one organization can do this. This is very much about partnership working, about how we develop that evidence, the digital infrastructure, the clinical pathways, the understanding of what people in the general public are comfortable with, so that we can really harness genomics to make a difference in the future. And so the UK can be the sort of place where people like yourselves, people from all sorts of different parts of the life sciences ecosystem, can know that if you've got a great idea, you can discover new things in that area. If it works, you can prove it, and if you can prove it, it can be taken up. So hopefully that's given a sense of how we're working, how we're thinking, and I'm looking forward to the panel discussion.
Thank you very much, Rich. A great talk. So last speaker in the session, Raghib Ali, who's an epidemiologist by background, but also works in acute medicine as well, according to his biography, is the chief executive of Our Future Health, the new kid on the block, but bigger in terms of numbers of people being recruited and so on. So Raghib, we look forward to hearing from you about our future health and where that's heading.
Okay, good morning. Thank you, Munir, and to excuse me, and to the British Pharmacology Society for inviting me. So actually, I did train in clinical pharmacology once upon a time, so I did speak at one of these meetings a long time ago, but it's great to be back. So as Munir said, we are a relatively new study, certainly much newer than the UK biobank, which is actually the first study I worked on nearly 20 years ago, and Genomics England. So as you'll see, although we only started recruitment a couple of years ago, we have made good progress. But I'm mainly going to speak about the opportunities for hopefully many of you as researchers to use the resource now, but to give a bit of background, because I know it's, as a relatively new study, not all of you are familiar with it. So in terms of why the study was established, all of us work here in healthcare and will be familiar with some of the challenges mentioned on this slide. I won't go through all of them, but with an aging population, increasing levels of multimorbidity, and healthy life expectancy hasn't increased for some time, but even now we're seeing decreases in life expectancy. So people are not even living longer anymore, let alone living longer in healthier years. So that's a huge challenge. Secondly, in working age populations, particularly actually in younger age groups, we're seeing very large increases in mental health disorders, which are having an impact on people's lives and on the economy. And thirdly, there's still very large inequalities, particularly by deprivation and geography, and also by certain ethnic groups. So there was a need to have a new prospective cohort study which could help to tackle some of these issues by focusing more on prevention of these diseases. So as Munir said, I have spent most of my clinical career in acute medicine and worked kind of on the right-hand side of this diagram in A&E, seeing patients with heart attacks, strokes, cancers that have already spread, advanced dementia, kidney failure, etc. And of course, all of those diseases have developed over years, if not decades, and we see the end point of that when it's too late both to cure people, certainly, but even for a good outcome. Most of them will have very poor outcomes by the time we see them in A&E. But as I said, those diseases do develop over decades generally, and we should be aiming to move towards a new model of care. So what's described here as precision public health, also called precision population health, where we can identify people at higher risk much earlier in the natural history of disease through risk prediction tools, through new biomarkers, through a combination of information that we can collect from participants, including genomic data, diagnose people as early as possible, or find those, as I said, who are at highest risk, and intervene as early as possible to prevent those diseases and keep people in good health for as long as possible. So on here I mentioned some of the tools that are now available that will help us to move towards this new model of care. It's not an exhaustive list, and in some ways it's slightly biased towards kind of more biomedical aspects of disease. Of course, most diseases, social determinants and lifestyle factors, behaviors, etc., are extremely important. In fact, the majority of differences are caused by those differences. But at the same time, these technologies will help us to identify those people as high risk much earlier, particularly things like polygenic risk scores, integrated risk scores, biomarkers, etc., proteomics, imaging, circulating tumor DNA, and then some of the earlier therapies. And again, this list focuses more on pharmacological interventions, but non-pharmacological interventions, digital interventions will be equally important, particularly if you're trying to start treatment much earlier in the natural history of disease. So our Future Health was established by the previous government as a public-private partnership between government, industry, and medical charities and the NHS. You can see here many of our kind of industry partners, charity partners, working across all areas of disease. And as we move towards the next phase of our Future Health, which is really about implementing what we learn from the data we've collected, the collaboration with the NHS will be even more important than it is now. So our Future Health is a research program. It's primarily about establishing the evidence base for new approaches to the early detection, prediction, and intervention of common chronic diseases. So the first thing we needed to do was to establish the resource. And this is what we've spent. I've been at our Future Health for about 3 years now, and we've spent most of the last 3 years really trying to put the foundations in place to build a resource of 5 million people. So a very ambitious target, but that was felt to be necessary because for prevention, most people don't develop individual diseases across a cohort of 5 million people, even over a 5-year period. In terms of a study that you wanted to start as early as possible, you need hundreds of thousands of people potentially to start those interventions. And so having 5 million people across all age groups, ethnic groups, geographic areas, etc., will enable us to do that. And it's for both discovery and translational research, so it's not just a observational cohort. As you'll see on the next bullet, we do have consent to go back to our participants and give them information about their own risk of disease. This was something different from some of the previous cohorts. So when I worked on biobank 20 years ago, there was some discussion about this at that time, but it was felt rightly at that time that it would be almost impossible to provide feedback to participants, and that was almost certainly the case then. Now, things have moved on. We do have the opportunity to use both existing systems, but hopefully to build new systems that will enable us to give people information about their risk of disease and have clinical pathways and service pathways in place to deal with that. As I said, both based on genetic and non-genetic information, and enabling risk-stratified screening programs. So currently, all screening programs are generally based on age. Age, of course, is a very important risk factor, but it misses people who are at higher risk, who are at younger ages, and it includes people who are at lower risk just on the basis of age. So by combining genetic and non-genetic information, we can have a much more tailored approach to screening based on risk. Thirdly, we have got consent to go back to our participants and to do additional studies. So these could be recalled by genotype studies, recalled by phenotype, collecting additional samples, questionnaires, and taking part in clinical trials. So interventional studies ultimately will provide the best evidence for these new approaches to prevention. And fourthly, if you get a cohort large enough, even with a couple of million people, which is across the whole country, that would be so much larger, particularly in terms of risk factor information, than we have anything at the moment. So most of the surveys of that type are in the tens of thousands. But it will also allow us to look at the impact of policies at national or regional level on people within the Our Future Health cohort. Okay, I know many of you, I'm not going to ask because it's between you and your kind of consent whether you've joined, but many of I know will have joined Our Future Health. But in case you haven't, this is what it involves. So we have, it's mainly a digital journey. So you join online after reading the participant information leak, consent, fill in the questionnaire, and then attend an appointment to give a blood sample and give some physical measures, blood pressure, height, weight, waist circumference, and for the first 1 million people, we had a point-of-care test for cholesterol. Now, of course, and speaking to this audience, you'll be very familiar with the unique opportunity we have in the UK by being able to link our data that we collect through our Future Health to the NHS datasets, and in the future to other datasets as well. But the NHS datasets on their own really are a gold mine, for useful health information, both looking retrospectively and prospectively. And you really can't do this at this scale anywhere else in the world. Our baseline, as I've described, we have relatively simple phenotyping. So the questionnaire, some physical measurements, and a genotype array. But looking ahead, hopefully within the next 12 months, we'll be able to start looking and adding in additional data sets. So primary care, we're told, is coming very soon, which will be really very important to enhance the resource. Also to move to imputed data from the genotype and then whole genome sequencing after that. Some additional data sets mentioned there, the data from the devolved nations as well. And then looking a bit further ahead using wearables, proteomics, methylation, etc. So the opportunities and administrative data sets. So in terms of social determinants of health, we can now get that from many of the administrative data sets, whether the census, DWP, HMRC, education, etc. data with a participant consent. So we would go back to participants to ask them if they are willing to share that information. So just to spend a couple of slides on our kind of genotype array. This, partly because any test on 5 million people is going to cost a lot overall. So we've started with the genotype array. And it's relatively low cost to do that now, not much more than some of the standard blood tests that we do. But even on a genotype array, we have enough information there for polygenic risk scores. We have got most of the GOS catalogue, and we have included some rare variants as well from the ACMG and ClinVar sets. It's also been enhanced for pharmacogenomic research and for predicting blood types, and we worked with Munir and colleagues to put that genotype array together a few years ago. So although it's a research use only genotype array, we will be able to run research studies, and the findings from those will be then applicable, hopefully, to be implemented by the whole NHS using clinical grade assays. So as I said, we have enhanced it for pharmacogenomic content. It mentions there some of the numbers in terms of the star-allele and variant coverage scores, so 1,700 targets for over 50 genes. We have over 6,000 annotated variants from the PGX database, PharmGKB, CPIC, PharmVar, and ClinVar. And we also have target amplification to allow for pseudogene disambiguation, which I hope means more to you than it does to me, for the experts in this area. So although I've trained in clinical pharmacy, one, it was a long time ago, and two, it was mainly in phase 3 clinical trials, so I haven't gone into pharmacogenomics in detail. The primary care data, as I said, will be coming soon. We've been promised that. We have the dispensed medicine data set, which is also very helpful, because it tells you not just about prescribed medicines, but also those that have been dispensed. And of course, the opportunity here is to ensure that people, participants initially, and then people across the UK receive drugs that are most likely to work for them, so enhanced efficacy, but also to reduce the risk of adverse drug reactions. And many of these, and the work that Munir and colleagues have done has shown that this is already ready. Many findings are already ready to be implemented clinically, but we hope that we can demonstrate further opportunities through our Future Health as well. Just to spend a couple of minutes on what we've also done to ensure that our cohort is as representative as possible. Most previous cohorts, it's been difficult to get large numbers of people from underrepresented groups for various reasons. So we have deliberately set up our cohort to try to ensure we have good representation from parts of the country that haven't been represented before, younger age groups, 18 to 40-year-olds particularly, better ethnic diversity, and certainly better socioeconomic diversity, given they have by far the worst outcomes. In terms of genomics, of course, this will be familiar to all of you here. Most of the data we have is from European ancestry populations. And of course, our Future Health will also be predominantly our European ancestry populations. But if we have enough people, so for example, if we can get 10% of the cohort from non-European ancestry populations, that's 500,000 people, which is an order of magnitude larger than anything we've had previously in the UK. So how have we done? So we launched recruitment in October 2022. This is slightly out of date, so this is maybe a couple of weeks ago now. I don't check it as often as I used to. I used to check it every day, but I've started to go to every week now. As the numbers have increased. I think we've got we're very fortunate, the British public, and many of you would have received invitations to join as well. Really an unprecedented response from people across the country in joining. So nearly 2.5 million people have already consented to join. And as of today, actually, 1.5 million people have now given a blood sample. So it is the largest cohort of its type with biobank bloods in the world. So that hopefully means we can maintain and build, on the work of Biobank Genomics England, and now with our Future Health to maintain our leadership in this field globally. One of the ways we've been able to do this is by having a lot more appointment venues, so over 400 appointment venues across the UK using a combination of pharmacy, mobile clinics, and shopping centres. Collaboration with the pharmacy is very important, not just for recruitment, but we can use it for ongoing clinical trials, or for example, now that most pharmacists can prescribe, there'll be opportunity for pharmacy to be much more a part of clinical services, including prevention in particular. So this just shows, going back to the point about geography, if you look at most cohort studies historically, they've been centered around universities for obvious reasons, and some parts of the country. So we took our clinics all over the country to rural areas, coastal areas, where often actually they have the worst outcomes. So places like Blackpool, northeast and northwest, East Anglia, parts of the southwest, really South Wales, Central Belt, Scotland, etc. Huge inequalities, 20-year differences in healthy life expectancy, 10-year differences in life expectancy. So for the first time, we now have a cohort that covers the whole of at least England so far, and over the coming year, increasingly Scotland, Wales, and Northern Ireland. We'll be opening our first clinics in Northern Ireland in a few months. So this is on the first 2.1 million consented participants. So in terms of diversity, we've done fairly well in terms of gender. Again, all cohorts of this type tend to have more women than men. In terms of age, again, we've got about 500,000 people aged 18 to 40, so a very large cohort, by far the largest cohort of that size in the world now. In terms of ethnicity, about 10% of the cohort is non-white ethnicity, and about 18% is non-British white ethnicity, so not very far away from census representation, although there are some groups that are much less likely to take part, so particularly Pakistani and Bangladeshi, and to an extent Black African and Black Caribbean. That's mainly driven by age. They're much younger populations. And then in terms of deprivation, we have about 250,000 people from the most deprived quintile, which is about, again, 10 times bigger than previous studies. This summarizes some of those numbers across ethnicity, deprivation, age, and gender. And we will continue to focus on this. So as we move towards our 5 million target, so we'll aim to have 500,000 people from ethnic minorities, 500,000 from the most deprived quintile, and 1 million from the age group 18 to 40. And that's particularly important, as I said, for prevention and mental health disorders, which, in fact, there'll be some data coming out from our cohort next week, just showing just how prevalent anxiety and depression have become, and particularly in younger age groups, much, much higher than in older age groups. And that's a big change over the last few years. As I said at the beginning, we have a lot of data now available. So in parallel to spending a lot of our time, effort, and resources on recruitment, we've also tried to make data available as quickly as possible. So as of today, if you go to our website and apply through our independent access board, you can access this data. So nearly 1.8 million people with questionnaire data, 1.5 million with NHS-linked secondary care data, 1.3 million with clinic measurements data, and 650,000 genotypes. And this will increase in the years to come. So as of, you've seen where we are now, by this time next year, we should have 1.5 million people with all of those data types, including imputed data on the genotype array. So really a huge, I hope, opportunity for all types of research, including pharmacogenomic research and risk-stratified medicine. And this will really help us to establish the evidence base, not just in more common diseases, but as is shown here, one of the limitations of previous cohorts has been that they were not large enough to look at rarer diseases. I mean, of course, Genomics England was established particularly to look at rare diseases in childhood, but many rare diseases also occur in adulthood. And this list, it's not exhaustive, but it shows some of those rarer conditions. And in fact, something like pancreatic cancer, although it's not a very common cancer, because it's so fatal, it's the 10th leading cause of mortality. Other diseases there which, Parkinson's disease, multiple sclerosis, motor neuron disease, etc. There really haven't been any cohorts globally that have been able to understand the risk factors for these because the numbers have just been too small. So we can really take a lead in understanding. There's no other cohort really big enough globally to do that. So just to summarize on this slide, some of the opportunities to use the resource for researchers. I won't go through all of them, and I suspect the slides will be shared later anyway. I just mentioned maybe at the end the opportunity. You can't really give a talk anymore without talking about AI and machine learning, but we have got a very large data set, and using traditional methods will obviously limit what we can do with that data. So AI and machine learning will be very important, particularly when we combine the data set coming in from primary care data, whole genome sequencing, proteomics, etc. And this will be, and hopefully in the future, imaging as well. These really are very large, unprecedentedly large data sets that will require new approaches and new methods to maximize the benefit from them. So this is what we're trying to achieve. Our kind of central box there is on our vision to enable everyone to live longer, healthier lives. And we look forward to working with all of you in order to achieve that. So thank you.
For that, Raghib. So we'll let the panel sit down, and then it's your chance to ask them any questions you have in terms of what they presented, but also other aspects as well. So maybe I can start off, and if I was to ask you what is the biggest limitation you have, I'm sure you're going to say it's funding. So we'll leave that off the cards. But maybe I can go to you first of all, Martin. What is the biggest limitation that UK Biobank has in terms of progressing further, where you want to get to? What is stopping you?
Yeah, it's a great question. I think the diversity of our cohort is maybe was mentioned, I think, when in the last talk, and that is potentially a limitation of Biobank.
I don't know whether that's working. So.
Yeah, sorry. So the cohort was representative of the UK population at the time of recruitment in terms of ethnic diversity and so on. It was slightly better off and generally healthier than the background population, but it is a limitation of Biobank. So I think it's fabulous to see Our Future Health, making big efforts to make their cohort as representative of the UK population as it is today. What Biobank is thinking about is a repeat assessment of our participants, but also to include offspring, and that'll be a way of potentially increasing the age diversity, and also possibly partners as well. So there's various ways that Biobank may evolve in the future to tackle this diversity issue, but it's probably, that doesn't mean to say that the results that are generated from Biobank are necessarily biased, because there's enough variation across the cohort to provide meaningful data. And so relationships that have been observed in Biobank have generally been replicated in other cohorts as well that are potentially more representative of the background population. So I think that's maybe just one aspect to focus on as a limitation for Biobank.
Okay, Rich, same question to you.
I think it's a great question. I guess when we look at what we see as our goal, which is to accelerate the impact that genomics can have alongside other advancing areas to improve health outcomes and to make the UK like the place to sort of discover, prove, and roll out these technologies and innovations. I think one just has to accept that the reason that we have that opportunity to retain leadership in the UK is because these are knotty problems. It's not just, oh, great, we have a great idea that there's this opportunity. Actually, the reason that we have the opportunity to continue to lead is because these are challenging things to play out end to end. And you take any one example, the diagnostic use of whole genome sequencing in a given setting. Often, for example, in our 100,000 genomes cancer program, it's not to undercook how amazing the advances were in whole genome sequencing, but often for the companies to generate these new technologies, actually the hardest thing was sometimes having a freezer on the right corridor that the surgery was happening in. And that's just one example of many. And it's why working as a whole ecosystem, you have to line these things up. And one of the great things I think we have here is the fact that you look at us three on this stage, but also many of you in the audience. This is a multi-member team effort. And you take the example of thinking about how lots of the opportunities that we've all talked about here might play out in terms of genomics or other sort of omic stratification for trials or genomics-enabled therapies. The current approaches to running trials, they just don't work. And I think June, for example, sat in the audience, has been really, in her role at MHRA, has been really progressive on thinking about how the UK can be on the front foot there and not pretend that we have all of the answers, not pretend that there's a perfect answer, and work through those. And that, I think we're really well placed to work through them, but it's not just that you click your fingers and it's done.
Raghib.
I think if you'd asked me any point over the last 3 years, I would have said recruitment of participants, but we seem to have made good progress on that. So I think the biggest challenge now is going to be implementing what we learn in our future health across the NHS. So the NHS is not set up for preventative healthcare. It's obviously, rightly so, it treats people who are sick first, but we need to move beyond that to have a sustainable healthcare system, going back to what I mentioned at the beginning. The current system is neither sustainable, it's not good for patients, and it's not good for the country either in terms of the economic cost as well. But it's not going to be straightforward to do that because of partly limited resources, partly because of priorities. We're looking forward to the NHS 10-year plan coming out hopefully next month. From what we know, there will be a greater focus on prevention, but there have been many plans in the past that have been focused on prevention, so it really will be implementation. But I think we do have an opportunity to lead in these new approaches globally. If, as I said, our future health will hopefully establish the evidence base for these new approaches, working with NHS, and then it's for the NHS to implement them for the whole population. But if we can do that, then that will be a model for the whole world.
Okay, thank you. Any questions from colleagues? Please. There's some roving microphones down here. Please introduce yourself when you...
Hi, I'm Laura Blackburn from the PhD Foundation. So I've got a couple of questions, if I may. The first one is about data and long-term storage of data. So it's something that has been a problem in the past, where, for example, you've had these amazing research cohorts being collected, and then the research project funding finished and the database died. Now, you all have, access to these incredible resources and data sets. How do you think we should think about the long-term storage and use of data? Because it is something that as a health system and also as a country that we are thinking about and getting to grips with. I think the Sudlow Review alluded to this as much, but how do you think that we collectively should think about this? And how should we treat data, and how should we make sure that it is maintained as a long-term resource?
Who wants to take it first? Well, you start with, since they've been there for longer.
Yeah, it's a good question. I mean, Biobank takes these questions very, very seriously. We have very robust data storage. Our security of our data is of paramount importance. We've created a research analysis platform recently to enable researchers to access our data on a server, which is secure, so de-identified. We've done that partly because of the physical size of our database. We're sort of getting into sort of 30 petabytes of data now with Biobank, which is just massive. So it was partly because of a sheer necessity to hold the data in a single place rather than to allow people to download the data. But it was also to protect it and to make sure that to reduce the chance of it being used inappropriately. And particularly when we get access to primary care data, we need to have very stringent safeguards in place to make sure that the data isn't mishandled by anybody. As far as the funding of the resource and the storage is concerned, this is part of our long-term plan. As you know, we get funding from various sources, the government, some charities, philanthropic funding, some industry funding. But it's top of our priority list to make sure that that aspect of our work is maintained, because obviously the data is absolutely precious to us. So I'm not quite sure how much more detail I can give, but it's a massive priority for us.
I mean, I think it's a great question, and I think it is one where our three programs, the long-term investment and the continued investment in them is something which is unusual internationally, and so it's an example where obviously data storage is only one component of it, but that longevity piece, which I think the government gets, the NHS gets, I think that's thing one. I think the second bit is, and a really key piece for us in terms of how we think about data. I think firstly, it's our participants' data, it's not our data. And so thinking about how they want it stored, how they, what their expectations are, what they've signed up for, what their emerging expectations are and how it should be used is central. The second thing is that just holding it is one thing, and making it useful for the purposes that our participants expect is different and really complex, and goes beyond actually broadly. The cost of data storage is definitely something that we need to continue considering really carefully, and as you accumulate more data it does cost more, but actually storage costs alone aren't the biggest challenge. Actually generating data, generating and having a new sample taken, there's both cost and opportunity cost to that. So actually the model of storing and reusing data is one that we think is the right one to pursue and explore. But actually getting the digital infrastructure so that it can be used for different purposes. A really simple one is for comparable analysis between different data sets, holding the data in a comparable format across the big cohorts is really important. We've done a lot of work with across the big cohorts saying, on whole genome data, we'll all hold it on this version of the Dragon pipeline, and that makes a real difference. For clinical use, actually, so often our role is to turn complex, often quite big genomic data into simple packages, sort of those small and simple packages of data that the end user systems can use to then make, and it's back to the point I made when I was speaking about actually making, not kidding ourselves that every system or every person is going to become an expert in genomics. It's to make the data presentable in a simple way. And I think, both Raghib and I have touched on this, recognizing the importance of research and clinical care being linked, and thinking how we make sure we look to the future so that the data can be used for both purposes, where people and drive that efficiency where people consent to that.
If you want to add?
Just briefly, I mean, I think the government certainly recognises the importance, particularly of our cohorts, but also the other cohorts, particularly those that include genomic data as kind of critical national infrastructure, and so they won't allow the resources to die. But the other new initiative, which many of you would have seen, was announced recently, is the Health Data Research Service, which I think will bring together in a much more coordinated way the various data sets, including ours. So that will be an important development over the coming years, this collaboration between government and Wellcome Trust. So yeah, I think that will help.
Was there another question you had?
Just a very quick one is, what are the plans, if any, for linkages between your three data sets?
For integration?
Linkages between
Linkage, I mean, we'd be very keen to...
all speaking about
we'd be very...
sources of data.
Very keen to, in principle, very keen in practice. Some of the challenges in terms of the various consents and the way the data is being collected and stored make it not as easy to implement, but in principle we're very happy to do that.
Yeah, I think there is a move in general to make sure that various data sets are held in similar formats. So for example, environmental exposure data, pollution data, making it available in the same format to researchers around the world, so that different biobanks make it available in the same format, and it will just allow large-scale research to be done, that links data from individual resources like biobank.
Yeah. Okay, great. Let's go to the next one. June, please.
Well, thank you, and I just think this is astonishing, incredibly impressive progress over all three, and I think we should, really log congratulations. This is incredible, and as we've heard, what a position it puts the UK in to drive forward the frontiers of public health, and in so many aspects unique. Are there, building on Laura's question, more opportunities for synergy? Not necessarily linkage, but more synergistic approaches, or even if there are areas of duplication, given the financial imperatives, have you thought about that?
Yeah. Great.
I'm happy to start on that. So just on one example, so I touched on it before, one example on genomic data formats is that one about actually ourselves, and then in the US, the All of Us program are all harmonized on a single approach to holding genomic data. And it's Dragon 3.7.8 format. We've also all spend a lot of time talking about how we can harmonize, how we can share learning on the platforms we use. With biobank, because we have some shared participants with cancer. One of the things that we've worked on with our participant panel is, are they happy for their data to be made available in biobank if their participants are both? And the answer is yes, and we are, and that's something that we're doing. We also see big synergies in terms of how our different skills can answer those quite knotty questions. They're not one organisation questions, a lot of this, and so as we look to think what evidence we need on how you might implement some of these things in clinical care, that's very much something with across our three cohorts, but also with many other people in the ecosystem. This is a team effort and working out how you best develop that evidence.
Yeah. I mean, I think there are conversations going on, between our groups. So I was in conversation yesterday with colleagues at Genomics England over digital histology, and there's work gone on in Genomics England that we'll learn from, and we'd like to make sure that there is the opportunity to present data to researchers in similar formats, so that large-scale research can be done more effectively.
These are not competing resources, they should be complementary, and we should avoid duplication, which is a danger sometimes. The overlap in terms of the three cohorts, I suspect, is relatively small because of the ways that we've recruited differently. But there are other cohorts, not represented here. There are many, I think probably I can't remember the number, but dozens of cohorts across the country with millions of people in them. And I think there should be an attempt to, as far as possible, link people who are in parts of multiple cohorts, because there's information that different cohorts have that are not in the other cohorts. And if we could have a unified kind of record of that, I think that'd be extremely powerful. And some of these cohorts go back decades. So that, again, that would help to get that additional information. And some are focused more on education, for example, or other social determinants. So there's a big opportunity there. We haven't done it yet, but I think it's a good challenge for us to do that.
There are bodies being established to bring those cohorts together to make people aware of the resources that are available across these cohorts. There's huge numbers of cohorts actually across the UK, and some of them are ideal for particular types of research, but it's very difficult for a researcher to identify which cohort to use for their particular research questions. So there are efforts underway at the moment to bring the metadata from those cohorts together so that researchers will be able to decide exactly where to focus their research effort.
Okay, thank you. So, please.
Hi, I'm Sarah White. I'm a GP. I work with Bupa Clinics at the moment. And you mentioned we're launching a product for polygenic risk scoring, but we're also doing some whole genome sequencing. We're also about to look at some pharmacogenomic interactions. Being GPs, our knowledge is growing enormously, but it's still a little bit shaken, and the science sounds absolutely fascinating. We've been given a bit of variable advice, so we understand if someone's having genomic testing, they shouldn't have had a bone marrow transplant because that changes things. But we've been given varying opinions on if you're looking at pharmacogenomic data, if you've had a liver or kidney transplant, that could be different than so someone who you're entirely looking at their own genes. Does that have any effect at all?
Might be a question for Munir.
Yeah, that's definitely above my pay grade.
Yeah. I certainly don't think biobanks will be able to cover something like that, and there are some complexities in terms of depending on what kind of transplant you have and so on, but we can talk offline if you want about that.
Okay. The other question is obviously this is all very much a developing area. As things stand, although obviously your genes stay exactly the same, but the interpretation of pharmacogenomic interactions, will that vary between different labs, or are they all sort of talking to each other and there's certain standards and they're all saying the same thing?
Again, like Munir, why don't I start and then Munir and others can correct me. But I think pharmacogenomics it's a really impressive field in terms of how established international standards have become, and CPIC as a resource is really useful. And I think, as with all of these areas of genomics, we need to recognise that we're still early in the learning curve, and I think there's been really good work done to date on much better actually than in a number of other areas at a similar stage in terms of harmonisation. But there's a lot left to learn. So we know about certain variations, and great research has been done on, but one of the key things, and the thinking behind our resources and much other work, is there's a lot left to learn, and I think acknowledging that, recognizing that's in all sorts of dimensions. Actually, Munir and I were talking about this before, like in the context of polypharmacy. And you've just given us another really knotty question about transplants, but also in different communities, and are there also more complex, are there rarer variations in that also have an impact that we don't yet understand. So there's a lot for us still to navigate that we need to bake into how we run these programs, so we keep learning. A bit like, that's a big lesson for us in the rare disease setting. It's great to do stuff today, but actually the power of what you can learn and do for tomorrow is even greater.
So as part of the CERC that I mentioned earlier, we're working on a panel. What is the current panel of pharmacogenes that we should be able to implement into the NHS today? But clearly that's going to be dynamic. As more advances are made, the panel will expand.
Absolutely. I was just thinking, are there differences at the moment between the current genes you can test for and how different labs might interpret that in their result reporting, or is there general harmony at the moment?
So I didn't quite hear all of that, sorry.
Sorry. I know obviously we're going to find more, not us, but the science will find more interactions and more drugs that there's an interaction with. but on the things that are known at the moment, is it likely that different genetic labs, as when they report back to you, might have a different interpretation of a certain finding?
No, so we're working on the standardization of that.
Right,
So we're
okay.
developing guidelines as part of that,
Yeah.
and also working the industry group to make sure that there's kind of standardization, harmonization going on, because that will lead to confusion if there's differences between different labs and different reporting structures.
Okay, thank you. It's just all just a developing thing. It's just amazing, isn't it? Absolutely fascinating. Thank you.
Okay, we've got a few more minutes. Any more questions? Chris, over here. Oh, is there somebody over there? Yeah, please, if you were there before, and then Chris. There's 2 more questions, please. You first, and then Chris there.
Hi there, I'm Sudanas. I'm an undergraduate student at University of Nottingham. So my question is actually about AI, so which is like machine learning. I see that with the developments of the machine learning, like your companies included AI as an analysis tool. So how does your companies address potential biases introduced by AI-driven image analysis, particularly when training models on imaging sub-cohorts that may not reflect the full population diversity? Because like I know UK Biobank does lots of things with the AI. So that was my question.
Who wants to take that?
So you're talking about the image, using image data, is that right? So you're thinking about algorithms, machine learning algorithms that identify phenotypes from images like MR, magnetic resonance imaging. Yeah. The position that biobank takes on this is that we provide the data, so we provide the raw data, the images from the MRI scans, and then it's for the researchers to apply their models, whatever machine learning models they would prefer to use. We don't dictate any particular methodologies. It's for researchers to use the data in the ways that they would like, and we actually embrace that sort of approach because we'd like a range of methods to be applied to the data that we present to researchers, and if there are differences in methods, different machine learning models, for example, and they generate somewhat different results, then those data will go back into the research domain, and then people will ask questions about why there are those differences, and the science actually moves forward because of those differences. So our role is really to provide the data, and then it's for the research community to find the most appropriate methods, including machine learning methods, to analyze that. We will be making machine learning tools available on our rep so that people will have a range of things that they can apply, but there may be some techniques that we wouldn't be able to support, and we'll find ways of allowing people to get access to the data and use those techniques as appropriate.
Thank you.
Okay, last question here to Chris Chamberlain.
Thank you. Chris Chamberlain from AstraZeneca. It's absolutely wonderful to see all 3 of you on the one panel representing the UK's significant leadership in this space. It's already apparent that beyond research, you're going to have a transformational impact on diagnostic practice, but this is going to be a significant challenge for the NHS pathology services and the diagnostic industry as well. And they probably need to get started already, given how quickly things are changing. What do you think we should be doing to prepare in this space? There were the molecular pathology nodes from the MRC a few years ago, but most of those have gone. What do we need to be doing now to use what you're going to deliver?
Okay, maybe Richard, might be better you start off with that one.
Yeah, I'm happy to start, thinking very much about genomics as an element, but recognizing your question is broader than that. It's one of the things where, as we work through the stages we're at so far, the expansion and organization of diagnostic use of genomics, that's one of the things we're working really closely with the NHS who have this 7-hub approach there, and we're working to continue to support them where they're wanting to move to a system where you keep certain amounts of the data analytics centralized, which we support, but recognizing that other elements don't necessarily impact. Often samples being closer to patients is better. So I think that sort of join up, and there's not a one size fits all for different, and I wouldn't pretend to be an expert on what the right answer for different elements of that pathway are, but I think that making sure there's a real link up between what the decision, recognition that some things do need to be done in a harmonized, sometimes centralized way, and deciding which bits don't. And that's something that I think we're privileged to have really good joined up thinking on in the genomics element. There's lots else that one needs to work through. Actually, almost all of the modalities that we've all talked about, there's a similar question and not necessarily always the same answer.
Do you want to add?
Maybe just, I think we pretty much have time to end on a positive note. I mean, I also talked about the challenges of the NHS being able to implement this, but I think for the first time, although there have been previous plans in terms of support from government, from the NHS, from the regulator, and the resources that we have, I'm much more confident now that that the NHS will take on this challenge and devote appropriate resources over the coming 5 years at least, during the course of this government, hopefully beyond that, to make that transformation really of the health service possible. So I think we should be optimistic, but recognizing there's still a lot of work for us to do.
Great, thank you very much. Okay, well, let's thank our panelists and the speakers for this morning. As June said, that these are fantastic resources. Does make the UK world leading in this area. So thank you very much for all the work that you're doing in this area, and hopefully we can support you in order to progress this further. Thank you. Okay, so coffee time. I think we've got about 20 minutes, and coffee's upstairs. Thank you.
So, ladies and gentlemen, welcome back to a session which is focused on emerging research leaders. So I'm Ros Smyth. I'm an academic paediatrician at UCL, and I'm Vice President Clinical of the Academy of Medical Sciences. And I'm really delighted that this session is in partnership with the Academy. So for those of you who don't know, the Academy funds a really fantastic scheme called the Starter Grants for clinical lecturers, although the eligibility has extended a little bit beyond lecturers to other people in that training grade at similar level. It was originally funded predominantly by the Wellcome Trust. It's now funded by Wellcome, MRC, but a whole host of other funders, including the Association of Physicians. And I've been a panel member, and then in a later incarnation was the panel chair, and Munir was one of our panel members. And it's a hugely impactful scheme that doesn't give a massive amount of money, but it gives the opportunity for people's, often their first grant, that can really help them to get important preliminary data that can then start them on a track to larger applications for external funding. And we have 3 such individuals speaking to us this morning. So I'm delighted to welcome the first speaker, who's Dr. Pamela Bowman. She's from Exeter, and she's going to talk about sulphonylurea therapy in iDEND, is that, have I said that correctly, syndrome, so from a great centre which has a big interest in this area. So let's welcome Pamela.
Thank you very much. I'm absolutely delighted to have been invited to speak today about a specific area of rare disease and precision medicine that I've researched over the years. And this is to do with sulphonylurea treatment in iDEND syndrome, so developmental delay epilepsy and neonatal diabetes, and how a genetic diagnosis really enables precision treatment for both the diabetes and the neurodevelopmental phenotypes in this condition. I'm going to start with a case. This is a little boy who was diagnosed with diabetic ketoacidosis at the age of only 5 weeks old. So presented extremely unwell with severe insulin deficiency. and he was diagnosed with neonatal diabetes. So he was initially put onto insulin treatment and quite poor control, incredibly difficult to manage in a tiny baby, these big fluctuations in glucose, lots of hypoglycemia, sometimes resulting in seizures, and parents often spoke about the real challenges of managing this on insulin. Now, neonatal diabetes is rare. It occurs in about 1 in 100,000 live births, and it can be divided into transient forms where the diabetes occurs in the first 6 months of life. It then remits and then typically relapses later on in childhood. And then permanent forms where the diabetes occurs and persists throughout life, and syndromic forms where there can be a constellation of additional multi-system features. And for the majority of this talk, I'm going to focus on permanent neonatal diabetes. And the main thing to remember about these conditions is that they are usually monogenic. So there's usually a single gene cause for this type of diabetes, and it's incredibly important to test babies, and in fact anybody who's presented with diabetes under the age of 6 months. And the National Genomic Test Directory actually allows us to test under the age of 9 months, because making a genetic diagnosis can be absolutely crucial for the clinical management, and that's what I'm going to talk a bit about now. So the commonest cause of neonatal diabetes is heterozygous activating variants in the KTP channel, the pancreatic potassium channel. Now, this is the channel that sits in the pancreatic beta cell. It links blood glucose to insulin secretion. And these variants essentially occurring in either the KR6.2 pore-forming subunit, now that's coded by KCNJ11, or the regulatory subunit, which is SUR1. These variants essentially cause the channel to be stuck in the open state, so activated, not responding to ATP. Metabolically generated ATP. And therefore, the beta cell remains hyperpolarized even in the presence of rising glucose. And insulin remains locked inside the beta cell. So patients present as insulin deficient. It's just that their insulin is locked inside their beta cells. So this SUR1 subunit is actually the same subunit, as many will be aware, that is bound by sulphonylurea medicines that are classically used to treat type 2 diabetes. So it was really groundbreaking when this genetic discovery was made, because it was then shown that this type of diabetes could be treated with sulphonylureas. And in fact, these drugs overcome the genetic defect, closing the channel independently of ATP. Children secrete their own insulin. They come off of injections. They really don't need to test their glucose very much at all, because their glycemic control is very good. And they all get improvements in glycemic control, essentially. Both the KCNJ11 and ABCC8 variants. And really, this was groundbreaking, and it completely changed the lives of individuals and families affected by this condition. Part of my PhD, I did some long-term follow-up studies over 10 years looking at the outcomes of these individuals. And it was really reassuring to see that actually over 10 years, this was a really successful treatment that was maintained long-term. So for both genes, 90% of people were staying on sulphonylurea alone long-term, typically glibenclamide, with really good glycemic control maintained without the need for additional therapies. In the small minority who didn't maintain that control long-term, there were other explanations. So, for example, puberty with increased insulin resistance, children growing and the dose not being increased with growth, and reduced compliance were some of the things that came out. But in the majority of cases, we were seeing a real success story. And what was also very reassuring was that this was a safe treatment. So no serious adverse effects reported over the duration of the follow-up, and only some mild GI side effects, but nobody had to stop treatment. We also didn't see any severe hypoglycemia in over 800 patient years of follow-up, and that was defined as kind of having seizures or losing consciousness. And this is in contrast to what you might find in type 1 diabetes, where severe hypoglycemia is more common. So this appears very safe, even with the high doses. Now, the doses we use are of the order of 2 to 10 times the magnitude of the doses in type 2 diabetes. But despite that, there's no severe hypos. People did describe some mild hypoglycemia, and that appeared to be related to food, rather paradoxically. But people would say, and patients came to us and said, look, I've gone onto a diet that's low in carbs, trying to lose weight, and I've started getting more hypos. So we thought, well, is it something about the regulation of the channel in this context that's different? Is the physiology different, which is making this mechanism of insulin secretion imperfect in the absence of these potassium channel pathways? So the hypothesis here is that actually the sulphonylurea is permissive in its action on the potassium channel in not directly stimulating it, hence you don't get the severe hypos, but actually allowing the channel to respond to amplifying pathways of insulin secretion. And that's supported by the fact that if you give these patients oral glucose, they secrete insulin when they're sulphonylurea treated, but if you give it IV, they don't. So it's something about the presence of food in the gut as opposed to the glucose level in the blood that's regulating the insulin secretion. And we wanted to test this out in a bit more detail. So I brought some patients and some controls who didn't have diabetes into our clinical research facility, and we gave them a high carbohydrate meal and a high protein meal on 2 separate days. The red lines are the insulin, and the blue lines are the glucose. We'll just focus on those at the moment. So these are the non-diabetic controls. You can see it's rise in glucose with the carb, and then a nice first phase insulin secretion that lowers the glucose and then is then switched off. And then here, with the protein meal, the insulin rises is less and the glucose stays fairly flat. When you look at the KCNJ11 cases that are treated with SU, what you see, if we just look at the insulin first, so that's pretty much the same between the 2 different meals, but the glucose is actually going in the opposite direction. So with carbohydrate, it's rising, with protein, it's actually falling over time, but the insulin is being made in response to the food and not in response to the glucose level. So that was supportive of this hypothesis that actually this was being driven by a different pathway and the physiology is unique. We did actually look also at GLP-1 and GIP levels to show that the incretin pathway was intact in these patients, and in fact I found that the levels were the same between the diabetes patients and the controls. And that's led to the potential for GLP-1 receptor agonists to be used as an adjunctive treatment in the minority of patients who don't get good long-term control on sulphonylureas. So knowing that this pathway is intact has enabled us to bring that in as a potential therapeutic option for people who need adjunctive therapy, so avoiding the needs for reintroduction of insulin. So dietary advice, we tell people to include carbohydrates to avoid that stimulation of insulin secretion without any carbs to sort of balance that, so have mixed meals. Avoid low carbohydrate diets and missed meals, and that the incretin pathway is actually very much intact and could be used as a therapeutic strategy. So the diabetes, really well managed. As I say, people don't really need to test their glucose very much. They have liberal diets. That's really, really good news. However, it's not the only problem. So I'm going to move on to talk about the neurological features in this sort of, in this diabetes, where what we see is that there's a spectrum of neurological features that range from very subtle neuropsychological impairments when you test for them that aren't clinically obvious, all the way to the severe DEND syndrome, so this diabetes, epilepsy, and developmental delay. And what we see is that there is a genotype-phenotype relationship. So there are some variants that are sort of almost always associated with a severe neurological phenotype. And there are others where you really just see diabetes, but you have these subtle impairments if you test for them. And that fits with the expression of KTP channels, because not only are they expressed in the pancreas, but they're also expressed in the brain, muscle, and nerve. Now, what was really interesting was that when people started switching from insulin to glibenclamide, not only did the diabetes get better, but there was also some improvement in the neurological features. And that was reported in some early case reports, and also supported by some neuroimaging studies that showed improved cerebellar perfusion and perfusion of other brain areas on transfer from insulin to SU. And one prospective study looked at this and found that the improvement was bigger in younger patients and was more around the motor abnormalities, attention deficits, and hypotonia. And for our case that I presented in the first slide, he had this V59M variant. This is one of the variants that is usually associated with quite a significant neurological phenotype. And he had iDEND syndrome. He had epilepsy, he had no speech at the age of 4, and he communicated using picture cards. But when he transferred to glibenclamide, school noticed a fairly rapid improvement in concentration. And in fact, he came out of school and spoke to his mum for the first time, said, "Hello, mummy." And that was a really just an amazing moment. It's amazing to hear his mum talk about that. But what we know is that despite having an initial improvement, which can be quite marked, people are left with residual neurological deficits. And part of our long-term follow-up study, we looked at the types of things that people were left with, even on sulphonylurea treatment. So there's intellectual disability, ADHD, are quite common, and then other things like the epilepsy, sleep problems, muscle weakness, anxiety, and in KCNJ11 patients in particular, they appear to have higher rates of autism than the general population. So we looked at this in a bit more detail with a subset of children in whom we did development and well-being assessment, which is a semi-structured questionnaire that can be used to diagnose neurodevelopmental and psychiatric disorders. And we found that within 10 children, 6 people had more than one neurodevelopmental disorder, and there were 14 disorders in total, but only 2 people had a diagnosis clinically, and that was autism. But these were ranging from autism, ADHD, anxiety, and behavioural disorders, and they had a really high impact, as evidenced by their Z scores on the Strengths and Difficulties questionnaire, scores for impact from parents and teachers. So it was suggested that perhaps this could be a feature of the metabolic disturbance and hyperglycemia from a very young age. You know, was that the main reason why these people were having the long-term CNS problems? So we tested this out by taking some adults on sulphonylureas with KCNJ11 variants, and we took them and compared them with cognitive testing to a group of people with INS variants that cause neonatal diabetes, but you wouldn't expect them to have a significant neurological phenotype based on the expression of the INS gene. So we were controlling in a way for the hyperglycemia from a very young age. And what we found was that the KCNJ11 patients essentially globally performed worse on these tests in comparison to this other form of neonatal diabetes. So it's quite convincing that this is a channel-specific problem because of the potassium channel being present in the brain. So why is the response incomplete in comparison to this really quite amazing beta cell response? There's probably a few reasons for that. 2 of the main ones, we think one is that we may not be achieving very good therapeutic levels of glibenclamide in the CSF. There's a rodent model that shows if you inject glibenclamide into the ventricles in a rat's brain, you rapidly find it in the circulating blood at higher levels than in the CSF, so there's an active transport mechanism. We don't know if that's the same in humans, but assuming that it is, we recommend higher doses in people with significant neurological features to try to mitigate that and try to get some decent therapeutic levels. The other thing that is the age of initiation of treatment. Now, are we starting treatment too late and missing a therapeutic window where there's more neuroplasticity and more ability to change outcome if you hit the channels in the brain at that young age? And that's the focus of a lot of the work that I'm doing at the moment. One study is supported by an Academy of Medical Sciences clinical lecturer starter grant. So just thinking about this early treatment concept. So these are 2 children both with V59M variant, so the genetics is the same. This is a girl who drew a man at the age of 9, the boy who drew a man at the age of 16, and this child has gone to a mainstream school, is now learning to drive and is on her way to independent living, and this person went to a special school and lives in supported accommodation as an adult with intellectual disability. Now, the main difference between these 2 is that this person was treated from the age of 6 weeks old with glibenclamide and also was serendipitously exposed to the drug in utero because her mum had gestational diabetes. This person started sulphonylurea treatment from the age of 4 years, and so we may have missed as I say, that therapeutic window where there's more neuroplasticity. Emphasises the need for early genetic diagnosis and early treatment. So this is some preliminary data from this study that's funded by my grant, which is ongoing, and this is taking people with the same variant, so V59M, to try to reduce the variability we see between the different variants. It's an international study. This is very early data, but what it's showing here on the left is the number of ICD-10 diagnoses. Again, these are mostly neurodevelopmental diagnoses like autism and ADHD, but that's higher if you treat people over the age of a year, and also the non-verbal IQ is lower. So this is a difference of about 1.7 standard deviations between people who started late versus early, which really is quite remarkable and takes people from a moderate to severe intellectual disability to a mild to moderate intellectual disability. I think this is really exciting. I'm hoping that it will hold out as we recruit more patients around the world and get a bigger cohort for this study. Lastly, just want to talk about another project that's ongoing that is taking treatment to the very earliest part of development. So this is a lady who came to us with neonatal diabetes, and she was pregnant. And just to say that the implications in terms of her pregnancy management really depend on whether the fetus has inherited the variant or not. So there can be really positive impacts, and because glibenclamide crosses the placenta, if it's inherited the variant from mum, we can potentially get glibenclamide into the fetus while it's in utero, normalizing insulin secretion, fetal growth, and potentially benefiting neurodevelopment, and also for that postnatal monitoring for hyperglycemia and avoidance of DKA. And if the fetus is unaffected, we want to avoid treating mum with glibenclamide in the 3rd trimester so that we don't promote insulin secretion in the fetus, overgrowth, macrosomia, and complications. So there's a new technology in Exeter, cell-free fetal DNA. We can do this non-invasively, both from a paternal or maternal sample, to see if the fetus has inherited the variant, and this will inform pregnancy management, and this is a study that we are currently recruiting to. So this is really exciting. I think I'm short of time, so I'm not going to labour the points on the last slide. It's just a summary to say we've got pancreatic and CNS phenotypes in this condition. Sulphonylureas have impacted on both really profoundly. It's impacted on our clinical advice for the management of the condition, and thank you very much to everyone who's made this research possible, most of all the patients and families who are just amazing, and I'll leave you with a few links for further information and some ongoing studies on our multidisciplinary clinics. So thank you very much for listening.
Thank you. So I think we've got time for maybe 1 or 2 quick questions, if I can see. Could I just start off by saying, is the pancreas the most sensitive organ? What I mean by that is, do all of these patients have diabetes and some of them then have neurological problems, or are there people with these conditions hiding in ADHD and other developmental disorders?
It's a very good question. Certainly for the variants causing permanent neonatal diabetes, the diabetes is very penetrant and presents in the first 6 months of life. We'd expect them to be presenting with diabetes. There is another form, the transient form of neonatal diabetes. The variants can be a bit more unusual in terms of within a family, someone could present with neonatal diabetes, but someone could have the same variant present with an adult with diabetes, or they might not have diabetes at all. So there's more variation in the diabetes phenotype with those transient neonatal diabetes phenotypes, but the neurological phenotypes in those patients are really not particularly significant as far as we know. It's another area, though, that we want to do some research on. Yeah.
Any other quick questions? Yes. We've got a roving microphone.
The early treatment data is quite profound.
Yeah.
You're talking about children and then young adults who could be able to live independently versus...
Yeah.
So how do you see testing being implemented into clinical practice? You know, what warning signs do you look out for, say, during pregnancy? What cohorts are you hoping to reach by this? Because, you know, if you can make that difference, just lifelong, that's such a
it's huge.
Profound difference.
It's huge, yeah.
How someone can live.
Absolutely agree. And just hearing those patient stories, you hear from the families just how different it is for their quality of life. For this particular condition, 80% of these have been de novo so far, so they occur. It's not easy to predict through any. But one thing that my colleague Tim McDonald Dexter has shown is that glucose tends to be raised in these individuals at day 5 of life, even before they've presented. So actually introducing glucose even as a screening tool may be helpful. The NIPT for the ones that are inherited is going to really revolutionize care, because I really think that because we'll be making that diagnosis really early, the in utero treatment is a really exciting idea, but even just knowing that we have to be monitoring postnatally and catching them before they develop DKA and getting them on that early SU treatment for the development is crucial. So I think there's different ways of ascertainment of these cases. What you ideally don't want is for them to be presenting, really sick with diabetic ketoacidosis. So, yeah.
Okay, thank you very much.
Thank you.
I'm afraid that's all we've got time for. So our next speaker is Dr. Hannah Nieto, I hope I pronounced that correctly, who's going to talk about genomic diagnostics in thyroid cancer.
So good morning, and thank you very much for the opportunity to present today. I'm Hannah. I'm a head, neck and thyroid surgeon currently working in Manchester and going to start in Birmingham in a month, and I'm also a clinician scientist there. So I'm going to give a little bit of background first, and thyroid cancer as a whole is increasing in incidence both in the UK and worldwide. We know that a lot of this that you can see on this graph where it's kind of flicking up alarmingly, it are incidental findings, so people are having more scans and so we're picking up more and more of them. What isn't kind of reflected quite so widely is the fact that actually the mortality rate from thyroid cancer is increasing as well very slightly. So some of these are the serious cancers. Some of these are cancers that people may not have known about otherwise. So there is a challenge here about which of these diseases are important and which are indolent. So when I talk about thyroid cancer in this talk, there are lots of different types of thyroid cancer, depending on where in the follicle it is derived from. So this picture here of some thyroid cancer cells. This is a follicle with colloid in the middle. These are little parafollicular cells. These are medullary. These create medullary thyroid cancers if things go awry, and that's associated with RET mutations, which you may be aware of. I'm going to talk predominantly today about the differentiated thyroid cancers, which come from these follicular cells. Majority of these are papillary thyroid cancers, but also follicular, and together these are differentiated thyroid cancers. We also have poorly differentiated, which then can de-differentiate even further to anaplastic cancer, which is a slightly different kind of disease really by the time it gets to that point, which I will kind of touch on a little bit later. So what do we do for patients when we see them in clinic at the moment? So patients tend to present, either they've had a scan saying they've got a nodule or they have a lump in their thyroid, and they come to us with their thyroid mass having been seen by their GP. And we grade it, we take a history, we examine them, and we grade it on ultrasound. And the UK grading system is the U classification, essentially from 1 to 5 in terms of risk of malignancy. So anybody that's U3 and up gets a needle biopsy, so FNA cytology, and then they're graded again from 1 to 5 in terms of likelihood of malignancy. I'm going to come back to this, but what I'm going to talk about very first of all are these patients who have indeterminate lesions in the middle. Now these patients here have a needle biopsy that can't tell whether they've got cancer or not. That's not because the test hasn't worked. It's worked, but the cells, you cannot tell you whether it's cancer or not because you have to excise the entire nodule. So for these follicular neoplasms that you can see here, the only way of telling if it's an adenoma versus carcinoma is invasion of the capsule. So you have to take the whole capsule out at the moment, which means having a hemithyroidectomy. So in the UK, people have diagnostic surgery, and then after their diagnostic surgery, they come back to us for their final diagnosis. And they even have confirmed thyroid cancer, and maybe they've had enough treatment at that point, but quite often patients have to have completion of their treatment, which is the other half of their thyroid out, so more surgery and radioactive iodine. And then you also have a cohort of patients, the majority from that group, that have no thyroid cancer, who have surgery for benign disease, for no cancer. So there's a couple of challenges here that you can see that have been going on for quite a number of years. And one of those is how do we prevent patients who are having surgery for no cancer from that occurring? And the other one is how do we get the correct treatment tailored for patients at the beginning of treatment? And in America, and kind of maybe a little bit further afield as well, some places in Europe, they have some commercial or even university-based molecular testing that they do. And essentially, there are lots of tests, but there are two commercial ones that dominate the market, Thyroseq and Afirma. Thyroseq is a DNA somatic mutation-based test that has now added in a bit of RNA-seq as well, and it gives you a grading as to the likelihood of this being cancer or not. Now, they are really developing this as a prognostic tool as well, but the main thing that we're talking about initially here is the ability of it to rule out cancer. Afirma is a RNA-seq-based test, which has added in a couple of somatic mutations, and they too would like it to be a prognostic test, but the main thing that we want to think about here is ruling out cancer. So why don't we use these in the UK at all? Not even in private hospitals at the moment. Nobody really uses them. They cost $3,000 per aspiration, like per test, per nodule. So in the UK, it's relatively cheap to have a hemothyroidectomy. So the system we work in pays the hospital or the trust about £2,500 to have a hemothyroidectomy, whereas in the US, it costs about $10,000 to do that same surgery. So you can see why in the USA these tests make sense, whereas here there's a little bit more of an economic challenge. My own thoughts on that is as well, I don't think the data was that robust when these were rolled out in the USA compared to the amount of information you have to have in the UK to bring this in. And that has started to change because they've been there for a number of years now, but it's probably not as robust as we might like. So just taking a step back again, I just want to talk to you briefly about these 5 patients here. So these are the people who are pretty sure have malignancy when they've had their needle test. And what do we do for them? So they have some surgery depending on various clinical features, which are determined by the MDT. Now, traditionally, for thyroid cancer, patients would have a total thyroidectomy, so all their thyroid removed, and radioactive iodine. And radioactive iodine is a really nice treatment that is targeted to the NIS, so sodium iodide symporter in the thyroid cells. So it's preferentially taken up by thyroid cells, and it's got the radioactive component, so it obliterates any thyroid cells left behind. So if we've left a remnant here or any micrometastases, they're obliterated. But as we spoke about before, some of these cancers are not likely to progress or not very maybe clinically significant. And now it's relatively well established that if you've got a tumour that's less than a centimetre, as long as it's a classical papillary thyroid carcinoma on the histopathology, you can have a hemothyroidectomy. We're starting to expand this criteria, and I think in a lot of major centres in the UK, you would only get a hemothyroidectomy for a thyroid cancer that was less than 2 centimetres now. And the iron and estimable 2 trials that have come out in the last year have been radioactive iodine trials that have said for low-risk thyroid cancer, you don't need radioactive iodine. So if you don't need radioactive iodine, you don't need the rest of your thyroid removing, because we only complete the thyroidectomy in order to facilitate radioactive iodine. So they keep the rest of their thyroid, and they have that normal thyroid hormone and that normal metabolism, or relatively normal, afterwards. Now we're kind of moving on a step further to active surveillance. Now, active surveillance sometimes refers to these patients who've had a hemothyroidectomy for small tumours, but it also refers to the NICE guidelines say that if you want to, for a tumour that's less than a centimetre or the patient's very keen, you can do no surgery at all and just keep an eye on it. The difficulty we have is that even abroad, it's not very well defined what kind of surveillance we do for these patients. And then what about the patients who have really, at the other end of the extreme, so have really advanced thyroid cancer? We have a few different people, different ways this might affect people. There might be local invasion, so it's grown into local structures. It might be inoperable, or they might need a really major surgery, like a tracheal resection or a laryngopharyngectomy. Have lymph node metastasis, so the cancer spread locally. This is relatively well treated surgically with a lateral neck dissection. Distant metastases can't really be treated surgically or not very well surgically, and so this is where we have radioactive iodine. But for some patients, they start to get radioactive iodine resistance when this starts happening, and so that becomes a challenge. And then we have the anaplastic thyroid cancers that I mentioned at the beginning. This is when things have de-differentiated, and they're often huge on presentation, invade local structures, and surgery is not a good idea for these patients as a general rule. And so another challenge is for these radioactive iodine resistant patients and the non-surgical patients, what do we do? Now, thinking about a bit of genomics behind this, there's some key issues in terms of thyroid cancer progression. How do differentiated tumours progress? How do tumours which progress differ from those that don't, especially at the outset? What mechanisms can we use to target these therapeutically? And radioactive iodine refractoriness, can this be overcome? And this kind of is a nice slide that shows that we now divide these up into BRAF V600-like and RAS-like nodules or or tumours. And that's from a Cell paper back in 2014. And actually, we think one day we'll probably reclassify tumours rather than papillary and follicular based on these mutational profile. And you can see here that we think things de-differentiated down from the nice tumours that we're talking about at the top to anaplastic thyroid cancer. And this was a Sentinel paper by Lander and the Fagan group in America, which shows that you acquire mutations such as the TERT promoter mutation and TP53 as you go from well-differentiated through to anaplastic thyroid cancer. So how does this relate to the work that I've been doing? So some key findings of my PhD are kind of outlined here. I did a somatic mutation, an RNA, somatic analysis, RNA analysis, and microRNA analysis as part of my PhD, and bioinformatics, and then took this into the lab for a wet lab kind of discovery science. And the main things that we found really were the RNA expression levels that we found were more important in terms of thyroid cancer recurrence than the somatic mutations that we identified. And that we managed to say that even at the outset, a lot of these things were identifiable at the initial disease, and that kind of gives us a clue to the patient's pathway later on. And we managed to create a risk score based on these 5 markers that can categorise patients in terms of their likelihood of recurrence later down the line. Kind of a step further on for me in my NHR ACL, during which I had my starter grant, I wanted to take a look at some more translational work and look back at those patients that we were saying had those indeterminate nodules, because it seemed to me there was a clear opening for doing something molecular with that bioinformatic training I'd had. Papillary thyroid cancers are pretty well defined mutationally now, and this is fairly well understood. Unfortunately, the follicular thyroid cancers, which are the ones that are very difficult to tell on aspiration cytology, whether they're cancerous or not, are also quite difficult genetics-wise, because they're often RAS-based, the mutations, and these are present in adenomas as well as carcinomas. But, you know, those commercial companies have managed it, and so we wanted to see if using a different technology, so the Oxford nanopore MinION that you're probably aware of, a very tiny sequencer that works in a different way using nanopore, rather than the kind of traditional sequencing, would be helpful. This is particularly good for translocations because it does long read sequencing, and therefore it's particularly helpful in thyroid cancer. And the idea being to try and avoid surgery if we can. So this is the pathway that I've set out on, that we were going to get FNAC samples, get the DNA out, do some nanopore sequencing and bioinformatic analysis. We optimised this with cell culture samples and then FNAC needle wash samples, and we managed to do multiplex PCR and get the pathway all working. However, multiplex PCR is a little bit limited. You can amplify it up, but when you're looking to look at hundreds of different areas of the genome and potentially RNA as well, we wanted to do whole genome amplification and use adaptive sampling, which is a very particular use of the nanopore technology, where you can use instead of using PCR or CRISPR, you can actually use the sequencer itself to feed back while it's live sequencing to target certain areas of the genome, but it is quite tricky to do. This is my little nanopore kind of working away in the lab relatively successfully. And I have a few clinical samples on my computer or on the high performance cluster waiting for me to analyse at the moment. Another project that we have running at the same time was the accelerated anaplastic project. So you remember I mentioned those patients who had the really severe form of thyroid cancer. These patients present with these massive thyroid masses and are often in extremis with airway compromise, or are very, very unwell when they present. We can get rapid sequencing done on these patients, but actually I think a lot of centres use histopathology, use IHC to find out if patients have a BRAF V600E mutation, because dabrafenib plus trametinib is the main treatment we have for these patients. Now the problem with that is, if you get your histopathology takes a little bit of time, and if you use IHC for that and then they don't have a BRAF V600E mutation, we don't have many treatment options for these patients. There's nothing really surgically. Chemotherapy doesn't work, radiotherapy doesn't work. So what we need to be doing, instead of doing the kind of extra stains, is trying to sequence these patients early on, and it is kind of in the National Test Directory funded for this to happen, so that if people have ALT or RET mutations, then they can be put into solid organ tumour trials, and other therapies can be helped. The problem is we need to do this really fast because these patients, when they present, are often inpatients, they're really sick, and we say 3 to 6 months life expectancy, but we see a lot of patients who are admitted, diagnosed, and actually pass away during the admission with this diagnosis. So a matter of days in terms of the diagnosis is really significant here. So we're hoping to use our nanopore technology to rapidly sequence a panel for these patients, and we've had a bit of issue with the local samples we've used. It's a relatively rare disease, and happily set up a collaboration with the Anaplastic Thyroid Cancer Bank in the last couple of months, and they're sending their samples over to us really soon. So then just a little touch on some clinical projects that I'm moving on to now, and I kind of said before that the data behind the kind of tests that have been brought in worldwide, or really predominantly in America, hasn't got a huge or very robust amount of evidence behind it. When you look, there is one RCT, which NICE rejected when it did its last kind of review for thyroid cancer. And this RCT, rather than looking kind of the outcomes that we might be most interested in, compared ThyroSeq to Afirma in terms of the sensitivity, specificity, so kind of test statistics rather than the impact on patients. It was a really nice systematic review done relatively recently. And what we really want to know is the negative predictive value for these tests, because this test in the UK, the only thing we want it to do is rule out cancer at the moment. So we only really worry too much about the negative predictive value. The problem with this data, and with a lot of data that's out there, is for a lot of these studies, they have assumed that if the test is negative, they can leave the nodule in and have just followed it with a few ultrasound scans. A year follow-up, sometimes 3 years. And if it hasn't progressed, they've had no surgery and you don't have a histological diagnosis. So the non-operated nodules, they've assumed they're benign if the test told you they're benign. So what we wanted to do was go back and do another systematic review and meta-analysis, and we looked at all the tests, all of the studies, but we also looked at just the surgically confirmed histology studies to see what the negative predictive value was there. And actually, you do still get a decent negative predictive value from Afirma and to a lesser extent with ThyroSeq, but it's not kind of 100% like it was on the studies that you might have seen before. So our research question that we're putting forward now, alongside the lab stuff that I'm doing, is what's the efficacy and cost effectiveness of molecular testing for adults here in the UK? And we have an agreement with Afirma, who are happy to support in kind, as in provide their tests for free for this study to a value of £650,000. And we're putting together this application to NIHR to try and fund the study. This trial design is slightly complex. It was designed by one of our diagnostic experts in Birmingham. But what it does is it means that we have a randomized cohort with a benign result. So we know for these patients, this is who would be affected by a negative result, comparing observation, which is our intervention, with surgery, which is our standard of care. And we'll also get a group of people who have a negative result and have had their thyroid out, so we can confirm the negative predictive value that we have in that USA population, because we don't have this in a UK population yet at all. So in summary, genomics for a kind of accurate pre-op diagnostics is coming in the UK, but it is not here yet at all yet. Potential to be used for prognostication, I think that's in development still, really. And for anaplastic thyroid cancer, the targetable mutations are going to be really, really important and are expanding all the time as new solid organ tumour trials open up to our patients. But we have to get the sequencing done fast, so our patients are getting in there, so we get the evidence for the future. So it does feel like we're maybe entering personalised medicine. We've been talking about for years and years now. And this is a picture of me. Hopefully I'll be doing less operations on thyroids in the future. And then I have a picture of just, oh, it's gone now, of the Birmingham Thyroid Group, who are doing lots of other research on Graves' disease, radioactive iodine resistance, and radiofrequency ablation. So all sorts of exciting things going on in the Birmingham Thyroid Group at the moment. And I'll be happy to answer any questions. Thank you.
So we've got time for one or two quick questions. Munir.
Thank you, that was very interesting. So, great that you're doing this in Birmingham, but have you thought if your RCT shows utility, implementation is going to be quite difficult? Because I imagine there aren't many thyroid cancer surgeons who do what you do in terms of genomics and so on.
Yeah, I mean, the commercial test, especially the one that we're using, is quite nice because when you get it back, it tells you, it calls it benign. or malignant, or suspicious, I think. So benign or suspicious. So not everyone has suspicious arm will have cancer, but they're saying that the ones that we can leave in are benign on the test. So actually it's relatively a simple, more simple than we get really from like a genomics report now. And then you also get a nice paragraph underneath telling you these have a TERT promoter mutation, a BRAF mutation. And then if you know a little bit more, or you discuss your MDT, you say, well, actually, this person probably just needs to go for a total thyroidectomy straight away, or might need genetic screening, or whatever else is implicated by that. So yeah, I think it is potentially feasible to roll it out, and this is why the RCT will be multi-centre, try and see if we can include other centres and DGHs, people without expertise. The samples are also potentially going to have to be shipped on dry ice, so there's a lot of potential for feasibility issues.
One more quick question. I've learned that ENT surgeons do thyroid surgery for the first time. So, but a very good talk indeed. Thank you very, very much. And then we move into neuropsychiatry with Dr. Antonio Pardinas, I hope I've got that right, who's going to talk to us about can pharmacogenomic markers improve clozapine prescribing.
Thank you so much. Thank you to the organizers for having me here. It's an absolute privilege. Good morning, everyone. I come from Cardiff University. Bore that to any Welsh speakers in the room. Differently to my colleagues before, I am a non-clinical academic. I am a biologist, and I have been supported by the Academy of Medical Sciences until last year with one of the Springboard grants, which is the basis for this presentation. But you're also going to see some data and results from complementary projects to this that we've developed. I don't have any competing interests on this. So for non-clinicians such as me, there is always a temptation of drawing a healthcare journey as a linear pipeline. And if this fails in most cases, because the reality is much more complex, it definitely fails in psychiatry, where we know that the pipeline leaks all throughout this journey. We know, and this is data from countries, from high-income countries, we know that only 20% of people with psychiatric disorder seek and access treatment. And for severe mental illnesses, your schizophrenia, bipolar disorder, psychotic depression, and others, we know that only 35 to 50% of individuals go through something that we would call an effective therapy. An effective therapy defined as something which is a combination of adequate treatment, correct diagnosis, and then, of course, response to the treatment. So in psychiatry, there is a big drive for genomics to be able to fix some of the leaks in this pipeline. We've been working for many years in this discipline on genome-wide association studies, and there is hope that the development of polygenic scores is going to help with diagnosis. And of course, pharmacogenomics has a real opportunity of making a difference in how do we bring people with an accurate diagnosis to the treatment that is going to be more optimal for them. But of course, in the realm of research and in the realm of translation of all of these things to the real world, there are many complexities, starting for the fact that all sorts of different things have been associated with response. Here you have a list of variables that come from psychiatry studies. All of them are associated with different rates of response to a given psychiatric medication. But something that pops up really consistently, as with many other drugs, is that metabolism is an important predictor of response. And many psychiatric drugs have recommendations for doing therapeutic drug monitoring, a relatively simple procedure which allows you to bring the blood concentrations of a drug to the therapeutic range in each individual, controlling or adjusting or circumventing that person's metabolism. If the blood concentrations of the drug are too high, we know that there's risk of toxic effects. If they're too low, we know that the effects are likely not to be enough. But because therapeutic drug monitoring is, even though it's recommended for many drugs, is not always implemented, we are on a quest in the field for predictors, and this is where pharmacogenomics comes in. If we go to the PharmGKB database, a website that any of you can browse at any time, you will find guidelines, drug gene pair guidelines for many psychiatric medications. But these guidelines are very well known in our field to be decision support tools, as phrased by the International Society for Psychiatric Genetics. They're not algorithms. They need to be implemented in a very careful way as part of an interaction between the clinician and the individual with the mental health disorder, because there are many different presentations, many different drugs, many different treatment alternatives. There are some cases where pharmacogenomic consortia have highlighted that the guidelines are very likely to be so robust as to potentially being grounds for preemptive genetic testing. And here you have some drugs where the latest review by the Dutch Pharmacogenomics Working Group highlighted that they could be that preemptive testing could be potentially useful. And I'm going to talk about a drug that is not highlighted here, which is clozapine. Clozapine is an antipsychotic which is given only to individuals with treatment-resistant schizophrenia. Treatment-resistant schizophrenia defined as the failure to respond to 2 antipsychotic medications. It's a drug that saves lives. About 60% of those treated with clozapine get a certain level of response and get some therapeutic benefit out of it. It has some major adverse effects, and I will be talking about that. And it's also a drug where we would want to be accessible to more people in a safe way, because for many concerns, including the adverse effects, only a third of people that should be eligible for clozapine in the UK actually receive the drug. So the complexities of clozapine management are many, and that is probably better indicated for my clinician colleagues to speak about. But what I will say is that at the moment, the evidence reviewed by the Dutch Pharmacogenomics Working Group is that there is no pharmacogenomic markers for clozapine. The reason for this is that the evidence is just not there. The majority of the studies done in this field, and there have been many, have been fairly small and have only analyzed particular pharmacogenomic markers in a candidate way. So the study that I got support from the Academy of Medical Sciences through the Springboard program to carry out was a study called PATRON for pharmacogenomics of treatment and response. And it was based on the analysis of a sample that we have been very lucky to be able to recruit in Cardiff, which is the CLOZUK sample. CLOZUK is much larger than what I say here. At the moment, we have around 15,000 individuals. But the individuals that I analyzed for this project were about over 4,000 that had all the data needed to carry out. The idea was to do a project, a genomic investigation of clozapine metabolism based on that genome-wide association study design, looking at all of the markers throughout the genome, not only certain pharmacogenomic markers and not only in certain pharmacogenomic genes, but to look in as many markers as possible, focus on individual variants, and then focus on the pharmacogenomic markers separately. So CLOZUK is a sample derived from clozapine clinics. People that are taking clozapine undergo regular blood monitoring precisely because of the risk of adverse effects, of hematological adverse effects. Because everyone who takes the drug has to do this monitoring, first weekly, then monthly. We were able, through collaboration with one of the pharmaceutical companies that manages the clozapine monitoring system in the UK, we were able to collect anonymized samples. And for some of those samples, we were able to link them to longitudinal measurements of clozapine metabolism. And this is the sample that you see here. We did 2 rounds of genotyping on this sample, on 2 halves of the sample. We got about 3 million markers there, and as you can see here, the sample is multi-ancestry, which is very important for the research that we carry out. We, of course, have a much greater proportion of European individuals. About 80% of the sample would be genetically similar to European populations, but we have representations for other ancestries in the UK as well. We also managed to get a sample of individuals sent for exome sequencing, and we were doing the analysis using some of the latest models for analyzing longitudinal data. What came out of the common variant analysis, of the genome-wide analysis of the GWAS, was that clozapine metabolism, as we were able to represent it, is an oligogenic trait. We saw only markers in a few genes. If you're not familiar with this plot, this is a Manhattan plot. Essentially, you can see all of the different human chromosomes here. Chromosome 23 would be the X chromosome, and each dot is a genetic marker. The taller the dot, the more confident we are on its association with the phenotype. In panel A, clozapine metabolism. In panel B, the metabolism of norclozapine, which is the first metabolite in the clozapine pathway. You can see here only a few genes get markers over this threshold for statistical significance, and all of those genes are genes that we know were implicated in the clozapine metabolic pathway. So what? You might be saying, well, what we didn't know is that common genetic variation in these genes could be predictive of clozapine metabolism, because when people analyzed classical pharmacogenomic markers in these genes, they found nothing. And we didn't replicate in this study some of the, with relatively weak evidence, associations in other markers that have been reported so far. Because of the oligogenic genetic architecture of clozapine metabolism, one of the properties of this from a genetic point of view is that the markers that arise are relatively strong. And we see, for example, that just individuals carriers of one variant had differences in their clozapine levels that made them more similar to non-carriers on lower doses. Potentially, and with a strength of the effect that is potentially actionable, particularly for people that are early in their course of clozapine, where treatment doses are smaller. We also did the analysis of rare genetic variation was much more complicated, and in analyzing rare variants, you're more constrained by the sample size. So we did focus only, even though we wanted to look exome-wide, we focused only on genes that were known to be associated to clozapine metabolism or general drug metabolism. And our main association came out of this set of 33 genes, in which we saw that rare genetic variation in those genes, when looked as a set, it did correlate with faster clozapine metabolism. And indeed, when focusing on CYP1A2, the main gene responsible for the clozapine pathway, even one rare allele showed comparable effects to the effects we saw for common variants. So we have effects on drug metabolism. Some of these have already been replicated by colleagues, but we need more. We need to build up a base of evidence that shows whether these variants are actionable or not, whether it could be any worth to adding them to any clinical decisions. And at the moment, there's been some research carried out by an independent group, which has shown that individuals that carry two variants from the common variant GWASs had around 3 times the chance of getting subtherapeutic clozapine levels when normally treated. So individuals that would be treated with the standard, but who wouldn't respond to clozapine. And we've also have some research done on an independent sample. This is research that we carry out ourselves, showing that if we try to use a combination of pharmacogenomic markers to predict the activity of that gene implicated in clozapine metabolism, CYP1A2, that basically associated with ameliorated adverse effects on cognition, which is another one of the known adverse reactions for clozapine. Our focus on trying to build up this evidence base comes, among others, from our interaction with the Royal College of Psychiatrists, which had this report in 2023, which, disclaimer, we contributed, but so did many others, that showed that at the moment, at that point in time, and I say still quite true. There was insufficient evidence of even potential clinical benefit to recommend routine testing of pharmacogenomic variants in psychiatry, with two exceptions. One, for an adverse effect of carbamazepine, a mood stabilizer, if used in people of East Asian ancestry. And another one, interestingly, for clozapine, for people of African ancestry. And I'm going to talk a little bit about this research, because this also came from research that our group did. And this is related to those adverse effects of clozapine that I was telling you about. The one that really worries people is the hematological adverse effects, whether people that take clozapine might get neutropenia, or the neutropenia can progress to a granulocytosis, which can then be fatal. Rapid decrease of white blood cells. Clozapine is not the only drug that has this adverse effect. Other drugs do as well. So anything that I say now could be relevant for other drugs potentially. So if neutropenia and a granulocytosis is detected, clozapine is discontinued, which is a major event for patients. And it's a major event because if they have treatment resistance, and that is the reason why they are having clozapine, clozapine is the only evidence-based medication. If you take clozapine out, you need to be really, really sure that that is a beneficial thing to do, because there is no effective medication without it that we know at the moment. So African individuals are known to be more likely to show neutropenia. And so when put on clozapine treatment, they have revised thresholds to define whether the drug should be taken away or not. This is a condition that historically was called benign ethnic neutropenia, which is a name that at the moment is considered a bit problematic because of how it was developed. And the reason is that it wasn't really known why African individuals could display this benign ethnic neutropenia that wasn't seen in other populations. Our own research, and following up from research of other people, have shown that benign ethnic neutropenia is very, very likely the presence of just one variant, a common variant in a gene called ACKR1, which causes a phenotype called the DAFI-null phenotype. DAFI-null is a blood group, basically, and DAFI-null is the absence of DAFI markers from the surface of blood cells. That is entirely determined by this marker. The interesting thing of this marker is that if you do a genome-wide association analysis of neutropenia in an European population, you don't find it. If you do a genome-wide association analysis in a mixed population where Europeans are the overrepresented ethnicity, as it's often done, you also don't find it. And the reason why you don't find it is because it's exclusive to African populations. So could this marker, and you can see here stark differences in the neutropene levels, in the clozapine levels between, neutrophil counts between carriers and non-carriers of that marker. Could this marker be a genetic predictor for that benign ethnic neutropenia? And so could it be a predictor that is worth testing before undergoing clozapine treatment? The reason why the Royal College of Psychiatrists has said yes, and some evidence, particularly in the United States, is mounting up to say, yes, this is the case, is because clozapine has now been shown to be safe for individuals that are carriers of this DAFI-null marker. I would love to be able to talk to you about the biochemistry of this and the molecular explanation. There is an interesting story in there, but just to say now, clozapine is safe for DAFI-null carriers as far as we can see from the current data. So, which brings me to my last point, which has been mentioned this morning. Genomics historically has had a failure in properly representing the diversity and the ancestry of the world. We have a chance to be doing more and more studies to try to correct that and to incorporate people of multiple populations in our research, and even to do population-specific analysis in non-Europeans, which, as is coming out from our research in clozapine and from many other lines of research, is interesting because in some cases the variants are population-specific, and we wouldn't know about them if we only look at European populations. So I'm just going to finish by thanking everybody for your attention, and my colleagues at Cardiff, postdocs, PhD students, and my academic colleagues for the collaboration in this work. Thank you very much.
Okay, well, we're somewhat up against the clock, but I think we do need to allow time for one or two questions. And what a surprise, Munir has probably got 6 questions, but over to you, Munir. Can we bring a microphone down to Munir? Have we got another question at the back? Yep, great. So I'll come to you next.
Thanks very much for that. So the reason people use therapeutic drug monitoring is because they haven't got anything better, really. And therapeutic monitoring is only part of the answer. Obviously, the drug has to get into the CNS. There are transporters which transport clozapine out of this, out of the blood-brain barrier, which haven't been characterized. And obviously, there's variation in some of the targets clozapine works on. So how are we going to tackle all that? How are we going to sort of find out how clozapine actually works at the CNS level and what the genetic predictors are at the CNS level?
Absolutely. I mean, it's a very pertinent question. It is indeed really complex. I think there's work out there, also partly your own work, that also shows that the transporters that have come out of genome-wide association studies don't seem to be the transporters that actually transport clozapine. So in and out of the CNS, so that is even more complex. We're taking the pragmatic route of focusing on outcomes. Essentially, we have something, a phenotype here, which is molecularly very close to what DNA variants do. That's metabolism. We have phenotypes that are much more distant to that, which is drug response, whether people are getting better, whether people are getting adverse drug reactions, and many others. Our work is going on that direction, going on focusing, on looking, thanks to samples such as the one we heard about this morning, but also our own samples, looking at patient-reported outcomes, clinician-reported outcomes, and seeing, okay, are people who have these pharmacogenomic variants getting different outcomes, and if so, why? There's many intermediate steps there that we might not be able to directly assess, like transport and what the drug is doing directly in the brain. It would be great if we could incorporate that evidence, but we're going for the pragmatic route of focusing on outcomes, and most of our work is going on that direction.
Great. So question at the back.
Yeah, can I kindly correct what you said about the prevalence of the RS2847778 SNP in populations? It is present in mixed populations, so in Brazil it's about 3% of people.
Yeah.
Of course, in Africans, in Nigerian-born Nigerians, it's 100% of people. Yeah. But in people of African ancestry outside Africa, you still see a very high prevalence, 70% in Jamaicans, 65% in African Americans, even slightly less than 1% in Northern Europeans. And that variance accounts for almost all unexplained neutropenia in Europeans.
No, it's a really fair point. It was definitely an oversimplification on my part. Is the reason why, when we have been talking about the potential preemptive testing, the route that ourselves and colleagues are following is that everyone should be tested, because you could find it, even though it is associated with African ancestry. But it is a much better predictor of neutropenia. It is the predictor of neutropenia, while African ancestry only explains part of it. So yeah.
And it's worth adding that there's a recent paper looking at people on clozapine, showing that genetic test identifies hitherto unrecognized cases of BEN, which hadn't been diagnosed clinically. In fact, there were more cases identified with a genetic test than had already been identified clinically.
Yeah. Okay, so thank you very much, Antonio, for that. And as we finish this session, I just want to also highlight the Springboard Awards of the Academy, which support non-clinical researchers who are early at the initiation of their academic appointment. And I think it used to be £100,000 was the award, but it may be more than that now. But certainly a substantial amount of funding to support people to get their work off the ground. And I think we've seen 3 fantastic examples of people who've benefited from those awards. And if there are funders in the audience, I can tell you the Academy is privileged to be able to assess and to make these awards, but it's really only because of the contributions of the funders. So I want to thank the funders, and if there are other funders who would be interested in contributing to any of these schemes, please do let us know. But a great session, thanks to all three speakers, and I think it's now time for lunch. Thank you.
Thank you.
So welcome back to session 3. We have a packed program today, and I'm very pleased to say we're going to move on and talk about the fascinating topic of highly personalized medicines. Personalized, not enough. We're going to move on to a new level. And we have a very diverse faculty to talk to you today. We've got folks looking after the regulatory side of this. We've got folks from the clinical and research communities. Most importantly, we have patient advocates, parents, and patients living with disease. And I think it's very important that we have an opportunity to recognize really why we're all doing this and why we're here today. So I think it's going to be a very entertaining session. I'm pleased to start off by introducing to you Dr. Shirley Hopper. Dr. Hopper comes from the Medicines and Healthcare Products Regulatory Agency. She has a history of working with pharmacogenomics, being on the original pharmacogenomics working panel at the EMA that set the European guidelines for pharmacogenetics, and she retains that special interest. Today, she's going to talk to us about the challenges and progress with the regulation of highly personalized therapies. Dr. Hopper.
Thank you very much. Okay, so this is just an outline of the presentation. I'm going to give an introduction and overview, and then really focus down on a particular type of product where we have been thinking about a regulatory pathway, and we published some draft guidance, which is individualized mRNA cancer immunotherapies, but then explaining how this approach could be broadened to other sorts of individualized and highly personalized therapies. Okay, so at the MHRA, we've been working for the last couple of years to provide a clear and comprehensive regulatory framework to support really the development, but also the evaluation and ultimately the licensing approval of highly personalized medicines. Because by really trying to streamline development and the regulatory review that we do, we hope to facilitate access to these innovative therapies by speeding up patient access, particularly in areas where there is high unmet need. So what we did in 2023 is we convened an independent highly personalized medicines expert working group to really advise us on both the development of these therapies, but also what might be the requirements for licensing. So thinking about quality, safety, and efficacy. Also post-licensing data collection that we might need for these therapies. And also how we would communicate to patients and the public and other stakeholders. So initially, as I said, we have been focusing on a particular type of therapy that is in quite late clinical development now, which is the individualized mRNA cancer immunotherapies. Now, these are commonly known as personalized cancer vaccines. And in February, we published a draft guideline, and that actually went out for consultation, a public consultation for 2 months. And we're currently reviewing all the responses that we got from a range of stakeholders with a view to updating the guidance later this year. And the principles that we are developing for the cancer immunotherapies will, in many cases, also apply to other types of highly personalized medicines that might utilize other technologies or be for other conditions such as rare diseases. So just to give you a bit of an overview of the cancer immunotherapies. Some of the products that are in later clinical development now are based on the COVID-19 vaccine technology using mRNA. And these medicines are generally being developed at the moment for patients with cancer, but with quite a low tumour burden. In the adjuvant setting, which is the post-operative setting. The active ingredient of these medicines is mRNA, and it's encapsulated in a lipid-based nano-drug delivery system, and that really allows the mRNA to be delivered to the cells where it has its effect. And what's really innovative about these therapies compared to the medicines that we normally review is that there is a fixed component of the mRNA, but also a variable component. And that component is tailored to each patient's unique tumour antigen profile or tumour mutation signature. And that is done in some cases using AI. And then after the medicine is administered to the patient, the mRNA molecule is delivered to host cells where tumour antigens are then expressed, and that generates an immune response against the patient's own tumour. And for that reason, what we emphasise in the guidance is that tracking and traceability is really, really important for all the steps from originally sampling the patient's tumour all the way back to administering the medicine to the patient. And that's shown here. So you've got the patient at the top. So what happens is their tumour is sampled, most often as part of the surgical resection. And they also give a germline sample, so for example, a blood sample. And then these samples are sent away for nucleic extraction and sequencing. And then there's a bioinformatics and artificial intelligence step that then designs the mRNA therapy individualized to that patient's tumour antigen profile. So that is then informs the design of the starting material for the manufacturing process. And then there's then an individual patient batch manufactured, which then has to be released and then administered to the patient that gave the original tumour sample. So it is a cycle. So we've been thinking, well, what would be our regulatory approach to these very innovative sort of medicines? So we would regulate them as advanced therapy medicinal products, or ATMPs, and we can do that under our existing human medicines regulations. However, because there is this product design aspect involving sequencing and then analysing and use of AI to inform the mRNA design, we are proposing in the draft guideline that this step would also be regulated under our UK medical devices legislation, and I'll talk a little bit more about that later. Now, these products are all slightly different. They're individualized. The active ingredient differs slightly, but we wouldn't be able to give a license for each individual version because obviously that just would not be feasible, especially in the oncology setting. So we're proposing that a single marketing authorization or drug license would cover use of a product across a target population, even though there is this variable component tailored to the individual. But it's really important that then the product design process, so including the sequencing, the AI, bioinformatics, also the manufacturing site and the process, all that would need to be the same across the different individualized batches. And there would need to be flexible but predefined processes and controls so that we can reasonably expect that the product can be safe and effective in clinical use once we license it. So as I've said, we would classify these as ATMPs, and under that we will classify them as gene therapies. Now, the advantage of ATMP classification is it gives us a flexible and risk-based approach to our regulatory requirements. For example, we do have some precedent for this, such as CAR T-cell therapies, where there are differences in the active that is administered to the individual patient. Now, this applies to the current products that are being developed using biological processes, such as a plasmid, but they would not necessarily apply to chemically synthesized mRNA therapies that we might see in the future. Another aspect is that the mechanism of action of these does not involve integration into the host genome. However, the classification currently is the same as for gene therapies that do edit the host genome. And this sort of lack of distinction can sometimes lead to at least a perception of overburdensome risk mitigations compared to similar technologies that we would sort of classify in a different way. For example, the COVID vaccines, which are a very similar technology, are not classified as ATMPs, but are classified as vaccines. And so there is a different requirement there. So in view of all of this, we are considering whether we need to introduce a new ATMP subclassification for nucleic acids that do not edit the patient's genome. Whether we might also classify chemically synthesized products as ATMPs in some circumstances. And also we're considering whether we need to introduce a specific provision for sort of individualized and other highly personalized medicines within our regulations. So just talking a little bit more about the product design step. So in the guidance that we published, we cover aspects such as sample collection and storage. Looking both at the tumor and the germline. We talk about genetic sequencing, bioinformatics, which is the part that distinguishes the tumor-associated gene variants from the germline variants. So these steps are actually covered by in vitro diagnostic medical device legislation. And then the sequencing platform, including software for secondary analysis, must also meet GMP requirements. And research use only platforms really should be avoided in this context because this design really has a medical purpose. So once we've got the sequencing analysis, the artificial intelligence system would receive the sequenced genome and output the potential tumor antigens for mRNA manufacturer. And this comes under software as a medical device regulations. And the guidance covers some considerations when using AI in this way, such as performance, generalizability, bias, which we heard a little bit about earlier, transparency, and also what happens if there's an update to the AI. Now, currently, algorithms using continuous learning are not currently compatible with UK medical device regulations. So another important aspect is that developers will need to put in place systems to record which version of the AI perform the antigen selection for each batch of the medicinal product so that we're able to trace back from the patient all the way back to see which version of the AI designed that mRNA sequence. Now, many of these principles are already being discussed among regulators, and the MHRA, along with the FDA and Health Canada, have produced some high-level principles, which you can have a look at in the link that I provided, which is a good machine learning practice. So just moving on to the manufacture of the drug substance. So this is standard in that it's regulated under good manufacturing practice guidance, and some of the key issues that we cover in the guidance, where there are specific considerations for individualized products, include the starting material, in vitro transcriptional synthesis, obviously the constant and variable elements in the molecule, characterization, process validation, and also potency and functionality testing of the active. And where there is prior knowledge from other products, such as similar mRNA technologies, then that can be used as supportive data. So moving on to the drug product, this is where the mRNA is incorporated into the drug delivery system, which currently we're seeing lipid-based. So there's sections on characterization, batch definition, stability. And what's really important, particularly for cancer patients who've had surgery and are now waiting for their therapy, is to have rapid release testing. And that's quite a challenge, but that's something that we're doing a lot of thinking about, how that can be streamlined, but still ensure the quality of the product that the patient receives. So then, as for most new products, we would expect non-clinical testing, and the initial testing should really prove the principle that the mRNA construct produces a specific immune response with an anti-tumor effect. And it could be acceptable to cross-refer to studies with other mRNA constructs if relevant. And testing would also be expected to explore safety in relevant settings. And then this data set from the non-clinical could then support the use of the individualized product with variable mRNA components. So again, clearly we couldn't have non-clinical testing happening you know, in a major way for each individualized batch while the patient is waiting for their treatment. However, if the mRNA backbone changes or the delivery system is new, you know, there may need to be new product testing in that situation. So moving on to the clinical aspects, which is more in my comfort zone. So we do make reference to the EMA guideline on the evaluation of anti-cancer products in humans because, you know, we are expecting a classic oncology drug development for these products. And we feel that that kind of clinical development can support an individualized product. Ideally, the product design step should be fixed for the duration of the pivotal trial. So we don't want big changes happening halfway through the trial because that would make the results really difficult to analyze. And obviously, if there are subsequent changes to the design of the product, such as the AI or bioinformatics, then we would need the license holder to really justify that doesn't impact efficacy and safety and that they don't need to generate, you know, new clinical data. So we also include some specific trial design considerations. We're not prescriptive about the sort of clinical trials that should be done. There will be cases where adaptive designs or single-arm studies might be acceptable. But in many cases, we would, as with most oncology drugs, we would want to see a randomized controlled trial if feasible. Now, what's challenging with randomized controlled trials is there is this need for a manufacturing time from the time that the patient is potentially randomized into the trial, so the time at which they have their surgery and then provide tumor for sampling, and the time that they actually then receive their immunotherapy. And also that time can be variable between patients. So how to actually incorporate that into the trial design is quite challenging. So, for example, you need to think about how you would maintain the study blind when you've got this variable manufacturing time. And it's also really important that any control group does receive standard of care. So if they are receiving standard treatment, you would not want them to have a delay or an undue delay to that treatment because the experimental arm are waiting for the immunotherapy. And it's also important for sponsors to think about what the study objectives are. So that might differ depending on the chosen study population. Is it going to be all patients that are being considered for the immunotherapy, or is it only those that actually then receive it once they've sent their tumour off and then the batch has been released? So then after licensing, or at least preparing for the post-authorization period, it's really important that the license applicant identifies all the relevant safety concerns, thinking about ATMPs and individualized products. And we would expect a post-authorization safety study. So we have included that in the guidance. And developers could consider how to tell if there is a post-marketing safety signal, so something that wasn't found in the trials but is a potential safety issue in the clinic. How can that be related back to the individualized mRNA sequence? So again, that comes back to this traceability. And obviously, again, post-authorization, developers need to consider what is the effect of any changes to the product design that selects the tumor antigen. Can we then still rely on the efficacy and safety data that came out of the clinical trials? And it may be that in this case, there are some additional risk minimization measures that may need to be implemented. So it may be important, for example, that healthcare professionals get some educational material to help manage risks. Okay, so the final section of the guidance really deals with information for patients, healthcare professionals and the public. And it really is, we feel it's important to try and provide good quality information for these groups. And the current format of our regulatory documents, so we provide a summary of product characteristics and a patient information leaflet, is quite challenging to convey adequate information about these novel therapies within that. But I think we're going to need to find another way, probably working with partners to be able to do that. So we're going to include in our update to the guidance a little bit more information on that based on really helpful consultation feedback that we've had. But really, the patient will need to consent to this treatment quite early, at least consent to their tumour being sequenced and potentially considering the therapy. And they also need to have sufficient time for an individualised risk-benefit discussion with their healthcare professional. So these are real challenges when providing this information. So I'm just going to quickly run through the consultation responses that we got back for Section 8, the patient, public and healthcare professional information section. So, we had 48 separate respondents across major stakeholder groups, so developers, academic organisations, public sector, and patient organisations and charities. And we had 600 lines of comments. And as you can see, yes, individuals provided the majority. So we had the most comments for the Section 8 information for patients, healthcare professionals and the public. And we asked about type of information, format, and also appropriate timing of providing the information. So we had a lot of comments about the type of information. I mean, I'll just, a lot of this you would expect, but people were really keen to know about what's the evidence for this. Is the clinical trial evidence relevant? And they also want to know what would be the other options for me at this time. We also had comments about what is the impact on my daily life of agreeing to this? What other procedures will it have to be delivered in a specialist setting? What is the impact on my carers, my relatives, travel time, all those sorts of things. And comments on also samples and data, who owns the data, and how will it be used, and will I have access to it? And then we had some more general comments on format. Again, multiple formats really, and accessible, and use of sort of video as well to try and explain some of these really complex messages. And then really early information, but sequence and plenty of opportunity for follow-up, and most importantly, time to ask questions. So I provided a link to the draft guidance, and as I've said, we are analysing that, and we hope to publish an update before the end of the year, and we will expand the guidance in due course to cover other types of highly personalised medicine. And we always advise scientific advice for your individual product. Thank you.
Thank you very much indeed. We have time for some questions. Please. There's a microphone on its way. It's a long way down.
This is Akram from Pakistan, visitor here. I learned the name of Pirmohamed Sahab, Sir Pirmohamed. So I thought I must go and join this conference. This is something relatively new for me, most of the. But one thing I'd like to make clear, to learn more from you. As we know, the viruses, they are known to develop changes in their RNA, DNA against the chemotherapy, for example. So then those antiviral therapies become ineffective. Similar biological changes can occur in human RNA also, and make problem for development of therapies, and ultimately in the clinical application also, after few application may become ineffective. What is the possibility of that?
Yeah, well, I mean, that's something that obviously we would look carefully, particularly as part of the non-clinical testing, to see whether there are any potential risks to the therapies, and also see what we've learned from our experience with other mRNA products. But obviously the reason why we ask for a post-authorization safety study and long-term follow-up is to really identify whether there are any longer-term important issues with this therapy.
Because those mutant changes can make problems in the diagnosis or setting the group also. So false positive, false negative results are possible during testing these groups.
Thank you.
Is there any studies for that also, for the relevant studies?
As I say, as part of the non-clinical testing, then we'd obviously look at the potential for those sort of risks.
Thank you. Question at the back.
Yeah, thanks, Shirley. Dan O'Connor, ABPI. Really nice to see this guidance coming through. So this is currently positioned in the oncology setting, and there's a potential for large sample sizes for you to be able to conduct a relatively normal development program. I thought what was really encouraging within the guidance was that you alluded to the fact that this might be applied, or at least some of the principles might be applied in the rare disease setting, which is really important when we think about some of these platform technologies that are coming through. Can you say any more at this stage around what elements of the guidance might be applicable in the rare disease setting? Thank you.
No, thanks, Dan, for your question. So I think really we're looking broadly at our legislative framework to really sort of future-proof that and to think not only about the mRNA cancer immunotherapies that obviously are in sort of late-stage development, but think about other disease settings such as rare therapies and also other sort of technologies that might not fit so easily in ATMP classification. So we really are having a look at our legislation and looking to future-proof that. You know, and as I said in the talk, looking at not only being able to incorporate sort of chemically synthesized products, but also have a clearer provision for highly personalised medicines to be able to give, you know, a single marketing authorisation for other types of medicines that where there is individualisation, such as some of the rare disease therapies that we hear about. So, yeah, and obviously, yes, the traditional clinical development certainly isn't necessarily appropriate. We do have already some mechanisms such as marketing authorization under exceptional circumstances where we can consider very, very small data sets where it just isn't possible to generate the usual amount of efficacy and safety data, and then we can have sort of annual follow-up of those. Certainly in areas where there's serious condition and very high need, obviously we would really sort of consider whether we could authorize these products. We're also obviously having a think about whether there are other routes other than licensing that might be appropriate for some of these products, and that's something that we're discussing, and we may be able to provide some published guidance on that in due course.
Thank you very much. Thank you kindly. So moving on, I'm delighted to introduce to you Alison Watson. Alison is the CEO and co-founder of Ring20 Research and Support and the founder of the UK Rare Epilepsies Together network. I'm pleased to invite her here to talk to us about living rare, from parent to protagonist, advocate to activist. Alison.
Thank you so much, first of all, for inviting me to speak today and to give you a bit of a whistle-stop tour of my journey over the last 20 years, what I've learned and how that might be relevant to this audience today. First and foremost, I'm going to come up with a disclaimer that I am from no scientific or medical background. First and foremost, I am mum to David. He's now 28. He's living with a rare chromosome disorder, ring chromosome 20 syndrome, which is a super rare form of epilepsy. Only apparently 200 cases reported worldwide. Yeah, we'll come back to that one. And some of what I'm going to say through my talk today might come across as a little provocative, but I say some of these things to promote thought about opportunities for rare disease and how maybe we can think more broadly, more laterally to help more people. So my journey starts from being parent, mum to David. And David, after normal childhood development, at 6 years of age, we found him one morning. I'll never forget, because it's the day after my husband's birthday, and we'd been out celebrating with friends, and we found David slumped in a corner in his bedroom, and he was lifeless, like a rag doll. Just scared the living daylights out of us. Rushed him to hospital, and they said, after a few investigations, "We think your son's had a seizure." I went, "Pardon? Yeah, where did this come from? Perfectly normal, healthy child. How did that happen?" And we fast forward through the next coming months, 6 months or so later, after several investigations, starting on some initial meds, And his behaviours were a little bit unusual and erratic, but we really didn't understand that. We had a sudden and severe onslaught of seizures, ramping up to 30 seizures every 24 hours, which was quite something to be dealing with. And our journey from that point in time has really been a roller coaster with this really refractory form of epilepsy that just does not respond to any form of treatment. We've been on over a dozen anti-seizure medications in various combinations over the last 20 years. He's tried ketogenic dietary therapy. He's had a VNS implant. None of these things have really got any real control over the seizures. He currently now has several non-convulsive status epilepticus episodes, NCSE, every single day, 4 to 6 per day, lasting 30, 40 minutes each time. And about once a month or so, he'll have a full-blown convulsive seizure at night, which, for those of you who know, now runs a high risk of sudden and unexpected death in epilepsy. So it's a truly scary condition that's not only changed our lives. I took a lot of time off work, went back down to part-time, and ended up in the space that I'm in today. It's been life-changing not only for David, but for his family. And it's life-limiting for David. It has impacted him through every aspect of his life so far, through educational support and us having to battle and fight to get that support in place, which you'll hear across the rare disease community, from accessing benefits to getting support from social services. And now, as he's moved into adulthood in the last 10 years or so, even though he's less mature for his age, you know, trying to achieve some level of independence, albeit on a supported basis, a supported living. You know, let's forget the word work. You know, people think he can go out to work, but he has to be supported for an hour or two's volunteering at a time. So from being a parent, that led me to be inquisitive by nature. I'm a business analyst by profession. I want to understand why things happen, and I want a logical progression on things. So I got immersed, firstly in the epilepsy, and got involved in the local epilepsy action group, tried to help and support them, helping to promote a local schools information booklet series that we got funding for, and I helped fund through fundraising, to helping out with a community patient advocacy group that existed at the time that David was diagnosed, which was fantastic, and finding my tribe, finding my community out there. But that died away about 14, 15 years ago, and that sense of community was hugely valuable. So with a colleague, it took several years for us to get to that space, but we founded a completely new patient advocacy group, Ring 20 Research and Support UK, and to date we are currently the only group for this particular condition, supporting families across the world. And we support something like 130 families, so over 60% of the reported number of cases in medical literature. And if I go back to that point, we've just had some members in Japan from our International Scientific Advisory Board who published a paper on 47 cases in one centre in Japan. So underdiagnosed, yes. Underreported, absolutely. So we've grown a patient support group and become a champion for Ring 20 across the world. But my advocacy then just exploded and explored. There I am, working part-time, caring for David on the side in the other 24 hours of the day, and immersing myself in volunteering, running the patient advocacy group, and then getting much more involved. What I found was that the epilepsy community kind of are an umbrella where all epilepsies are one. And we've heard about cancers today, and I think all of you in this audience wouldn't think of cancer as one condition, but actually lots of people consider epilepsy as one condition. And epilepsy, or the epilepsies, particularly the rare and complex, the very refractory and comorbid conditions, are rare diseases in their own right. And there are very many of them. And the rare disease community is much more welcoming of this. And that's where I've found my advocacy space. And I've had to do a huge amount of learning. I've had to immerse myself and start to understand genetics and genomics, because my son has an ultra-rare chromosomal structural variant, not a single gene disorder that I can get my name around some horrible code, ABCD1. No, he has a ring chromosome, but we don't know why that ring has formed, and we don't know what genomic change, because something's changed to make the ends of those chromosomes sticky, and we don't know in any ring chromosome, in any of the chromosomes in the body of rings, why those ends stick together. And without that, it's difficult to diagnose, and we can't address new therapeutic advancement. So I'm out there now talking, speaking, networking, trying to generate interest in research. And because we're a complex biology, and if one person tells me one more time that ring chromosomes are complex, I'm going to scream. You know, we still need research. And so we're driving patient-led research from patient impact reported study that's coming out later this year. It's been about 2 years in the making, and it will be the largest study of any kind beyond the clinical and genetic studies, which have studied 25, 30 patients at a time. We're talking about 84 patients around the world that have contributed. But beyond my work within Ring 20, I got involved at the European level, so I got invited to join the European Patient Advocacy Group for EpiCare, the European Reference Network for Rare and Complex Epilepsies. And I ended up co-leading the EPAD group for 5 years, where I not only learned where Ring 20 fit within the realm of rare conditions and rare epilepsies, but also the differences and the similarities across rare epilepsies and rare diseases generally, and the challenges that we face, but the opportunities to make change happen. I became a Eurodis volunteer, and I did some work then that led to a short stint of work for Rare Diseases International, where I looked at the global picture of the collaborative global network for rare diseases as that was emerging. And what that taught me was that before you can look at rare diseases on a global scale or even a regional, European scale, sometimes we have to look a little bit more closer to home and leverage what we can do nationally right here in the UK with the fantastic resources, the fantastic NHS, the science and technology and companies and minds and academics within this country that can really change the things. And so, about 3 years ago, I then took on a little bit more just because I like a project. So I created the UK Rare Epilepsies Together network, and I brought together the majority of UK-based rare epilepsy patient advocacy groups and the umbrella national groups to work on a collective proposal to really address the unmet needs across this group of diseases in diagnosis, treatment and care, and really to address the challenges within NHS service delivery, within the research progression, and really try and move the dial forward. I'm really proud to say that we have done a number of projects already. We had our first patient advocacy group roundtable with Genomics England, which was fantastic, and we have a white paper style report coming out that you can see. This was an extract from the challenges roundtable, but the solutions that we're coming up with is due out in the next month or so, and we're looking at that as a major call to action. So let's reflect on rare diseases, because we haven't heard about this morning, so quite a good spot to come back here. 8,000, might be 9,000, depends on who's counting, a lot of rare diseases out there. Amongst those, there's something like 160 EpiCare talk about rare and complex epilepsies. And within that, 1,000 nearly pathogenic genes for epilepsy. And yet they're not 1,000 different phenotypes, 1,000 different syndromes of epilepsy. There is a lot of commonality. So where are those common underlying mechanisms of action? And where can we look at those to address change? But we have heard today that 5% of rare diseases typically only have effective treatments, so there's a woeful shortage, but opportunity there to move the dial forward. And diagnostic yield even with the fantastic work of whole genome sequencing and the newer standards and technology, is still woefully low at around 30 or 40%. There's still an awful lot of people that have severe problems that remain undiagnosed. And that whilst there are millions of pounds, and this came out in the rare disease research landscape recently, being invested in rare disease research, it's in a handful of rare diseases, not the thousands that exist out there. So the unmet needs of the rare disease community, you know, we're all looking to genetics and now genomics to unlock the key to diagnosis, and we're seeing this revolution, this explosion in diagnosis. It's not fast enough, but it's definitely ramping up and it's getting better. And that then leads to understanding of these genomic variants that we can then apply to scientific advancement and really look at precision treatments for the future. We've heard about the precision medicines. But not all rare diseases are genetic in origin, and not all genetic diseases are monogenic. And there's a massive focus on single gene disorders, less so in the large CMVs, the chromosomal variants, or the structural variants. I'll illustrate this and come to my son's condition as an exemplar of this. In ring chromosome 20 syndrome, we've actually plotted, as you can see here, diagnostic decline over the last 5 to 10 years, and that goes against the trend for most genetic disorders. Why is that? That's because none of the newer sequencing techniques being used out there, and most of those that are being lauded within the Genomics test directory, can detect a structural variant. We have to go back to old-fashioned karyotyping. And so this advancement in technology has actually made us go backwards. And that's a really worrying trend. And it's not acceptable. But why is this complex variant? Well, as I said earlier, you know, we have this ring chromosome, but we don't know the cause. Is ring chromosome, that's not really the cause of the problems. That's another symptom. We've yet to find the cause. And until you can find a root cause, you can't address the problem, can you? And what makes it even harder is ring chromosomes typically occur in a mosaic form, and in the majority of our patient population, the majority are mosaic. So some of the cells within the body are rings, and some are normal wild-type chromosome 20s. So when you're sequencing, if you don't look at enough cells, and this is where the newer technologies often fall down, and even karyotyping, if you don't look at enough cells, you're not going to see the rings. So underdiagnosed, undiagnosed, misdiagnosed. So they're causing barriers not only to diagnosis, but to therapeutic discovery. Because we don't know the cause, we can't apply our minds to gene therapies and the ASOs, all this fantastic technology that's emerging, the gene editing. We can't even look at drug repurposing because we've got no biomarkers. We've got no reference points to look at. And where can we apply pharmacogenomics or genetics to this cohort? Because they're getting karyotyped mostly. None of them have been whole genome sequenced because the end of the line for their diagnosis is you've got a ring chromosome. You don't want to look further. And actually, what more could the genome of these individuals tell us? So these come to a couple of unanswered questions. You know, what more can we learn from studying the wider genome? Why stop at looking at the physical symptom that we see under the microscope? And yeah, is that structural variant really a diagnosis? But so what? Well, you know, there is, as I've said, a big focus and concentration on single genes, but actually the larger CMVs, the structural variants, they're not the low-hanging fruit. And I come from a rare disease background where the mantra is leave no one behind. I'm actually seeing the opposite here. So I'm a bit concerned. And my request to you is to stay open-minded. I want you to look to the future. Yes, keep this huge explosion and progression going, but don't ignore what we already know and continue to explore the human genome, because we need to look at it. I've learned, you know, there are many different techniques at looking at the genome, and all have their purpose. So, you know, unlock those secrets and try and find the answers for the many, not just the few. If I leave you with a couple of passing thoughts, you know, I'm talking for rare disease, but, you know, the epilepsies is really where I champion, and, you know, we're massively under-researched. We're massively undervalued, and we're massively misunderstood. Within those neurological conditions, there's a huge move towards looking towards dementia. And dementia typically is there for people within old age, maybe people who have lived long and fulfilled lives, and the ends of their lives, physiologically, the body's deteriorating. You know, this is something that's happening. But actually, epilepsy is going to become more prevalent in old age too, because as people are being kept through stroke and heart conditions and other conditions, you know, possibly we're going to get an explosion in late onset epilepsies too. So we think it's time for change. Do you agree? So I'll leave you there, and, you know, precision medicine, pharmacogenomics, genomics, it can tell us a huge amount. And precision medicine, you know, it's here to stay, but how can we precise to the many millions of people around the world? And just coming back on a passing thought on the regulators and the payers, the concern from the rare disease community is, are we ready for this? Are the regulators ready? And I've heard hints just now, so that gives me some hope that the regulators are thinking differently at how they need to think about, you know, these new technologies and these new therapeutics coming forward, and how we can license them and approve them. But are our payers ready? Is NICE ready for this explosion? Because it's coming, they're in the pipelines, all of these new therapeutics. Can we afford them? So even if their technology is out there, will the patients be able to see the benefits in our lifetimes? I hope so. I hope you can help. Thank you.
Thank you very much. We have time for some questions. May I ask a very selfish question here? I work for a pharmaceutical company. How can the industry be the best partner for patient groups like yourselves?
Talk to us. You know, that was part of the reason for setting up the UK Rare Epilepsies Together network, because we recognise that some of the more common or interesting to research rare conditions have those relationships with pharma, so there is a more understanding on that handful of rare conditions. But there are so many, aren't there, rare conditions? So you don't know what you don't know. So talk to us. We become the patient experts, the expert patients in our conditions. We have a lot of knowledge that we can share with you. And I've had to learn this whole journey of genomics over my lifetime. And it's really interesting that I'm here today. And we've heard Dr. Rich Scott talk this morning. And actually 20 years ago, I was in clinic with him with my son. And it's really sad to think that 20 years on, we really haven't moved much further forwards. We know no more about ring chromosomes. But actually, there is a lot that is moving in this space. So, you know, I use my experience with Ring 20 as an example of so many other advocates in other rare conditions. Talk to us, engage with us, because we absolutely want to talk to you. And if you can support us in our work of advocacy, in our information sharing, our opportunity for networking and raising awareness, perhaps together we all play a part, and that we can be a valued player in this space.
Thank you.
Can I make a little comment?
Well, Munir first, and then perhaps if we come back.
Thank you very much. That's really amazing story. Just to understand the variability in getting to a diagnosis of the 80-odd patients' families that you are in contact with, I presume you got to Great Ormond Street eventually, but probably took you a while to get there. So what's the variability in terms of time, people getting specialist advice and the diagnosis? There will be a big diagnostic odyssey here in this patient group.
It's huge. So in centres that have an awareness in tertiary centres that do have an awareness of Ring 20, they may spot the red flags, and we as a patient community believe there are some key flags, hallucinogenic type seizures, particularly at night, etc., etc. These very prolonged NCSE, rather unusual in the refractory epilepsy population, so should be red flags. Those centres that recognise that then put forward for appropriate diagnostic testing, and we've actually got a map on our website where we plot the cases around the world and around the country, and there are hotspots, and you can see those hotspots in the different centres where they're more aware of signs and symptoms and correct diagnostic testing. But in terms of range of phenotype and how long that diagnostic odyssey is, we've heard cases diagnosed within a few months to, I have a case in Canada where it took over 20 years for an individual, and she had stroke-like episodes, and her neurologist said to her, and she quotes, he said, "It's all in your mind." So it's being misdiagnosed as psychiatric disorder, goodness knows what, and the degree of severity, we are talking about low-level mosaicism that have mild cognitive impairment and less seizures and later seizure onset, right through to non-mosaic cases or high levels of mosaicism with multiple daily refractory seizures. We've lost 4 individuals out of the whole patient population in the last 2 years from seizure-related or SUDEP deaths. This is life-threatening, and people in wheelchairs, tube-fed, residential care. It is quite devastating condition, and it needs to be acknowledged as such, and awareness needs to be out there. Thank you.
Okay. Last quick question here at the front, and then we'll move on.
It was a really very nice presentation and very informative. What I learned from this, so far, all the anti-epileptic therapies, they are based on empirical basis. We try and epilepsy is controlled. We are not sure how they are controlled, except very little sodium ion channel involved, this and that. But we know very little about it, which is true. I think we need to, maybe we are able to find some genetic factors and other relevant factors. But I think we should concentrate on epilepsies based on our therapeutic goals. Which type of changes are more controlled by which type of anti-epileptic drugs. So this may help us to find a better approach for therapy, epileptic therapies.
Yeah, absolutely. That's a really good point about, for what can, we know anti-seizure medications, a third of people with epilepsy are refractory, have uncontrolled seizures. But within that, with the new anti-seizure medications coming through, there's always typically that 40% responder rate in terms of successful therapies. And we don't know with people with the same phenotype, same etiology, whether they fall into the 40% they're going to have a good response or not. And there's really great hope for some of the work that I know Sanjay Sisodia, for example, is doing, looking at the wider genome to see whether that can tell us anything in terms of drug response to existing anti-seizure medications, and which people will respond better to which meds. But also in terms of response to other interventions for comorbidities. Because if we can't control the seizures, can we improve quality of life elsewhere with these really complex cases? So, thank you.
Thank you. Thank you very much. And so now it's with considerable pleasure that I invite Jack and Alex Johnston to come and talk to us about the most important perspective in this, the patient perspective. And they're going to talk to us about time is muscle. Thank you very much.
Thank you. Actually, your talk's a very lovely segue into my talk, so thank you. So, in 2011, when Jack was 3 years old, we received the devastating news that he had Duchenne muscular dystrophy, which, for those of you who don't know, is sadly a life-limiting progressive muscle wasting conditions. There were no approved treatments, and it actually felt like there was no hope. But all those years ago, we were given a choice. We either accepted the disease and what it was going to do, or we were going to fight it. And we chose to fight it because we were not going to be defined by the emotional and physical response to it. We were going to be defined by what we were going to do. So that's Emily Crossley, who I actually found my tribe with her. She's another Duchenne mum, and her son Eli, who's become one of my son's best friends. So we came together to found Duchenne UK. We begun purely as a medical research charity with the aim of pushing forward new treatments. In 13 years, we've raised over £30 million, and we're slowly seeing those treatments become a reality. It's an exciting time, and it feels like we're on the cusp of real change. I'm not going to try and present this whole slide, but as Duchenne UK, we've solved problems as they've appeared, and some initiatives, like for instance, the DMD hub was created to expand clinical trial capacity. We had no economic model for Duchenne, so we set up Project Hercules. We've developed care guidelines through a project called DMD Care UK. We actually funded the first gene therapy clinical trials, the first stem cell trials in the US, and we've even got into technology and we're developing an upper body suit. But today, more importantly, you're here to hear Jack's clinical trial journey and why we founded the Time is Muscle campaign.
Hi, I'm Jack and I'm 17 years old. I was diagnosed with Duchenne muscular dystrophy when I was 3 years old. Since my diagnosis, my friends and family have campaigned tirelessly to fund research into finding a treatment for Duchenne. I can never thank people enough for everything they have done to help me and the Duchenne community. In my short life, I have taken part in 3 tough clinical trials. The first 2 clinical trials failed dramatically, which was heartbreaking, not just for me, but my family. Clinical trials are very hard to take part in. You must give your blood, sweat and tears to take part. What I want to tell you about today is the clinical trial that I am on now, Givinostat. The black and white results of the data show the drug is showing the slowing down progression of the disease, but I would like to add some colour to the data by sharing my story with you. I started the treatment when I was 12 years old, an age at which boys with Duchenne typically stop walking. I don't remember much about the start of the clinical trial, other than the drug tasted a bit funny and I went a bit picky about the favourite foods I liked. I did have a platelet count drop, which meant I had to watch for bruises, which I never got. My bloods are now fine and I don't worry about what I eat. I never noticed any major improvements when I started taking the drug, but remarkably the thing was, at 12 I stabilised and seemed to stay the same for years, which was about 4 years. I managed to walk until I was 16 years old. At 16 years old, I went through a difficult time at school, which impacted my mental health. During that time, I accidentally flipped my wheelchair, which had no tippers, which ended up leading to me breaking my shoulder. My bones are weaker because I take steroids, so they break more easily. The impact of the fractures meant I struggled to balance to walk, and I felt safer staying in my chair. Despite a difficult time in year 11, I passed my GCSEs and managed a place at college doing a BTEC level 3 in graphics, which I love. Unlike my friends with Duchenne, I have never had to take a reduced timetable due to tiredness. At college and home, I manage all my bathroom needs. This gives me the dignity and privacy. I can transfer from my bed to my wheelchair myself, which reduces the burden of caring for me, like my parents. They don't have to get up in the night to take me to the toilet. This means they are not as tired and can both hold down a full-time job. I do seem to have good energy levels, and I need them because it takes a lot to manage my condition. I can independently catch the bus from college to home, which gives me a lot of freedom. I do also have plans to my journey, so I don't encounter steps. I'm lucky that I can still self-propel in my manual wheelchair, as it makes it a lot of places and vehicles more accessible than when I'm in my power chair. Being able to get out and about independently means I can get out and mix with other people and make friends and contain friendships. I noticed I don't get sick with bugs like my Duchenne friends, and I've never been hospitalised with illness, which is common for people with Duchenne. I do believe I have stayed well and had the stamina to do well because of Genomics. My greatest passion in life is wheelchair sports. I play power hockey and power football. I do this on top of a full timetable at college. I train 3 times a week and play on the weekends. My wish is to stay on this drug so I can achieve my dreams of representing England in power chair sports. And I want to go to university to study graphics. Through playing power sports, I have made many friends with Duchenne. I have watched them deteriorate over the past 5 years when I've stayed pretty much the same. I've watched as my friends with Duchenne need help having to lift drinks and cut food up. I don't have these problems. It breaks my heart to see them struggling. My friends at power hockey watch me independently transfer while they are hoisted, and it has felt unfair that they haven't been given the same treatment as me. So, well, they need to be able to be given this treatment so they can achieve the same dreams I've been able to achieve. At times, I did feel judged by them because I was the boy that got the drug and they didn't. I'm glad Genomics has been approved by the MHRA for ambulant and non-ambulant boys with no age restrictions in place. And I want my friends to get the same chance that I got almost 5 years ago and given access to Genomics.
So, the Genomics timeline. We were very, very lucky that Jack got onto the clinical trial. He was one of a handful of boys in the UK that got access. So much to our delight, in June 2022, the positive data from Jack's trial came out, which actually for Duchenne, which is a rare disease, was quite a large, placebo-controlled phase 3 clinical trial. We then waited through the whole of 2023 while regulatory filings were happening. And then in March 2024, the FDA approved Genomics in the US, and US patients got access. We then became aware that the company had actually opened a compassionate use scheme in the UK, the Early Access Programme. So we informed our community in November 2024. About 500 boys in the UK are eligible for the EAP. We very, very naively thought this was going to be easy. The company were giving the drug free of charge. It's an oral suspension drug which patients take at home. But it very quickly became clear to us that the rollout was going to be nothing like we hoped for. Complexities not foreseen and barriers to access and issues preventing rollout quickly became apparent, and I'll talk you through the next milestones. One thing we quickly learned though is, as I said in the beginning of my talk, back in 2011 when Jack was diagnosed, I actually believed there was nothing worse than being told your son had a disease for which there was no treatment. I actually learned there's something worse. Your son having a disease, but there is a treatment. It's been made available free of charge and you cannot get access for your son. So some of the barriers we've faced are lack of resource within the NHS and capacity issues. Families went to their trusts and were told it was going to take months longer before they could open the EAP. A lot of them did want to wait for NICE to approve. There was lots of different barriers, but one site said they couldn't do it because they didn't have a fridge to be able to store the medicine. So what we did is we launched the Time is Muscle campaign to drive home the importance of this new medicine. It was being made available free to the NHS, and we needed to share what this meant for families to try and speed up the rollout and help families access it. It had one clear aim: Everyone who could benefit from Givinostat, like Jack's friends, should be given access swiftly. We were lucky that the MHRA approved the drug in December, and actually what that meant for the EAP was it did remove some of the uncertainties from like the drugs and therapeutics committees. It did give clinicians more confidence that it was safe and effective. It also resulted in a more stable version of the drug being produced, so we didn't need the fridges anymore. But the Trust and Health Boards were still hesitant to participate in the early access programme. So our campaign is really focused around meeting with politicians, MPs, ministers, regional mayors like Andy Burnham, Steve Rotheram, the London Assembly, really building a network of support in Parliament. We've had parliamentary debates, parliamentary questions on this issue. We've done lots of different events in Parliament to try and highlight how the EAP exists, but patient can't get access. So here's some of the photos from some of the events. That's actually our MP, Lisa Nandy, who's the Secretary of State for Sports and Social Media. So we had 30 MPs come along to an event, and I believe this man then received a lot of text messages from various MPs saying, you need to come and meet these 3 young men and you need to hear their story. So he came along, he stayed for 40 minutes, and Ben, Eli and Jack were absolutely fantastic ambassadors. I was incredibly proud of Jack for sharing what it would meant to him to receive Givinostat and the unfairness of it not being available to everyone. Eli and Ben shared what it would mean for them and why everyone who could benefit, whether they're ambulant or not, should get access. They highlighted the quality of life benefits and the importance of maintaining arm and lung function. So following the parliamentary event, Wes invited us to the Department of Health, where we've been discussing the challenges with him of the EAP. And I think it's fair to say he wants to support us, but his hands are tied. What's become clear is he has very little power over the individual trusts to open the EAP, but he's trying to see what he can do to help us speed up access. So what we've achieved: national and local media coverage, access now at 9 sites with a further 7 ready to start. What we're trying to do is facilitate best practice sharing and highlight innovative approaches and really meeting with trusts. In March, the first site actually opened, which was in Leicester. It was a small patient cohort, but they thought creatively, rather than making their cohort, which is I think about 13 boys, come in for individual appointments, they got all the boys together in one room, explained Givinostat, how the programme was going to work, and actually did a group consent, so it saved clinic time and the number of hours. And similarly, that happened in Wales, which was the second site to open in April. Interestingly though, access in Scotland has been completely different to the rest of England. Every single child who is eligible now, who wants access in Scotland, will get it, because the Scottish minister was able to intervene there and direct the trusts in Scotland to give access. So if you're a child with Duchenne in Scotland, you get access. But, you know, Jack's friends at Alderhay in Liverpool, they don't get access. So we have built positive, constructive relationships with the NHS, and we've always sought to work with them since the inception of Duchenne UK. We see clinicians and senior leaders as our partners. It really has been difficult to navigate this campaign and wanting to maintain this approach, but also support our community with fighting for access. The lack of collaboration sometimes between different hospitals has been really unhelpful. And what we've tried to do is facilitate data and information sharing between the clinicians to speed up rollout. So Wes came back to us very recently, and although he can't help with the EAP, what he has promised to do is, if NICE approve, he will then use the Innovative Medicines Fund to speed up access for patients. So where we are now, we've got the first NICE committee meeting on the 10th of July. So we'll see what happens there. I think lots of people will know in this room, you rarely get a positive decision at the first meeting. There's usually some negotiations to take place, but we've got our fingers crossed. We've got promises from the government that if it's recommended by NICE, it will have a fast rollout. Access is currently piecemeal with glaring differences between the UK's devolved health systems. There's still zero access for non-ambulant patients, a whole cohort who desperately need access to this, and there's no clear timescales on when they will get access. So what we've learnt so far, the challenges, there's a chronic under-resourcing of specialist neuromuscular clinics, especially in staff. We have a national shortage of neurologists. There's a lack of interest and engagement from hospital leaders unless you apply pressure. We wrote to every single trust who could deliver the AP, 24 trusts, only one CEO replied to our letter. It's only when the media got involved that they then reply. There's an inability of the central government to intervene on compassionate use schemes like the EAP. Fragmentation and lack of cooperation between hospitals. For example, because the boys can have the platelet counts drop, they have to have the bloods monitored. Why can't local hospitals do the blood draws instead of the bloods being done at the neuromuscular centres? But that collaboration is very difficult to achieve. So what we've learnt so far, practicalities. So in Scotland, the patient access scheme could be adopted for the rest of the UK so that all sites can approve it at the same time. Some sites have been able to pool pharmacy resources, which is helpful. Innovation is key, thinking about things like group consents to reduce impact on clinic time, but collaboration is key. When sites have worked together, they've been able to set up quicker. And then finally, areas for future work. We're actually going to write a white paper on what we've learned and make recommendations for the future. Working with the NHS to understand what we could have done differently and what would have helped them roll out the early access schemes in the future. We want to work with regulators and the government to determine how schemes like the Early Access to Medicine scheme could be improved and maybe used more instead of this EAP. How do we ensure that new treatment is fair, equitable and safe for all patients? So, thank you.
Thank you. Thank you both. Remarkable. We have time for some questions. Gentleman at the back.
Thanks, Alex and Jack. Really, very powerful profile story and amazing to see the timeline of what you've achieved. You touched on it, Alex, at the end around the Early Access to Medicine scheme, which we know the Duchenne community has some experience with a few years ago. So what, in your view, needs to change for that scheme to be more attractive to avoid some of the potential problems that you might see within the EAP? Thank you.
So I think actually there's a couple of things. Actually, I think the company should have come and spoke to the UK. They're an Italian company who developed the drug. They should have come and spoke to the patient groups first, because we could have told them more about the Early Access to Medicine scheme, which would have encouraged its use over the EAP. I think the problem for the company was, if you look at the timeline, they opened the EAP in November and they got the MHRA approval in December, and then the NICE process began. And what we've been told is that they couldn't then continue to enrol patients through that period, but if they had the EAP, they've been allowed to continue rolling patients. So I think if EAMS could change to allow patients to enrol in the scheme until it's actually approved by NICE, I think that would be helpful.
That does exist now in the new legislation, is what's called the winding down period. So that was put in the new legislation specifically to avoid that. So you would have this kind of gap where you have maybe not a NICE opinion, but you had a marketing authorization. In the past, you'd have had to close the scientific opinion. But now the MHRA has the power to keep that scientific opinion open until a much later point. So just to know that does now exist.
Well, that's brilliant news, because that's something we wanted to call for, so it's good to hear that.
If I can ask a question, one of the things that sometimes concerns us as trialists, people running the trials, is when we're talking to young people, making sure that we explain what the pros and cons of the process is. Would you like to comment what's the best way for us to do that? I mean, clearly you've been through a number of trials. How's the best way to talk to younger patients about making sure they understand what's involved in that process?
I feel like it's important to explain to them and also keep an eye on them through the clinical trial, and also being able to talk to them, you know, about what the side effects are and how it is going to affect you, you know, I think.
And we shouldn't worry too much that that's a bit scary sometimes, because I think that's what we do, you know, we don't want to burden people unnecessarily.
Yeah, sometimes it can be scary, but I feel like there's a way to do it instead of using like more words, using like maybe like a presentation or something, or using, you know, pictures and stuff to try and explain it that way. I feel like that is a more friendly way, you know, for younger boys to understand it when they started the trial, because I was only 12 when I started the trial, and I feel like it on a piece of paper with writing, it is quite a lot to take in. So I feel like if it can be done with picture diagrams and maybe a little less writing, and maybe actually having it in person explained to you, know what I mean, if that makes sense.
A bit more work in it from our side.
I think it is scary for patients, though. We're having the first gene therapy trials for Duchenne, and sadly we've had 2 boys die in a gene therapy clinical trial. And I've often, as a parent, thought now, you know, Jack's 17, if he tried to enrol on a gene therapy trial, those conversations that we're going to have would be very, very, very difficult.
Yeah. Thank you.
Is he receiving immunostat?
Yes. Yeah. He started quite late at 12. The label's from 6. You know, we wish the treatment was available when he was 6, and he'd started during the
assessment about improvement, say before starting and after a few months of its use.
I think this drug slows the progression of the disease down. It's not a miracle.
Is there any question there are some tests we can define for this improvement?
I think there's work we can do on clinical endpoints to try.
I think the trials to date have been really quite definitive, and we've seen the MHRA approval. So you know, we're in a good space there at least. Sometimes we think when we've done that, that's the job over. Clearly, you've shown us today that's not the case. Thank you very much. Appreciate you have to. Thank you. Thank you very much. Really appreciate it. So now we will move over, and I'm pleased to invite Professor Rinaldi, who is the Professor of Molecular and Translational Neuroscience at the University of Oxford, working at the John Radcliffe Hospital. He's going to speak to us about unlocking potential, a scalable framework for genetic therapies in rare neurological disorders. Thank you very much.
Yeah, thanks very much. First of all, I should say thanks very much for the kind invitation to this wonderful meeting, and thanks for sharing those fantastic experiences. It's going to be difficult to switch gears a little bit, but I will try. So I'm Carlo Rinaldi. I'm a clinician scientist at the University of Oxford, and in the next few minutes, I will try to tell you a little bit what are the activities in Oxford and beyond on scaling up therapy development for rare diseases, particularly rare neurological diseases. I'll give a bit of background about myself. I'm Italian originally. I did a PhD in United States. I'm based in this building, which is the newest building so far at the University of Oxford, which is called the IDRM, or Institute of Developmental and Regenerative Medicine, that is structurally and thematically divided into these three themes of neurology, cardiology, and immunology, with the overarching goal of trying to tackle diseases in these areas. In the lab, which is we call a molecular and translational neuroscience lab, quite friendly, we call it the R lab. We use a number of tools, including stem cells that we derive from patients, stem cells including motor neurons, cortical neurons, muscle cells. We use advanced microscopy, more and more genomic data to inform our hypothesis. And as therapeutic approaches, we really are interested in exploring genetic therapies, particularly developing better and safer next-generation antisense oligonucleotides and CRISPR gene editing approaches. We're deeply passionate about therapy development, where as a clinician, this circle of bed to bench site is what fuels our research, my research, the possibility of going back to the lab to answer the questions that a patient poses us in the clinic, and then bring back those answers potentially, or even more questions back to the patients, with the general goal or trying to achieve impact and achieve implementation through practice for our approaches. So historically, we've been invested particularly in this area of the circle, which is the identification of molecular targets, understanding what is it that we need to target in rare diseases. Again, the goal is to try to more and more transition into clinical trial phase. So since I'm a clinician by training, I would like to start, let's say, with a clinical case. And this is a real case, is a work that is currently ongoing, very much unpublished work in a way. So this is the case of a woman, 19 years old, that has always had a lack of sensitivity for pain, both noxious stimuli and inflammation and temperature. With self-mutilating injuries of the fingertips. And you can see this is an image not of the actual patient, but taken from gene reviews, mutilation of the mouth, and bone deformities as a result of past fractures, and anosmia, which is inability to detect smell and odor. This patient was originally seen in the clinic of Professor David Bennett at the University of Oxford, and with a combined effort with also Professor Jenny Taylor, we came up with the actual solution from a genetic standpoint, why this patient was having these symptoms. So this patient is actually compound heterozygote for mutation in this gene, SCN9A, which is a gene that is known to be associated with a congenital pain insensitivity syndrome. And what's interesting is that this patient has two mutations, one that is a well-known inactivating mutation, and the second one, which is a deep intronic mutation between exon 24 and exon 25 that effectively shifts the reading frame and allows for the production of a cryptic exon and eventually a stop of the premature stop of the mRNA, so stop of translation. So effectively, this is a very, very clear cause of the disease of a loss of function. So the patient basically has symptoms because there is no production of this protein, which is a critical channel in neurons and neuronal communication. So David Bennett approached me and simply asked me, can we develop a therapy, a genetic therapy for this disease? And the answer is yes. This is work that actually we performed in only 2 weeks. We have the luck of having in the lab an oligonucleotide synthetizer. So we screened very few antisense oligonucleotides for basically sequences that were able to splice switch this cryptic exon, because we know that this technology allows for basically splice switching quite well. And of the sequences that we tested, at least in patient fibroblasts, at least 5 were very, very effectively in reducing the production of this cryptic exon and therefore allowing the translation of this protein. So there is a medical student that is currently working on this project, and every day he asks me, how can we make this into a product? And he very well understands, as everybody else in the lab, that we have in the hand potentially a product that could actually make a difference for this patient. So this is the question that we ask ourselves in the lab all the time. So the technology is there. The science is actually fairly mature in the space of antisense oligonucleotide in this case and genetic therapy in general. How can we scale up this type of approaches? And this is just a survival curve from a very seminal paper published in the Lancet journal in 2016 by Richard Finkel and his group, basically showing the remarkable effect of another splice-switching antisense oligonucleotide, which is nusinersen, that was able to effectively change the course of a disease. This is a very, very good example in the field of neurology and neuromuscular diseases in the context of SMA, which is another devastating neuromuscular disease, spinal muscular atrophy. Also in this case, as I was saying before, this is a splice-switching antisense oligonucleotide. And this work paved the way for a number of other clinical trials and eventually the approval of nusinersen in 2018, which again was a seminal approval, which has really changed the course of this disease. And more there is data that is published out there. This is just the most recent dropped in bioRxiv in 2025, just a few weeks ago, showing the pharmacokinetics and pharmacodynamics of antisense oligonucleotides delivered via intrathecal administration in non-human primates. And as you can see, the level of reduction of MALAT1, which is the target of this antisense oligonucleotide, is pretty much reduced in many areas of the brain, basically showing that we can actually achieve what we want to achieve with ASOs quite well. There is certain areas of the brain which are the deep nuclei, globus pallidus, putamen, where the effect, as expected, is not as dramatic. And this correlates quite well with the amount of oligos that we can detect, in fact, in these neurons and these neuronal groups. And this is also another paper in Science Translational Medicine in early this year in 2025, showing that we have developed as a scientific community, this is not work done in my lab, a tracer, a PET tracer that allows to basically identify and find where this antisense oligonucleotide goes. And this is, again, non-human primate data after intrathecal administration, effectively showing that after 7 days, you can still see signal of ASOs in these neurons and neuronal population. So the challenges that were set up to try to give an answer to, in a way, I've tried to divide them into these three big questions. How to identify systematically patients amenable for nucleic acid therapy intervention, how to platformize the development of effective nucleic acid therapy, and how to safely treat patients with nucleic acid therapies at scale. And as you can see, the common theme among all these elements is the systematic, the ability to do this at scale, because we can actually do this right now for single patients, but how we can do it as a society, let's say, for all patients that are amenable for this sort of approaches. So I'll start with the first question. And as an immigrant in this country, I probably am allowed to say that this is probably the best country to do this sort of research. We really have an outstanding pool of genetic information through the 100,000 genomes and a centralized clinical systems such as the NHS. And on this basis, we were lucky enough to be funded the first of the MRC Therapeutic Center of Excellence, which is called the Therapeutic Genomics, which is, has a number of stakeholders and, of course, patients, institutions, University of Oxford leading it under the, in fact, the leadership of Professor Sanders, which really has this shared vision of producing a genome-led, patient-centered, data-driven vision, again, to bring genome-targeted therapies to the clinic, again, at scale. So this, I'll give you just a simple example of what this means when we're trying to do that. And this is a long program of over 14 years. So we're focused particularly on antisense oligonucleotides and CRISPR editing. We call this genetic therapies. And I'll give you an example of the programmability of this sort of approaches, how we can learn from certain features of this type of therapies and apply those features a bit more globally. So this is the case of transthyretin amyloidosis, where basically, effectively, a vector was used. Inside the vector, there was the Cas9 protein and a guide RNA that would bring the Cas protein into a specific target to allow for a specific gene editing. And effectively, exactly the same approach was used for another clinical trials in hereditary angioedema, which is another neurological and multisystemic condition, where basically all the components of the genetic approaches have been remain the same. The only difference here is the guide sequence. And there is element of programmability is very common in this sort of genetic diseases. So in the course of this next 14 or 20 years, that's what we're trying to do, relying on a lot on the genetic approaches and data that we have, is how do we identify the most tractable targets, the ones that are easy to treat. And I'm thinking, for example, of the patient that I showed you before, which is one of those low-hanging fruit in a way. How do we treat this disorder at scale? How we deliver therapies to complex tissues? We're going to focus initially on brain, blood, and retina. How do we share the data? This is very much about a data problem. We want to be able to share data and produce this iterative learning where we learn consistently and constantly about from what we're doing, and how we build, of course, a diverse workforce that is able to deliver this vision. And I'll give you an example of what we do actually in my lab on trying to platformize the development of effective nucleic acid therapies. How can we make sure that quickly we can generate the best oligonucleotide sequence for a given disease? And this is work that is currently in bioRxiv in second rounds of revision. We basically used and identified a new class of Cas13 proteins, which are proteins that bind effectively the DNA, which is Cas13b. We call this new Cas a CRISPR lock that is able to effectively bind to a specific mRNA and function as a steric blocker. So it basically doesn't allow other RNA binding proteins to bind to that specific RNA. We validated the system in a model of Duchenne muscular dystrophy, which is a severe, again, neuromuscular condition caused by a CTG repeat expansion in the 3' UTR of the DMPK gene. And effectively, what we did is that we genetically encoded the Cas13b inside cells of patients, human myoblasts, effectively having this specific mutation. And the mechanism of the mutation in this case is very well known. So the element that is important here is that we need to understand the mechanism of the disease in order, back to the point, in order to be able to develop an effective treatment. And the way this system works is that basically Cas13 decorates these loops of RNA and prevents the sequestration of specific binding proteins that actually are what cause the disease. So we validated this system in the context of myotonic dystrophy. This is just to show that basically the spliceopathy, which is typical of this disease, is completely corrected, almost completely corrected when using a guide for Cas13b that is only 21 nucleotide long. The reason why I'm saying this is because we can use now this new technology, if you want, CRISPR lock, to interrogate at scale, again, all the functional elements of RNAs. And we can basically do that in a single experiment. And what we're trying to do right now is that we have identified 600 haploinsufficient disease genes. These are these are mutations that cause a disease in humans that are caused genetically by a heterozygote loss of function mutation in one of the two alleles, where the other allele is insufficient in a way to compensate for the loss of function and the lack of activity of this allele here. So the idea here is to use CRISPR lock to basically identify the best possible sequence in that mRNA that allows for an upregulation of this wild-type allele, and we hope that this can be translated then into a sequence against which we can design an antisense oligonucleotide. And again, we can do this at scale for over 600 haploinsufficient genes. This is work that is currently being done by a brilliant student in my lab, Salma Ali, and the readout will be a single-cell RNA sequencing output. And the advantage of this approach is, again, as I was saying before, is that you can do this effectively in a single experiment. And in one experiment, you know which is the best sequence that you can use to then eventually design an antisense oligonucleotide for the approach that you want. Then down to the last question, which is probably the most difficult one. How do we safely treat patients with nucleic acid therapies at scale again? And I decided to tell you, I could have told you many stories, but I've decided to focus on this activity right here that I'm very much involved in, which is the UK platform from nucleic acid therapies. This is one of 11 nodes in the rare disease space that have been funded by the MRC and the government about a couple of years back, some of which are disease-specific, some of which are cross-cutting. Our node is a cross-cutting node. And the idea basically of this node is to provide exactly that, a roadmap for rapid bench-to-bedside translation of nucleic acid therapy, starting with antisense oligonucleotides for rare diseases. Our aim is quite broad. We want to establish a cross-disciplinary network to promote knowledge exchange, target selection, preclinical development, down to the clinical translation. I'll give you a bit of a flavor of the type of activities that we're trying to do. So for work package 1, that is led by Mina Wright and Annalise Davidson, is about target selection and preclinical development. We're trying to develop and generate guidelines for selection of patients. We want to be able to identify those low-hanging fruit, those cases, again, at scale in all the genetics clinics across the country. So there are guidelines that are currently being implemented, produced, and will be published soon. The second work package is led by Nick Clench and Hayan Zoon, which is to test the UK capability in nucleic acid therapy development for, again, rare diseases. So basically, the idea is that we will take one exemplary study. This is going to be done in collaboration with NATA, which is an institute that, again, works on the development of nucleic acid therapy, and understand what are the requirements for a scale-up synthesis, the manufacturing, the toxicology, what it takes to get basically an antisense into the clinic. And then the last one that I will probably tell you a little bit more about it, this is the work package 3 on clinical trial and regulatory approval. This is work that I'm doing together with Professor Muntoni, is to basically build a multidisciplinary forum to exchange information on nucleic acid therapy clinical trials and the regulatory approval process between the key stakeholders. And the idea here is to work with people like you in this audience, patient communities, the MHRA, of course, the RTLP that I will tell you a little bit about it later, to understand, again, what are the clinical requirements for the advancement of such therapies into the clinic. And one element that we're very much focusing on is the concept of safety. So we want to make sure that whatever antisense oligonucleotide or nucleic acid therapy we bring into the clinic as a scientific community, we want to make sure that these are safe drugs, safe medications. This is very, again, preliminary. I'm just to give you an idea of the type of thinking that is behind. Again, this is work done by a brilliant student and another brilliant student, I have to say, in my lab, which is Barney Hill. So basically, he's using now a machine learning approach to extract all the information of antisense oligonucleotide for patents published since 2005. So this is an extremely powerful approach. And this is just an histogram to show you that basically since 2021, there's been an absolute explosion of patents for antisense oligonucleotides, which is interesting. So we are investigating and trying to understand more about this enormous pool of information. One thing that I can tell you, for example, which is something that was absolutely unexpected, we found that actually when an ASO is designed to target the 3' UTR of a gene, usually is more effective. And this is something that we didn't know before. And this tells you the idea of the power of this type of approach. And what we aim to do, and this is hopefully will be published very soon, is to generate a web tool, which we call it OligoScan1, that will allow any investigator in the UK and in the world to input their gene and have the best possible ASO sequence for inhibition of that specific target. Why am I telling you this? I'm telling you this because basically we're using exactly the same approach to extract all the information on safety data for nucleic acid therapy that is currently available in the world. And this is something incredibly difficult because much of this data is either hidden in companies or hidden in academic labs. Sometimes it's released by documents, by the FDA, the MHRA, but they're very difficult to extract this information. So we're using a machine learning approach to try to do that. We want to be comfortable as a scientific community that these drugs are indeed a safe drug. And I would like to end by mentioning, of course, the work of the RTLP, or Rare Therapies Launchpad. Daniel O'Connor is here in the room, so I won't say too much about it. This is established in the UK as a model to model a scalable and sustainable system to meet effectively this new area of individualized medicine. And the goal here is to put in place an innovative, a new way, regulatory way to reimburse end-to-end this type of treatments in the clinic. And this is an approach that very much relies effectively by this gained experience that allows the transition effectively from a research activity right here to an actual treatment in what are called accredited centers. And I won't dwell too much in this because Daniel again is here. I'm sure he can answer all the questions about this. And I would like to end with this picture. This is the picture of baby KJ. This was published earlier this year in 2025. This is the first kid to receive a gene editing approach, a base editor, treated 3 times with a base editor to effectively correct a mutation that is causing a very severe neurometabolic condition. And this is a kid that, against all odds, is doing well and is currently thriving. And this is to say that this is the present. This is not the future. This is what's happening already. Science is not the limiting factor anymore. We need to understand and make sure that the process follows through well. And with this, I would like to end here, of course, thanking everybody in the lab, our lab, Therapeutic Genomics, and of course, all the other partners involved that are listed here. I'm happy to take any questions. Thank you.
Thank you. We've got time for some questions now. So I wonder if I could start off by saying, I mean, clearly this has tremendous application within rare disease, but is this also to some extent, a novel discovery forum for new targets for more conventional drug therapy?
Absolutely, yes. So the idea is that as long as the mechanism is an obvious mechanism, this could be applied to any sort of new targets or new gene that comes to the attention. Absolutely, yes.
It just seems to be a very tractable way to get in there and do something a lot quicker than we do on the other side of the fence.
Absolutely right.
Munir, there a microphone, please.
It's a great talk. Thank you very much. So just to understand the safety aspects, really. Obviously, there are some safety aspects which we've already reported, certain SOs, etc. So how much mechanistic work has been undertaken to understand why platelets go down, or, you know, the sort of hydrocephalus occurs and so on and so on? Because it seems to me that it's going to be difficult to collect the safety data because of the rare nature of the disease. It's going to take many years, but we need to understand mechanisms of those cases which have occurred to help us to be able to predict for the future.
Yeah. That's an excellent question. Some of those information is available and we can track it, but what we are understanding, and the more we treat patients, and I'm thinking of these N01 trials, for example, from Team U, etc., is that actually some of these safety aspects cannot be replicated in the models that are being used to get the drug up to that stage. So lots of non-human primates data even, we don't quite see the same odd cases that we see in the clinic. And there's probably a number of reasons for this. Certainly lack of translation of the models is certainly one. Another component is probably the fact that treating patients that have already a steady inflammation state, let's say, compared to non-human primates that are unaffected, let's say, might also play a role. But this is an active area of investigation. And this is exactly why we want to extract this information. We're trying to talk with pharmaceutical companies to make sure that they release this information. I understand that it's difficult for pharmaceutical companies to release this information, but at least they can do it in an anonymized way so that we can learn from all the clinical trials work that have been done. And this is exactly the work that we're trying to do to systematize this sort of safety profile.
Thank you.
Just behind.
Thank you. It's June Rain, formerly MHRA. Really exciting work, right up to date. A couple of thoughts. Could you say more about what your plans are for the 19-year-old you talked about at the beginning, where you're making a product? And then secondly, relevant to the regulatory piece, the editorial on progress in N=1 trials now recommends the elements that could be an accredited platform. What do you think of that? Do you think this is becoming a real proposal?
Regarding the first question, that's a difficult question. It's difficult, I have to say, and this is the first time in my career that you're working in a lab trying to optimize, let's say, further optimize a product, a molecule that could very potentially be something that has an impact for the life of a patient that comes to the clinic next door. So there is a moral, I feel more and more a moral obligation to having to advance this therapy. And I have to say the ecosystem around me allows me to do that. You know, we could potentially think of doing that. So effectively, what we're trying to do at this point is to de-risk this sort of molecules, to talk specifically about this molecule in the best possible way, so that whoever manufacturer comes along and decides to take this forward, could be the RTLP as opposed to any other, company, we could try we could treat the patient exactly the way Team U has treated its patients as an N of 1. So this is very much work that is in progress and ongoing. With regards to the accredited centers, I really do think that this is probably a good way to move forward. We're not talking about rare diseases. I mean, rare diseases are an incredible large family of diseases, but among those rare diseases, there are those that are extremely amenable for this type of approaches. And the case that I presented here of this woman of 19 years old represent that exact case. We don't have to do much work on the biology side of things. This is a splice switching oligo that can be treated, that can be used. The likelihood of finding a patient with the same mutation, a cryptic exon, where we found a cryptic exon, are very, very low. We have looked around in the 100,000 genomes in the world. This is probably really an N of 1, if anything. There's probably, it's unfathomable that we would be able to do a standard clinical trials in this case. Probably the approach of the RTLP to move this into a treatment in an accredited center where we share the data, we learn iteratively the process, so that for the next one we can be better and the risk even more, I think is the right way. For this group of diseases, probably the right thing to do.
Thank you very much. I'd like to draw the session to an end now. I'd remind you there's coffee. Please be back for session 4, 20 to 4. I'd like to finish by thanking all the speakers. Thank you. Thank you. Thank you.
Good afternoon, everyone. Thank you for coming back, and great to see you for this final session where we'll continue talking about some of the really exciting innovations in advanced therapeutics. So I'm very pleased to introduce our first speaker in this session. It's Professor Bobby Gaspar, who is the Honorary Professor of Pediatrics and Immunology at the UCL Great Ormond Street Institute of Child Health. He is a pioneer in gene therapy, and in particular in exploring the evolution of hematopoietic stem cell gene therapy technology. So Bobby has conducted some of the first studies in patients with severe primary immune deficiencies, and has also conducted the first studies in patients and also late-stage clinical trials. And Bobby is also the co-founder and chief executive officer of Orchard Therapeutics. And today he's going to talk about gene therapy for rare diseases.
Thank you very much, and it's a real privilege to be here and talk to you all about some of the work that we've been doing in this area. And thank you for the kind introduction. And just say, I was a physician and at Great Ormond Street Hospital for 25 years, working with children with severe genetic diseases and looking at ways of treating them using gene therapy. And so I had a dual career at UCL and at Great Ormond Street. And then the results of those clinical trials led us to form Orchard Therapeutics to try and take these forward to become standardized medicines that could be used for patients worldwide. And so I've been running the company as CEO. So we've heard about different types of gene therapy, but this, and I just like to use the kind of phrase that gene therapy comes in lots of different flavors, or there's lots of different types of gene therapy, and they're not all the same. So where I have focused, and the work that I will talk about, is hematopoietic stem cell gene therapy, which has the opportunity to address some specific disorders. Not all genetic diseases, but some specific genetic disorders. And I think it's very important to understand that. And so what we are doing is taking an individual's own blood stem cells, where there is a faulty gene or a defective gene, and introducing into those cells, into those patient cells, ex vivo, so outside of the body, a working copy of the gene. And we use a lentiviral vector in order to introduce that working copy of the gene, and there are reasons for that. And one is that the lentiviral vector integrates the new genetic information into the DNA of those host stem cells. Once it's integrated, that means with every cell division, that new genetic information is passed on. So that gives a durability to the therapy. The other really important thing is these hematopoietic stem cells, when they're returned to the patient or when they engraft in the patient, are capable of self-renewal. So you create a pool within the individual of self-renewing, gene-modified hematopoietic stem cells, which means that they are potentially there for the lifetime of the individual. So with one administration, you can provide that new genetic information potentially for the lifetime of that individual, and hence potentially a cure. I'll just say that the first patients that we treated at Great Ormond Street were treated in 2001 with a single administration of gene-modified cells, and that child has a functional immune system now, and on social media you can see him living essentially a normal life. These cells, because they're hematopoietic stem cells, have the opportunity, or have the ability, to become multiple different cells of the bloodstream. So by correcting these cells, you're able to correct these cells in the periphery and therefore have the opportunity to correct multiple different disorders. So by correcting immune cells, we can correct immune deficiencies. By correcting red blood cells, we can correct hemoglobinopathies. But the other area that is really very important, has come to light through the work that has been done, is that these cells, monocytes and macrophages, have a natural ability to migrate into different cell types. So monocytes and macrophages can migrate to the brain, where they become microglia, and therefore gives us an opportunity to deliver genes to the CNS. And I'll show you examples of that in just a second. By migrating to the bone, they can become osteoclasts. Osteoclasts are naturally derived from the macrophage lineage, and therefore we can remodel bone. And I'll show you examples of how that has been translated in the clinical setting. But there are opportunities for migration to the liver, and we have programs where we're using this to correct a specific form of Crohn's disease through the migration of these cells to become gene-modified tissue-resident macrophages. Now, this is all theoretical, but actually this has been translated into clinical practice. And what you see here is that there have been clinical proof of concept and multiple publications showing the use of HSC gene therapy for correction of neurometabolic conditions, leukodystrophies, mucopolysaccharidoses, and other lysosomal storage diseases, for correction of immune disorders, severe combined immunodeficiency, Wiskott-Ulrich syndrome, chronic granulomatous disease, and also for correction of red blood cell disorders as well. And with respect to our conversation today, some of these have also become licensed medicines in the EMA and in the FDA. And these are some of the first approved genetic medicines. And how do we do this? This is just a process by which this is done. We're able, through apheresis, to collect blood cells. We're able to purify them, the stem cell population, from that apheresis population. And ex vivo, these cells are cultured with lentiviral vectors carrying that genetic information, and now you have gene-modified cells. What we've evolved to is a commercial process whereby these cells that are modified can be cryopreserved, and therefore quality control can be done around those cells. They can then be released, and those cells, having been cryopreserved, can then be shipped back to the patient site where they can be infused into the patient. In all of the conditions treated so far, some form of conditioning has to be given to the patient in order to make room for the incoming gene-modified cells. What I've talked about, and where a lot you may have heard already, is the science of this, some of the clinical trials. But in order to make a medicine, there are a number of challenges that we have to go through. And what I want to talk about is not just the clinical information or the science behind this, but also the path that we've taken to take this from extraordinary clinical data into medicines that are now being used by patients across Europe, across the US, and in the Middle East as well. And so it's about not only, say, making the clinical, getting approval, but also taking it to access for patients. And so there's a number of things. So there's manufacturing and regulatory approvals, identifying patients, especially in the rare disease setting, having skilled treatment facilities, and also ensuring that there is access of patients through reimbursement as well. So there's a whole ecosystem that needs to be addressed in order to get access for patients with these devastating conditions. And I will just focus on a couple of conditions, but this is metachromatic leukodystrophy, and I'll use this as a case in point to illustrate some of those challenges that I've talked about. This is a condition that is really very devastating. Until very recently, there was no treatment available for this condition. It's a single gene disorder that leads to the defect in the lysosomal enzyme. The lack of that enzyme leads to the buildup of substrates called sulphatides, which are particularly damaging to peripheral and central nerves. These children are born normally, but after a few years, they lose the ability to walk, the ability to talk, the ability to interact with the family, and it's essentially palliative and supportive care until all children uniformly will die in either the first or the second decade of life. There is no treatment available for this condition. This poor child was, you can see her at the age of 5, prior to diagnosis, living essentially a normal life, and this is her 4 years later when she is moribund. And unfortunately, since these pictures were taken, this child has died. This is the defect. This is aryl sulfatase A is the enzyme that is missing, the gene codes for this enzyme. The lack of that enzyme leads to the buildup of these sulfatides, and it leads to central and peripheral neuronal degeneration. The disease is classified into an early onset form of the disease and late onset, early onset being symptoms starting before 7 years of age, and further subdivided into an infantile form and a juvenile form. And again, the difference between the two is the time at which the disease symptoms start to manifest. And typically, the children with the most severe form of the condition, where symptoms start before 2.5 years of age, have null mutation. So there is no enzymatic activity being produced. There, in the juvenile form, there is some residual activity, and so you can see that the level of enzymatic activity dictates the clinical manifestations, as is the case in a number of these kinds of conditions. This early onset grouping of infantile and early juvenile form about 80% of all forms of metachromatic leukodystrophy, and that is where the medicine has been developed for. And how does a hematopoietic stem cell therapy allow the correction of essentially a neurological disease? And it comes back to the biology of these hematopoietic cells, and especially cells of the monocyte macrophage lineage that can actually have license to migrate across the blood-brain barrier. And we know that neurological diseases have been very difficult to address because of the blood-brain barrier, which is designed to repel large proteins, viruses, and all kinds of other invaders as such, but not your own cells. These cells have a natural license to migrate across the blood-brain barrier. They distribute throughout the brain. They become microglial type cells, and this has been shown in animal models that they take on the characteristics of microglia. And now, having been fitted with that new gene, they can express that lysosomal enzyme, and there is a mechanism by which the enzyme product that is produced by these cells can be naturally taken up by those neurons. So through using the mannose-6 receptor phosphate pathway, this enzyme can be taken up by these neurons. These neurons now have the enzyme that was previously lacking and can prevent the buildup of sulfatides and prevent neuronal degeneration. Part of that mechanism that I've talked about tells you that once a neuron has started to degenerate, it is very difficult to reverse that process. So the important thing here is to identify patients early so you can preserve neuronal function. Also, through family screening, we can identify patients before symptoms have started, and therefore, again, this is where the best outcomes are, when you start to treat before the onset of symptoms. More of that later. The path to creating this medicine started in 2004 with the first publication of a mouse model that could be corrected using hematopoietic stem cell gene therapy, and this was started in Milan by Luigi Naldini and Alessandra Biffi. And this used that for the first time, the concept that hematopoietic stem cells could correct a neurological disease. This was then moved into clinical trials. The program was initially taken on by GSK and then taken on by Orchard in 2018 for further development. These are some of the data that have been previously published and led to the approval of the condition in the EMA and subsequently in the US. This is just looking at the level of enzymatic activity in peripheral blood mononuclear cells. The gray shaded area is just the normal range. These individuals start off with virtually no or limited levels of activity, but they have supraphysiological levels of activity following the single administration of gene-modified cells. And the follow-up here is beyond 8 years. The first children to be treated on this trial are now 15 years after gene therapy, and still, having had a single administration, continue to have elevated levels of RSAT activity. The activity in the CNS is also in the normal range and is sustained over time. More importantly is of course, the clinical data. And so what we're looking at here, and this, I've just shown the data here on the late infantile form, and especially in patients who were treated before symptoms started, and these were all identified as a result of having an older sibling who had severe manifestations. So these children were screened as a younger sibling and then treated. And this is compared with those children who are not able to receive any therapy, and these are children who often were the older siblings. And so we've used we constructed a severe motor impairment-free survival, which looked at, and this was classed as the interval from birth to the first occurrence, or when they were able to, when they passed the point at which they were able to sit unsupported, or to death. And so you can see that the natural history cohort, untreated population, by the age of 4.5, all of those children have either died or have lost the ability to sit unsupported. For the children that were treated, apart from one, all are alive and continue to have a preserved motor function. And this is data now goes on beyond 15 years. This is looking at cognitive performance. So again, if we look at this graph here, this is chronological age, and this is the performance age equivalence. And very simply, if you have a chronological age of 4, you should have a performance age of 4. So the natural trajectory here is this gray, is this dotted line and the gray shaded area. Again, you can see that the children in the natural history cohort have all lost that learning ability very soon after life and have very, very low scores. The children who have been treated continue to have normal performance. The vast majority continue to have normal performance, even many years after the gene therapy, and again, after a single administration of the gene-modified cells. But as always, pictures say an awful lot more than the graphs. And so this is children within the same family. This is the little girl that I showed you at the beginning, and this is her 3 years after the onset of the disease, and you can see how severe that is. She is no longer able to sit unsupported. She has to be fed. She has seizures, and as I say, she unfortunately passed away. Her brother, when she was diagnosed, her brother was much younger. He did not have any symptoms, but he had exactly the same mutation. He was treated on the clinical trial, and this is him 8 years after gene therapy and 12 years of age. And this is him showing the motor function, and as you can see, the difference that he between the two is obviously very dramatic. And and again, there are pictures of him now. He's a teenager on social media. He plays with his friends. He goes to school. He lives essentially a normal life. This is more than a decade after the single administration of the gene-modified cells. This was approved by the EMA in 2020 and was really greeted with huge excitement by the MLD community because they finally had something for this patient population. The timeline to getting FDA approval was more arduous because the initial conversations with the FDA had been more limited. We had to rework the endpoints, we had to rework the clinical data, and through many, many interactions with the FDA, we secured approval in 2024. It's called LENMELD in the US and LIBMELD in the EMA. It also has MHRA approval. But as I said to you before, just getting the approval is just the start of a process of making this available for patients on a global basis. There are a number of things that we needed to do. One was, how do we achieve global distribution? How do we provide access for patients in specialist centers? How do we get reimbursement for patients through the various different jurisdictions? And also, how do we find those patients early enough so they can benefit from the therapy? So there are a number of things that we had to do. And one of the first concepts that we had when we founded the company was it should be the cells that travel rather than the patients. When I was at Great Ormond Street, we had patients coming to Great Ormond Street from all over Europe. We had a child from Australia come to access therapy, and that is not sustainable in the longer term when you want to provide this for large numbers of patients. So we came up with the concept that patients should go to treatment centers, cells would be taken, cells would be shipped to a central manufacturing site, which would be able to modify the cells and then do the quality control around their cells, and then the product could be shipped back to the site in order to be able to be reinfused into patients. And one important part of that is that the cells need to be cryopreserved after transduction in order then to have the time span to be then shipped back to the treatment site where they could be thawed at the bedside and reinfused into patients. And so that model has now been established. So we now have a single a manufacturing site in Milan, in Italy. There are qualified treatment centers around Europe. There are 6 of those now in the UK, in Manchester, in Paris, in Tübingen, in Germany, in Lund, in Sweden, and in Milan as well. And also, we're recently qualifying a site in Spain. There are now sites following the approval that have been qualified in the US as well, who are now treating patients, so in Minnesota. And we've shown that you can ship cells from the US to a site in Milan, manufacture, and send them back without any loss of quality of those cells. So this is now, I say, available for patients both in the US, in the EU, and also we've treated patients in the Middle East as well. Getting reimbursement is also a challenge as well. And so we've had to go through numerous different processes in the EU because of the different approval processes for reimbursement in different countries. But now there is access for patients through agreements with the different agencies as in the UK and all of these different countries in Europe as well. There is reimbursed early access in France, and also there's this cross-border pathway as well within Europe, where patients who are identified in a country where there is no specific reimbursement agreement in place can travel to another country where there is access in order to be able to be treated. And then we've used that pathway for patients from Central Europe and other areas as well. In addition to that, there are treatment abroad pathways, and patients from Saudi Arabia have traveled to Europe in order to be able to access therapy. So this is now genuinely becoming a global therapy for patients. There are obviously other areas of the world that we plan to expand into, Japan being the next country, but also for LATAM countries and also into the Asia-Pacific area as well. The other really important part of this is the ability to identify patients prior to the onset of symptoms. The sad thing is that we identify many patients. The therapy is now available. Physicians are identifying patients of having metachromatic leukodystrophy, but sadly, it's too late for most of these patients. The label is for patients who still have normal cognitive function and who are still able to walk independently. And because of the severe onset and the rapid progression of the disease, many patients are beyond that level of function and therefore cannot access therapy. In some cases, there are younger siblings, as I talked about, where we're able to make that diagnosis and offer the treatment. And in some cases, physicians have been able, through disease awareness, to identify patients and make the diagnosis, and so we're able to treat them in the very early symptomatic phase. But the only real way to be able to offer this to all patients is through newborn screening. This is a condition that fits all the requirements for newborn screening. There is we can identify the patients. We know what the pathway is. There is a treatment now that is approved and able to offer significant outcomes. And so we have invested in bringing the stakeholder network together and have now started pilots in different parts of Europe and also in the US as well. And so far, having screened, and now this data to update is over 500,000 babies that have been screened in these pilots. 8 individuals have now been identified, and 7 have either received treatment or will receive treatment very soon. For these individuals and for their families, they will never know what MLD is like because they won't see the symptoms, and they will go ahead and have, hopefully, a potentially normal life. So this is a huge amount of effort, but this is a model of how, using our abilities to make diagnoses and these innovative treatments, we can change the landscape of a previously completely devastating condition. This path in MLD has allowed us to think about what we can do for other severe conditions as well, especially in the lysosomal storage disease space. We could use the same synergies in terms of regulatory, supply chain, manufacturing infrastructure for the same treatment sites. We can use the same treatment sites and referral networks to treat these patients as well. So MLD paves the way for MPS IH, which is Hurler syndrome, which I'll talk about in just a second, but also similarly for other mucopolysaccharidoses, such as MPS IIIA as well. The learnings from this potentially allows us to go beyond that to other lysosomal storage diseases, but also by delivering to the CNS for rare diseases, can we deliver to the CNS for larger forms of neurodegeneration as well? And so we have a program in a specific genetic form of frontotemporal dementia that's shown proof of concept in the preclinical setting. In Hurler syndrome, this is again a rare lysosomal storage disease, and here you get not only the major problems, there are many problems, but the major problems are in neurocognitive function, but also in skeletal function as well. These patients have a severe skeletal dysplasia, as well as, I say, many other problems. And you can see some of the deformities here in children who have not had any treatment. There is allogeneic hematopoietic stem cell transplant that can correct some of the features of the condition, but does not effectively correct CNS problems or skeletal problems. And also enzyme replacement therapy, which again has some effect, but leaves many aspects untouched. And again, by infusing these gene-modified cells and overexpressing enzyme, we hope to be able to address all of the different aspects of the disease. A proof of concept study was initiated. Again, this was started in TGET in Milan. 8 patients were treated. And again, I'll just show you some of the data from this study. So once again, we see increase in enzymatic activity in the CSF. So again, this is supraphysiological levels of activity which is sustained after a significant period of time. But that leads to the decrease in the abnormal substrate. So dermatan sulfate and heparan sulfate are significantly reduced and remain reduced after the infusion of the gene-modified cells. In terms of the cognitive function, and again, we're looking for that normal trajectory, and you can see, apart from one child, and I won't go into the details here, but the other children all show, after gene therapy, continued development of learning function. The MRI scores remain low. There is no progression of leukodystrophy and inflammation in the CNS. The growth in these individuals remains normal, and again, you would expect that there would be a drop-off in growth because of the skeletal dysplasia, but these individuals all have normal growth. And then when we look at shoulder and connective tissue movement, the substrate normally limits the ability of these children to move their limbs, and this is compared, patients treated with gene therapy compared to patients with an allotransplant, and they all start off with some level of restriction, but the patients who have had gene therapy by 3 years post-treatment have no restriction, whereas there continues to be restriction in the children who have had allotransplant because there is not effective clearance of the substrate. And again, just to show you what can happen, this is a child prior to receiving the therapy, and you can see him here. You can see the hunched shoulders and the restrictions that he has. And then again, this is when you can see that he has limitations in the way he lifts his shoulders. But then this is 3 years after the treatment. And again, he has much more freedom of movement. The phase 1-2 study led to us having further interactions with the regulators. And again, when we talk about challenges, the FDA were insistent that there is a standard of care in allogeneic transplantation and insisted that we do a randomized trial of HSC gene therapy against allogeneic transplant, despite the fact that the allogeneic transplant has a significant chance of mortality, and mortality is not an issue associated with HSC gene therapy. You can tell by the tone of my voice that I did not agree with that, but the FDA insisted that we did a randomized trial. We did do a randomized trial, 20 patients in the gene therapy arm, 20 patients in the allo arm. The enthusiasm for gene therapy by patients and by the physicians meant that we recruited a year in advance of our initial projections, and we are due to have treated all patients in both allo and HSC gene therapy a year and a half after starting the treatment. So this is quite possibly a first for the field to have a randomized trial of gene therapy against allo transplant, and we'll see what the outcomes are. We're looking at both safety outcomes in terms of death, rescue, allo transplant, treatment failure, as well as efficacy outcomes in terms of cognitive impairment and stature and growth as well. So the readout of that will be 2 years after the patients have been treated. So that's the primary endpoint analysis. So I will finish here just to say that this is, we have to start with great science. We have to start with a hypothesis, a biological hypothesis, and a technology that can address these genetic diseases. Show that in both the preclinical models and in clinical treatment. But to make a medicine, we have significant challenges, both in the manufacturing and ensuring the stringency of regulatory approvals, which of course is absolutely key for the safety of patients. But beyond that, especially in these rare conditions where there are severe clinical manifestations, the ability to rescue or to treat effectively depends upon diagnosis, disease awareness, and I would argue in many cases newborn screening as well. And these severe conditions with these advanced therapies need to be treated in skilled treatment sites or qualified treatment sites where there is expertise, not only in delivering the treatment, but also in understanding the clinical manifestations of disease and following these patients effectively. And we need to be able to think about access for patients. We have a different model here. We have a single treatment. We're used to paying for treatments on a weekly basis, on a monthly basis. We're now paying for a one-time treatment, and that requires a different model, a different assessment of value for these medicines as well. So this is an ecosystem that has a huge opportunity to benefit individuals, to potentially cure individuals. And we have to work as an ecosystem to ensure that this gets out to patients. And I would just say a huge thank you to the many people who work passionately to deliver these therapies. Thank you very much.
Thank you. I think we've got time for one quick question. Munir, could we have a microphone down at the front, please? Thank you. No, it's all right, it's coming.
I can hear you. I'm not sure.
Yeah.
Thank you. Amazing sort of videos of kids running around and so on. Just, so it's obviously cure.
Yeah.
So that loop you showed in terms of taking the cells to delivery, how long does that take?
So that takes about 4 weeks.
4 weeks.
It's about 4 weeks. And the majority, so actually once the cells are received by the site and to correct them, it only takes 48 hours, and then the cells can be cryopreserved. But we have to do quality control around those cells. So a lot of that is about doing the quality control, doing the release specifications, the documentation, etc. So that's actually what takes the time to then have all of that before the product is released and then shipped back to the patient site.
Okay,
so,
then the diagram you showed of the FDA trial, FDA's of mandated trial, 20 versus 20. Did you interact with other regulators, and was that?
So we actually had parallel scientific advice between the FDA and the EMA. The EMA accepted our initial proposal, but the FDA did not. The FDA insisted on a randomized trial, which we then took back to the EMA. So the same trial was the iDEND and the CTA for the two were the same, and so.
So if you just use historical controls, which is presumably what they.
That's what we originally were going to use, was to use historical controls, but the FDA said that they felt both treatments were in equipoise, and therefore we need to do a randomized study.
Interesting.
Thank you. Thank you very much, Bobby.
Thank you very much.
We'll move on to the next speaker, who is Dr. Ruben Rizzi from BioNTech in Germany. So he's the Senior Vice President of Global Regulatory Affairs, and then particularly led the global regulatory aspect of their work during the COVID-19 vaccine development. He's still working on the vaccine, but also with the teams who are developing further pipelines in BioNTech. And today he's going to talk about individualized mRNA vaccines. Over to you.
Thank you so much. Thank you. Thank you. Good afternoon. Thank you so much to the British Pharmacological Society for inviting me to be part of this amazing, remarkable lineup of speakers, fueling my imposter syndrome today. I'm going to try and navigate you through the perspective of BioNTech, who is developing together with Genentech fully individualized messenger RNA-based immunotherapy. You see here the word vaccine. We're going to discuss that later. For people working in regulatory affairs, it's a bit of an itchy word when it comes to cancer immunotherapies, but there's a reason why we still use it. You have heard already this afternoon from Dr. Hopper from MHRA the pioneering work that the UK agency is doing in terms of creating a framework to regulate mRNA-based individualized therapies. Hopefully, I'm going to be able to share with you a complementary perspective coming from drug developers of what it means for us and what are the uncertainties that we see. Disclaimer. Obviously, I speak on behalf of myself. I am biased. I'm an employee of BioNTech. I own shares of the company. A short introduction about the journey of BioNTech in the world of immuno-oncology. I'm sure you know our company from the COVID-19 vaccines that we developed together with Pfizer and that we're still working on. And we created basically a messenger RNA-based infectious diseases pipeline in the meantime on the wake of the COVID-19 experience. But BioNTech was born as an immuno-oncology company in first place, and our founders are oncologists themselves. So our real nature is really working on immuno-oncology. And at the end of the day, we were born as a messenger RNA organization. Today, as you will see in the next couple of slides, we're not really anymore just the messenger RNA oncology company, because we have diversified our pipeline quite a bit. And what you can see here is a summary of the rationale behind diversifying the pipeline, which is not just creating more opportunities and more shots at target, but it is really about having complementary approaches to address the challenges of today's oncology. We want to be able to use different modalities to be able to address the continuum of cancer, which is not just one niche in one indication, in one treatment line, but it's really going from the diagnosis, the adjuvant setting, and the late stage. And this can only be achieved in several cases with a combination of different modalities, different mechanisms of actions, and different technologies. This is why we have today mRNA immunotherapies, obviously still part of what we do on a daily basis, but also ADCs, CAR T cells, and also bispecific antibodies. And this is what you can see also in our pipeline. This is just the oncology pipeline. It's 2 slides. This is the first part. I'm not going to read through anything, but you can see the platforms on the very left. When you have mRNA, you have CAR Ts, you have protein-based therapeutics, antibodies, ADCs, bispecifics, and so on. Last but not least, I want to mention, you have seen the news, we have recently partnered with BMS for the development, in particular, of our PNT327, which is a bispecific antibody, VEGF and PD-L1. Very promising target. We're not alone in this race, but it's something that can be transformational for a number of oncology settings, as most of you probably appreciate. Let's now take a step back and focus on the messenger RNA immunotherapies that we are developing. We have a few different platforms. I'm not going into the details of all of them. We will try to identify what are the things that they have in common and what is specific to our fully individualized platforms, which are a kind of a special thing. This is the 2 main approaches that we have for immunotherapies that are messenger RNA based in oncology. What you see on top is what we call iNeST, and this is what we will focus on today. It's individualized neoantigen specific immunotherapy. This is a fully individualized product, so each patient, they get their own vaccine, so to say. What you see on the bottom is called FixVac. It means fixed antigens. It's probably not as fancy in terms of the name. It's an off-the-shelf therapy, so we predefine the antigens that we want to target for different tumor types and depending what they generally express. It's a typical, if you want, precision medicine approach where you identify your targets and then you have your off-the-shelf therapy, which is targeting those biomarkers. Today we're going to talk are mostly about what you see on top. This is the clinical trials that we are conducting for both platforms. I will give you some details shortly about what you see on the left, which is the clinical trials that we conduct for the individualized vaccines. You will see some things that these trials have in common in terms of the settings and the indications and the type of population where we are exploring this full individualized platform on. Just one maybe housekeeping note before we go through all the slides. Obviously, it's past 4 p.m. I don't think many of you want to really spend a lot of time going through regulatory guidelines and references. I'm going to go fast on some parts of the presentation for the sake of everybody's time and sanity, but you always find references, hyperlinks, and at the bottom of the slides, being men of science, you can always find some references of what I'm talking about, if there's a guideline, if there's a publication, articles, so you can go and check them out if you really want to go into the details of something while maybe I skim a bit too fast. So, this is in a nutshell what our platform, iNeST, is. It's messenger RNA. It can encode up to 20 neoantigens. I mean, up to 20 doesn't mean that it's always 20 neoantigens. And it's formulated in lipoplexes for intravenous administration. It sounds quite straightforward. Obviously, it is not. What is really important is not just this snapshot that gives you an idea of what the product looks like, but it's really the process that is behind coming to that product. And this is what the workflow looks like. We start from patient samples. There's a sequencing step where we do map all the mutations that can be identified in the patient's tumor. And then there's a neoantigen prediction step, and I'm going to tell you something more shortly about this, where we identify those mutations that are suitable targets or are better targets for a potential mRNA vaccine. And then what happens, we say downstream in steps 4 and 5 is the actual manufacturing of the vaccine and then obviously the administration of the vaccine to the patient. Messenger RNA in itself comes handy for the last 2 steps because it can be manufactured rapidly. It's easy, fairly easy to design in a way once you have established your platform. So it technically allows this to be delivered in a clinically reasonable time to be able to be administered to patients. But obviously from a regulatory point of view, what is a bit more complicated is what happens between 2 and 3. There is these key steps that are actually defining what the final product is for each individual patient. And eventually they define the composition and they define the benefit risk, obviously. And we will come to that later. Why do we need individualized products? No matter whether it's our platform or someone else's, obviously we all know cancer is not one disease. We tend to cluster cancers. This is how we've always done, based on histology, based on location of the primary tumor, staging. It doesn't mean that all these cancers that we group together are the same. Biologically, they're not. We know even within the same tumor, we have different cells populations that are going to keep evolving, accumulating mutations. And fully individualized products are really the only way we can address this heterogeneity. So at the end of the day, this is the best tool that we have today to be able to address cancer heterogeneity in itself. It doesn't mean that it's easy, but we have today the technologies, and it's not just about messenger RNA, it's also the softwares and the AI, machine learning systems that can help us identify those targets that enable us to try these developments. And indeed, what you see on the right is that we have, in most common cancers, we have a huge mutational burden in terms of the number of mutations that we can find. These are all new mutations. These are non-synonymous mutations that you can find in different tumor types. And as I said, mRNA, you see on the left, the general mechanism of any mRNA therapy or vaccine is a good tool to be able to express neoantigens that are actually targeting those mutations that we can identify in each patient's samples. The step of identifying these neoantigens, obviously, is not easy. We tend to oversimplify when we go through the whole workflow, but it's actually an incredibly complex exercise. it's not just about having sequencing data, it's also about being able to identify those mutations that we have in this sequencing data, and it's also going to be able to prioritize these mutations. Can we really identify not just the mutations that exist in that specific patients, but also which of these mutations are good targets? Which of these mutations need to be excluded and cannot be targeted with an immunotherapy because maybe they're too close to self, their products are going to be proteins that are too close to the normal proteome. So we need to be able to prioritize, and we need to be able to exclude some of these mutations, and we need to be able to identify those mutations that have the characteristics of being better targets for immunotherapy. We have a system that does that, and one of the big topics from a regulatory point of view, and Dr. Hopper mentioned that earlier today, is how the regulatory agencies are going to look into these systems, whether we call them algorithms, machine learning systems, or AI. There is this system that is actually looking into sequencing data and deciding what are the best mutations to be targeted for this patient. In terms of going back to the clinical development, how do we translate this workflow, this process, into a clinical candidate that can actually be investigated in trials? So there is one hypothesis, and I want to emphasize hypothesis because obviously we need to generate data, and the data are going to eventually confirm and back up what we assume. We assume that intuitively this type of immunotherapies are going to be most effective in early lines, in the adjuvant setting, for example. And there are different reasons for that. Again, I want to say intuitively, maybe it's not so intuitive, but you can see some of these reasons here. Once you have a bigger tumor burden, it becomes more complicated. There are resistance mechanisms that are already embedded within the tumor. It's more difficult to address. The immune system with the different treatment lines, as soon as you start getting chemo in, the immune system is going down, so it's more difficult to achieve a successful immune response to the tumor antigens in any case, and so on and so forth. And this is one of the principles, not the only one, but one of the principles that has been used to drive, at least partially, our clinical development strategy, and this is resulting in the phase 2 studies that you see at the bottom, and I'm going to go through a little bit more of the details of these studies shortly. This was the first phase 1 study for the technology, conducted by Genentech. I wouldn't say typical phase 1 first-in-human study, safety as a main objective. We could see proof of concept that the vaccine itself could elicit CD8 plus T-cell responses. One key aspect for this first-in-human, for this type of product, is also verifying the feasibility from a technical point of view, because we're talking about the workflow that I showed you before. It comes to being able to adapt that workflow to the clinical practice and being able to show in a clinical trial setting that that workflow is compatible with the timelines, with the deadlines, and in general with the clinical setting. And this is what we could achieve in this phase 1 study. We started with those phase 2 studies afterwards. This one is a study that is currently running in stage 2 high risk and stage 3 colorectal cancer. This is ongoing in many countries, including UK. In this case, we are using ctDNA to identify those patients that after resections are going to be more likely to relapse. There are different assumptions here. The first assumption is that ctDNA can be used to identify those patients early, because today what happens is that after surgery, they get adjuvant chemo, and then a good proportion of these subjects is still going to relapse. What we want to do with ctDNA, we want to intercept these subjects that are more likely to relapse and treat them with our immunotherapy and see if we can delay, postpone the relapse of colorectal cancer. This is the study design in a nutshell. It's particularly interesting because besides being a fancy design, it's particularly interesting because it's monotherapy in this case. So the subjects are just receiving our mRNA immunotherapy, which is manufactured for them. And it is compared with what is the standard of care today for this population, which is watchful waiting, basically, after they get the adjuvant chemo. So we're going to get to be able to see a signal of whether we are really doing a benefit in this population, which is a very interesting one. We have readouts later this year, so everyone is very curious about this. This is not a study that we or Genentech sponsored. This is from MSKCC It's a phase 1 study in pancreatic cancer. We all know what is the unmet need for pancreatic cancer. We all know how difficult this disease is to treat. And we wouldn't necessarily expect an immunotherapy to be the best option, but they initiated this study together with Atezo, and the data that they could generate in this relatively small phase 1 study have been really impressive. Not just we could see good data confirming the ability of the immunotherapy to generate T-cell responses and to generate responses in the tumor infiltrate, and we could really see the expansion after dosing and the persistence of these responses over time, over a fairly long period of time. But even more interesting, we could see how these responses correlate with clinical outcomes. Again, small sample, it's a phase 1 study sponsored by MSKCC, but still you could see when you have responders, you can see how this is showing up in the actual clinical outcomes. So it was interesting enough and exciting enough. These are not even the most recent data. There's more recent publication you can go look up for. We initiated, and Genentech is conducting this, another study. It's a phase 2 study in the same setting of pancreatic cancer. It basically mimics, to an extent, the same design. Obviously, it becomes a randomized study where we are comparing our immunotherapy plus Atezo plus FOLFIRINOX against FOLFIRINOX, and we're going to see whether we can show a benefit of the combination. Obviously, it's very promising. It's very exciting, to be honest. Last but not least, we're just initiating now a phase 2 study. This is bladder cancer, comparing with, in combination with nivolumab. And we're going to have to, in this case, again, the combination of the immunotherapy with Nivo. You will see that the combination with PD-L1s comes quite often. Another signal-seeking study in a different setting. So long story short, we have this fully individualized platform that is currently undergoing several phase 2 studies globally. We're going to have readouts in the next 2, 3, 4, 5 years for most of these indications. We're going to be able to tell whether we can really show benefit for these indications for a full individualized product. And obviously, we're not alone. Other companies are doing the same. Moderna, Merck, other companies are developing fully individualized therapies. And now the question comes, are we ready to have these therapies deployed in our healthcare systems? Are we ready from a logistical point of view, from a regulatory point of view, from an access point of view? These are not necessarily ultra-rare diseases. These are fairly normal oncology settings, but obviously fully individualized therapies require something slightly different than the average drug. I will use the last few minutes to just give you a sense of what are the ongoing discussions, some of the uncertainties, and some of the interesting things that are being discussed right now on this topic. First of all, I take a step back on what mRNA really is from a regulatory perspective, because messenger RNA can be used in a number of ways. That's a tool. Like all of these technologies, they are tools. So messenger RNA can be used for different types of medicinal products. We go from protein replacement to an extent, infectious diseases, vaccines, obviously, immunotherapy. Is it always the same entity? Well, you can argue, obviously, from a clinical point of view, different populations, different routes of administrations, different benefit risk. With infectious diseases, vaccines, we give them to healthy volunteers, obviously. It's not the same as going in a last line oncology setting. Different types of antigens, also different amounts of mRNA. So there are differences. At the same time, the flexibility of messenger RNA as a technology in itself means that in principle, you could have an infectious diseases vaccine, which is based on messenger RNA, and a cancer immunotherapy, which is based on mRNA, and they can be based on basically the same mRNA format, the same backbone, very similar formulations. So at the end of the day, from a technology point of view, they can be quite close to each other. At the same time, they are classified differently in most situations. And we have questions about whether that's right or wrong. Everyone can have different opinions, but it's obvious that this is an area where we are still learning, the regulators are still learning, and we still need to develop probably what is going to be the long-term classification for messenger RNA, as for any new technology at the end of the day. Regulatory guidance is emerging. These are just a few examples. These are not just guidelines for mRNA for individualized oncology immunotherapies. This is general guidance that is emerging on on mRNA. So the first one is a WHO guidance from 3 years ago on infectious diseases vaccines, and then you see a quality guideline from EMA, an FDA guideline on therapeutic cancer vaccines. I'm going to come to the MHRA guideline later on. The classification remains a bit inconsistent, maybe. We still have, what happens today is there's basically an mRNA vaccine that is used as an infectious diseases prophylactic vaccine falls in the big bucket of vaccines. And this is why using the word vaccine for oncology immunotherapies is always a bit confusing. It's not necessarily wrong. You give the product, you get an immune response against an antigen. But from a regulatory point of view, it can be confusing because vaccines are, in most cases, infectious diseases vaccines. And it doesn't matter what is the technology they come from. So they can be protein vaccine, they can be inactivated vaccines. They always fall more or less in the same regulatory bucket in most regions. At the same time, a very similar product that, however, is expressing tumor antigens, then it falls into a completely different bucket of most cases gene therapies or ATMPs. Whether that's right or wrong, it's not for us to discuss, but it's interesting that we have a very similar technology that can be regulated in very different ways depending on the application. I'm going to mention platform technologies for a reason. We have heard the word platform a couple of times, unsurprisingly, already today. There is something that connects the different applications of messenger RNA as well, and this is going back to how we can regulate full individualized products. This is an example that comes from COVID-19, which sounds very strange, but you're going to see how this can be close to the world of full individualized products. This is what happens from a regulatory point of view when we had the Omicron BA.1. It was a B-specific vaccine, apologies, Omicron original, and it was Comirnaty original and Omicron BA.1 vaccine approved. This is what happened in Europe, in UK. We had generated data for a BA.1 vaccine because the epidemiology initially suggested that BA.1 was going to be the major variant of concern. But then it came out last minute that BA.4/5 was probably going to be emerging in a stronger way, and we had to adapt to that. So the whole process of strain update at that time was still at the very early phase. It was the first time, basically, that we were going through this kind of process with the global regulators and WHO, and we had to switch rapidly. We didn't have clinical data available at that time for a bivalent original Omicron BA.4/5 vaccine because it just came up too late for us to be able to set up a clinical trial, manufacture a candidate, and run a trial and generate data and submit the data on time for approving for seasonal vaccinations with the new vaccine. What the regulators could do, though, was looking into the totality of the evidence that was already generated for BA.1, as well as the clinical data that we had already generated with other variant-adapted vaccines that had been brought into the clinics and never approved, because we were obviously testing different variant-adapted vaccines, and looking into a panel of non-clinical data suggesting that there's a good predictivity of the animal immunogenicity models to the human immunogenicity. This whole package allowed the regulators to approve the BA.4/5 bivalent vaccine without having clinical data on that candidate, and that was the first time, actually, that they did that for COVID-19 vaccines. This became later part of the strain update process for COVID-19 vaccines. These are slides from an FDA presentation for a VRBPAC meeting, I think in 2023. Where basically you don't have to read it through. The FDA said, if you have an established platform or technology or an existing vaccine, you don't need to generate data for every clinical data before approval for every single variant-adapted vaccine that you develop. And this has become the process, at least until this year, for variant-adapted vaccines for COVID-19. And what the regulators were doing in that case was adopting a platform approach. So they would look into platform data generated for the same technology with different antigens, with different variations of the same spike protein antigens, and they were extrapolating those data to support the benefit risk of a new vaccine for the season. This is exactly what we will have to do with additional complexity when it comes to assessing the benefit risk for full individualized mRNA-based immunotherapies in oncology. This is the workflow that I showed you before. At the end of the day, our product is what you have in step 5, which is specific to that individual patient, and it has been manufactured for that individual patient, so it cannot be compared easily from a clinical outcomes perspective at least to any other product manufactured with the same platform. So a lot of the things that we have to do are going to be based on platform principles. The CMC package is going to be a CMC package that describes the process, that describes the backbone and the common features of the technology, but it's not going to be specific to the actual encoded antigens for each patient. At the same time, the clinical package the non-clinical package is going to cover the whole technology as a platform, but is not going to cover the specific antigens that are expressed by every single candidate. And obviously, we're not going to be able to investigate clinically all the potential combinations of neoantigens because they're close to infinite. The neoantigen prediction, as I mentioned before, from a regulatory point of view, this is a critical step. How do we define it from a regulatory perspective? Can we improve this package? I think Dr. Hopper already mentioned what is the current debate around this topic. Obviously, industry is eager to say we can update this technology because it's what is happening today with AI systems, that you can have a continuous update of your software in order to be able to improve its ability to predict. But can we really do that in a regulated environment, and to what extent, what kind of validation can we provide? Because this piece of system is what is actually defining the composition of our vaccine at the end of the day. So how the life cycle of this kind of system looks like. So there are platforms approaches that are going to be required for regulating fully individualized products. It's not a platform-specific consideration. This is true for everyone who is working on this type of program. The potential from a clinical perspective is huge. We are moving from a typical precision medicine, let's chase a biomarker approach, into something that is really tailored to the individual patient. And I'm 100% positive this is what the future of oncology is going to look like, at least in some settings, because we have the technologies today to enable that, whatever the platform is going to be. mRNA products are still a relatively new entity. As I said, this goes beyond the fully individualized platform in any case. The regulatory world of definitions and frameworks around messenger RNA is being established now. And it is the same for AI systems and machine learning tools for drug development and for definitions, selection of antigens or definition of compositions of vaccines and so on. It's an ongoing process, and it's under development. So we really need all to work and provide our contribution and our perspectives so that we can build what the regulatory framework is going to be in the future. As I already mentioned, technologies are enablers, they are tools, and then the data are going to be able to tell us whether we're doing things in the right direction or not. I want to mention, last but not least, again, the draft guidance that MHRA put out. This has been a very important step forward because it's really specifically addressing what is the regulatory framework for individualized mRNA products. There are a lot of areas that are up for discussion. Do we really need to long-term follow-up before we know what the data are going to look like eventually? What is the right regulatory framework for what happens in the upstream part? Is IVD okay to define that kind of application, or do we need to look for something more specific? This is exactly the discussion that is taking place right now with the MHRA and the different stakeholders, and this is a very important discussion, and the most important thing that probably this draft guidance from the MHRA did was exactly to encourage this conversation with all the stakeholders that are working in this field. Last but not least, I want to mention this collaboration. You know, BioNTech is working with the UK government since years. This development in particular I mentioned today with autogenous cevumeran, our individualized mRNA vaccine, is part of that collaboration as well. Our clinical trial is running in UK in a number of sites, and this is an incredibly positive experience in terms of being able to really leverage the capability of the UK healthcare system. Why this is so important? It's not just for running clinical trials in a more efficient way. We're going to need to ask ourselves the questions in the future, not just for our platform, but for all individualized platforms. What do we need to do to deploy these type of products through the entire healthcare system? And this is not so obvious. These are special products that require special logistics. What we do at clinical trial setting is just a small bit, and then we're going to have to discuss what does it mean from NHS deployment in order to be successful. That's it for me. I hope it was helpful and interesting. I'm happy to take questions. In any case, there's my email over there, so you can reach out if we don't have enough time now, or if you have an afterthought, you can reach out to me via email. I will try to follow up with everyone. Thank you.
Thank you.
You have time?
One question?
Yeah, sure.
Yes, right at the back, please.
Hello. Thank you. Fascinating talk. And you're right, it really is the future. I was just wondering about side effects. If you are giving essentially a different treatment every time because of your individualized vaccine, do you think there'll be any commonality in potential adverse effects?
That's a good question. This is actually one of the topics that we're going to have eventually when we come to the point of having the final readouts and we get to the point of getting close to the regulators for an actual approval. The question is going to be, what do they want to see to be able to extrapolate the benefit risk across different patients that have received a different composition? The good thing that we have observed so far is that in terms of safety and tolerability, we have a very consistent profile, which is based mainly on reactogenicity, which is what you expect for this type of immunotherapy due to the immune response. But obviously the question is, what kind of extrapolation can be made across the different individual subjects within the same population and also across different indications, because at the end of the day, you have different tumor types that are going to be targeted, and then you have different compositions that are looking into different mutations. So this is actually one of the topics, how you can extrapolate your clinical experience to inform the benefit risk across the board.
Thank you very much.
Thank you.
Thank you. So thank you very much, Ruben.
Sure.
In the interest of time, we'll move on to the next speaker, and the final speaker in today's session, who's Professor Peter Johnson. Peter is Professor of Medical Oncology at the University of Southampton and also the National Clinical Director for Cancer at NHS England. He has a particular policy interest in improving cancer survival, particularly the earlier and faster diagnosis of cancer and its treatment. And today is going to talk about cancer precision medicine in the NHS. Peter.
Thank you very much, and congratulations for making it this far on a hot day. I'm going to talk you briefly through some of the areas where looking at the application of molecular diagnostics and precision medicine to practice in the NHS is going, and what we're doing to try and support it as part of that. Certainly in the systemic treatment of cancer, there are basically two classes of entity that have been most effective in changing the outlook from treatment. Firstly is small molecules, and the diagram here shows you the external loop of the EGFR receptor with a gefitinib sitting in it, and you can see on the bottom the effect on a very large adenocarcinoma of the lung, shrinking away from a very large tumour mass to nothing at all as a result of the inhibition of signalling in that particular activation loop. On the right-hand side are the effects of immunotherapy. This is checkpoint blocking antibodies stimulating T cells to react against tumour cells, shown in the scanning EM at the top. And at the bottom is a series of photographs of somebody with melanoma en cuirasse in the chest, going through the chest wall, as you can see on the CT scan, gradually melting back as the active immune response is developed by the disinhibition of T cell recognition of the antigens in the melanoma. So we already have in cancer medicine a lot of evidence that precision medicine will be effective and can do things differently for patients. So what are the issues in delivering precision for our patients and for the healthcare system as a whole? Well, firstly and clearly fundamentally is identifying an actionable phenotype, and in most cases in cancer that involves analysis of somatic changes in cancer cells as the specific targets of therapy, as in EGFR mutations in non-small cell lung cancer. There is also critically, as you've heard already, the question of biological heterogeneity, clonal evolution, divergent evolution of tumours, and the point at which you can interrupt the dysregulated growth pathways, whether this is a truncal mutation or whether it's something which only takes place in a branch in the subset of the tumour. Also fundamental, clearly, are having agents which will target the lesion, whether these are small molecule inhibitors or whether these are therapeutic agents. And clearly, to underpin that, we need, as you've heard a lot already this afternoon, the appropriate drug approvals process. And then critically for the healthcare system, we have to have a means of joining this all up. So some means of identifying the people who are potentially suitable for this type of treatment. So whether this is prospective screening of populations at the time of diagnosis, or very rapid turnaround somatic testing at the point at which we wish to give treatment. Whether we do this on fresh or fixed tissue, increasingly we're looking at circulating DNA, and I'll talk a little bit about some of the data we're generating from looking at the minute amounts of DNA which are liberated by tumours. And of course, as we try to make this cost effective, moving from single gene targets to panels to exomes to potentially whole genomes, in order to derive the information in as cost effective a way as possible. And fundamental to all this, and a problem which I wouldn't pretend that we have entirely cracked yet, is having really rapid turnaround, fully validated tests for all the potential targets. So just to give you a bit of perspective, this is a heat map, although you can see that there isn't a lot of heat in most of the map. Looking at down the vertical axis the names of the large drug companies, and across the top there all the different therapeutic areas, and this is a reflection of R&D spend by different pharma companies in those different areas. And you can see that there's one column which is absolutely red hot, which is oncology drugs, and then there are one or two bits in respiratory medicine and I think one or two other illnesses. But you can see that the pipeline is hugely dominated at the moment by cancer drugs, and if we look at that, it corresponds to about 84% of the novel drug pipeline globally that we see. Our response to that in the UK has been reasonably effective. So this is oncology drug availability over a 3-year period from 2020 to 2023. This is an independently produced report that looks at that, and the green bars are fully available and the red bars are not available at all, and the orange are intermediate levels of availability. And you can see that England, which I've highlighted, is above the middle of the pack in terms of the availability of drugs. So I think our regulatory and our reimbursement and our approval processes are serving us quite well with the new cancer drugs as they come through, bringing them into the system. Similarly, the length of time it takes us to go from licensing to rollout of the drugs, thanks to the processes in NICE and the MHRA, and latterly the Cancer Drugs Fund as well, have also meant that English drug approvals and the rollout to availability in the healthcare system as a whole are generally at the faster end of the spectrum across Europe. And again, I've highlighted England in that. And that's so important, because you heard earlier this afternoon the agonizing tale of waiting to get a new treatment for Duchenne muscular dystrophy. And of course, cancer patients' trajectories are measured in days, not years, in many cases. And so making sure that we have a really effective approvals and availability process is critical to the system. So I'd wanted to give you 3 specific examples of precision medicine and how the NHS is tackling this. Firstly, the question of molecular diagnostics for somatic tumour changes and how we're dealing with this need for a rapid information on somatic analysis. Secondly, a bit about mutanome-based therapeutics, which is another way of saying RNA vaccines based upon the patterns of mutation in tumours, which Ruben has just set up very nicely. And thirdly, a little bit, because it says pharmacogenomics in the title, a little bit about cancer risk and pharmacogenomics at the end. So the implementation of genomic medicine in the UK is driven by a concatenation of different levels. Firstly, our fantastic research and innovation ecosystem. Secondly, the NHS and its enthusiasm for innovating and for bringing new treatments in as quickly as we can prove them to be effective. A very vibrant industry, as I've already shown you in this area, producing a pipeline of agents, and also very importantly, political will to support this, and a number of strategies over the years, whether it's the life sciences strategy, the genomics strategy, and the support of the Secretary of State, all of them really important in making sure that there is a continued focus on how we do this, how we turn molecular insight into pathogenesis and characteristics of tumours into things that we can do in the clinic. And whilst everybody's very well aware of some of the strictures and limitations of the way the NHS behaves and some of the things that we're able to do, nonetheless, I think when we get it right, we can demonstrate that we can really do this at speed and at scale. Moving genomics from relatively experimental into mainstream diagnostic delivery is a journey that the NHS is undoubtedly still on. Lots of different elements in that, a national leadership model led by Sue Hill and the Genomic Medicine Service in the NHS, priorities of guidance, really important public engagement and faith and confidence in the system, in the way it's regulated, and ambassadors in all the different levels in the genomic medicine service across the country for making sure that when we roll these things out, we're doing it effectively and appropriately. Lots of communities of practice, lots of work raising awareness of healthcare professionals, recognizing that this up until quite recently was quite a niche interest, but is now absolutely mainstream, for example, in diagnostic pathology or in mainstream oncology. And making sure that we've got our clinical workforce coming with us on this is terribly important. What we're in the process of doing is moving from the old single gene tests where we would look at an EGFR mutation or a RAS mutation in a tumor to small next generation sequencing panels to much more extensive panels incorporating both DNA and RNA in order to pick up all the multitude of targets that might be potentially actionable in tumors. And this is undoubtedly gathering pace, and the speed of approval of new targets and new agents that we need to target the testing for is driving a lot of the activity. And I want to pick up in particular, as I say, about the area of liquid biopsies circulating DNA as a means of short-circuiting at least some of the bottlenecks in this system. But this transition, and when we're sort of here at the moment in most of the country, it's a variable pace, and the implementation of it is not entirely straightforward. But I think very importantly, this is very much a work in progress. If we take non-small cell lung cancer as an example of this, there are currently 8 small molecule targeted treatments for non-small cell lung cancer. I wouldn't invite you to try and read the slide. All I would highlight is that if you look at the different mutations to which they're targeted and the point in the trajectory of the illness at which they're targeted, the great majority of them are for initial therapy. And the point about that, of course, is that people who present in the clinic with lung cancer with symptoms very often have quite advanced disease, and the length of time that you have to before you act has to be very, very short. And so we need a turnaround time for a sophisticated molecular analysis of at least the 8 genomic targets in non-small cell lung cancer to be in the order of a week or 2 during the time it takes us to get the bronchoscopy, the tissue analysis, the histopathology organized in order that we can start treatment in as rapid a way as possible. So what that does is it focuses the mind on how you do this at scale and how you do this at speed in an accurate way, and trying to incorporate a number of different potential targets for targeted therapeutics. So we've been wrestling for a few years with doing this with conventional tissue sections, and the histopathology service is a very dispersed service, lots of hospitals across the country sectioning bronchial biopsies, transbronchial biopsies, and doing immunohistochemistry on them. And then sequentially, in the most part, doing molecular analyses. What we wanted to do was to see whether circulating DNA might give us an alternative route to molecular diagnosis. So we started a couple of years ago now in a pilot phase, taking blood samples, extracting DNA from plasma at the point at which patients presented with appearances on their cross-sectional imaging which suggested lung cancer. So we took the blood sample even as we arranged the bronchoscopy and started the histopathology off. And we've gradually expanded this from 700 samples in the first year to 10,000 samples in the last year. And most importantly, what we've shown is that if you do the usual thing, which is take a CT scan, arrange a bronchoscopy, arrange the histopathology, and then do the molecular testing, it takes you on average about between 35 and 40 days to arrive at a molecular diagnosis. If, on the other hand, you take the blood sample as soon as the CT scan shows you something abnormal, we can pretty much halve that time to molecular diagnosis. And that's incredibly important, of course, for patients diagnosed with lung cancer, because it means they're not sitting there waiting for us to tell them whether they can have first-line treatment with a kinase inhibitor as opposed to conventional chemotherapy. They can get straight on with the treatment as soon as we have the ctDNA result. So I think this type of technology, which we're now starting to use increasingly for looking at targeted therapies in, for example, breast cancer, and we're testing also in hepatobiliary cancers where the biopsy issue of getting a good sample from a deep pancreatic tumor can be really quite time-consuming, trying to see whether we can set up these alternative pathways to diagnosis as part of the NHS. So there are potentially quite a lot of different use cases for circulating DNA, and I won't go through them all. We've been looking at the screening of asymptomatic populations using patterns of abnormal methylation in plasma DNA as a very early marker of carcinogenesis. And interestingly, methylation patterns vary according to the tissue of origin. So these multi-cancer early detection tests, which we've been testing out, can potentially show us not only that there's a tumour present, but the likely tissue of origin. So we completed a very large randomised trial of that with the Grail company, which completed accrual of 3 years of sequential blood sampling last summer, and we'll see the results of that early next year to see whether we see a stage shift and a change in prognosis. The alternative route to diagnosis I've already mentioned. So at the moment, of course, we're doing it in parallel to tissue sampling. There is probably a subset of people with lung cancer where we don't actually need the histopathology, where we could act on the molecular findings in circulating DNA, and that's our next pilot scheme, is to see whether there's a population of people in whom we can safely omit the biopsy and go straight to therapy based on the molecular diagnostics. Similarly, the selection of therapy for advanced disease is based on the same idea, that you target the findings that you make in the circulating DNA. Similarly, looking for the possible recurrence, this is an area which has been known about for a long time in hematological malignancy, where, for example, in childhood leukemia, we monitor for the presence of clonal B cells during remission, or indeed in the treatment of chronic myeloid leukemia with kinase inhibitors, we monitor for the presence of the Philadelphia translocation in circulating DNA. And increasingly, we can look at how we do that in solid tumors that are in remission following targeted agents in order to test for the emergence of clonal resistance. And that's the other idea, is that one can potentially switch therapeutics in response to emergent clones in the face of ongoing therapy. So what I hope that tells you is that we are doing a lot of work not only to optimize the tissue-based molecular diagnostics, but also to look at potential alternative routes, both in terms of detecting cancers earlier, but also moving more rapidly to targeted therapies. And this is just one of the stories. We've done a bit of press about this, and the statement from this young lady whose lung cancer got picked up as a testament to the power of this particular analysis. So clearly being told you have lung cancer, particularly at an early age, which is something we increasingly see with these peripheral adenocarcinomas. But finding that she had an ALK fusion in particular allowed us to give her an ALK inhibitor with very good therapeutic results. So moving now to the cancer vaccine question, you already heard from Ruben about the technology underpinning this, and when we started to think about this for the NHS, what became rapidly apparent was that we would need to move differently both in supporting the trials and also readying the system for the potential adoption of these treatments if the trials prove positive and they're effective and value for money. And if you say it quickly, the idea that you take a tissue biopsy, you do whole exome sequencing and you generate the sequences to create a vaccine sounds highly plausible, but then you think about it in the context of NHS histopathology laboratories in 95 different hospitals across the country, and how are you going to make sure that you get high quality tumour material for sequence analysis in a timely way in order to be able to produce, again, a vaccine in a very short space of time. And the workflow that Ruben described in the laboratories has, of course, a whole antecedent workflow in the diagnostic service in the NHS. And when we thought about this, we were very worried that you would have this marvelous technology, but it would just take us too long to get the tissue through the laboratories out the other side and out for sequencing. So we established a partnership with BioNTech a couple of years ago to support the delivery of this particular technology. Firstly, to address some of the bottlenecks which we know exist between histopathology and molecular diagnostics, to make sure that we were putting resource into that interface between the pathology laboratory and the extraction of DNA and RNA and sequence analysis. And secondly, of course, to test in the system how you might do this, because if this works and everybody with stage 2 and 3 bowel cancer needs to get a personalised vaccine generated, we need to make sure that we've got a system that is ready to do this. So the bottleneck between cell path and molecular diagnostics, to accelerate the vaccine trials, because again, you can anticipate in this type of trial that they will have a very high attrition rate from people having resection of their bowel cancer to people who have positive circulating DNA, to people in whom you can construct a personalised vaccine. You can imagine that you need to test a lot of people in order to run a trial in a reasonable space of time. So we wanted to make sure we could accelerate the trials, and thirdly, as I say, to prepare the NHS for this kind of technology and its rollout. So we've established this platform to support patient referrals, not just in the trial sites where the vaccine is going to be administered, so the usual dozen or so places where patients go to have the actual vaccination procedure, but also the much larger network of places where people have their cancers diagnosed, to make sure that we're putting resource into that wider network to make sure that we're streamlining as far as possible the pathology process. And we've done this obviously in collaboration with Genomics England and the Office for Life Sciences and the NIHR. And we've set it up as a platform to allow us to set up trials across the country in the large number of sites, not just for BioNTech, but for any company that has this kind of technology, which requires biological selection and stratification and trafficking from a large number of places into fewer. And our next partner is Scancell, who have an advanced melanoma vaccine trial called SCOPE, which is dependent on HLA type. Another 4 trials in the planning at the moment. So essentially what happens is that out at the Cancer Vaccine Launchpad site, patients are identified as potentially suitable for this. They give a very light consent to the collection of their demographic details and to the analysis of their tissue for molecular diagnosis. So they don't consent to the trial at this point, they consent to being part of the Cancer Vaccine Launchpad. They have their blood sample taken, they have their tissue sample taken when they have their resection. Both of those go to a processing in a cellular pathology genomic center, which is a larger regional pathology laboratory, which we've put resource into for the processing of the tissue. Those then go to the genomic laboratory hub. At the moment, the sequencing is done in the company, but ultimately we hope that we will do the sequencing in-house in the NHS, and then the sequencing data will be stored, and the vaccine is obviously generated at this point. If the patient is suitable, they then consent to the trial and can then move to the trial site. So a whole pre-screening and molecular analysis pre-phase to potential participation in the trial. And so this was the trial sites for the adjuvant colorectal trial that you've already heard about in the usual distribution of places across the country. But by setting up the Cancer Vaccine Launchpad to feed into each of these sites, we were able to spread the availability of trial much wider. So 13 trial sites out to 55 recruitment sites, which equates to going from about 17% of the population who'd have their colorectal cancer treated in the trial sites to over 60% of the population who could potentially take part. So this enabled us not only to accelerate recruitment into the trial, but also, of course, to make sure that we have a trial population which is much better representative of the whole population of the country, and not just those who live in close proximity to teaching centres. So, at the moment, we're just in the process of expanding this to Scotland and Wales as well, to make sure that we make this a truly national study. Through having structured setup of the pre-screening sites, we know a lot about the feasibility. We know through introductory site calls how we can get things set up. And there's an average of 16 days from the initiation visit to activating the site. So very rapid setup, and the first patient recruited within a month of activation in these sites. Lots of training going on to make sure that people on site at the recruitment sites are familiar, and also very importantly to make sure that the pathology laboratories are processing the samples rapidly. So the average turnaround time for a tissue sample in a histopathology laboratory coming in on the Cancer Vaccine Launchpad to being sent for sequencing was 2.5 days, which is an enormous shrinkage of the time it usually takes us to get samples out of the pathology laboratory for molecular diagnostics. And then the other thing we've developed is an electronic referral tool, which essentially links up the screening site to the trial site and allows us to track in real time both the patient and also their biological samples and the analysis that's been done on them. And when this was up and running, we managed to very rapidly increase the rate of recruitment to this particular trial. And before we closed to recruitment, which happened a few months ago, the NHS was screening as many patients as the rest of the world put together for this particular study, which was running in places like Spain and Germany and the US. So I think what this demonstrates is that by setting up a targeted and properly founded system for doing this, the NHS can actually rise to the challenge of bringing in material, enabling it to do genomic analysis, and feeding it through into this kind of endeavour. So these are our 16 cellular pathology genomic centres across the country where we've concentrated this resource, and they've prepared, as I say, almost all of the samples within the target turnaround time, 97% of them, very high rate of acceptance for molecular analysis, because the other thing is you need to make sure that you have high quality tumour material in order to derive your sequence. So that has been, I think, a real boost not only for the trial portfolio, but also for the way in which we run histopathology and molecular diagnostics. And again, an intended benefit of this approach is to have geared up these cellular pathology genomic centres not just to support personalised vaccine trials, but also to support the wider molecular diagnostic ecosystem in the NHS. So for the future, we plan to extend this to as many NHS trusts as we possibly can. As I said, we're planning to repatriate at the very least the nucleic acid extraction, which we've already piloted and shown we can do, and in due course also the sequencing, which currently takes place at the company. Multiple different cancer vaccine trials coming through, and we're expanding the portfolio. And again, to return to the point, getting the NHS ready for the rollout of these drugs and the effective delivery of these if we prove them to be effective and cost effective. To swing back now to my third brief topic, the germline abnormalities. Clearly cancer predisposition, relatively small number of genes that are cancer predisposing, but Lynch syndrome, the mismatch repair deficiencies, has traditionally been a big one. We know that the prevalence is about 1 in 400 adults, and that actually historically we've diagnosed only a relative minority of people who are affected by Lynch syndrome. So we set up a few years ago a Lynch program to try and identify as many of these kindreds as possible. We set up champions across the country in different places where people were presenting mainly with bowel and endometrial cancer, ran a number of workshops and made sure that people understood what we were trying to do, set up some data solutions to make sure that we could track this, and set up a registry to make sure that we could not only track these kindreds but also keep tabs on what became of them. And most importantly, we started auditing the pathology laboratories to look at the rates of testing either by immunohistochemistry or molecular analysis for mismatch repair deficiencies, and again went up from about 50% of samples of colorectal cancer being tested for MMR to 94%, and similarly from about 30% of endometrial cancers to 96% in the course of that evaluation. And what we now have is a very effective regional network of identification for people with Lynch syndrome. And of course, this is increasingly important because the primary treatment of people, for example, with colorectal cancer with mismatch repair deficiency will in due course move towards checkpoint inhibitors, because the mismatch repair deficiency results in a very high mutational burden within these tumours, which makes them highly visible to the immune system. So the pathways of care for people with germline predisposition are also changing. Finally, pharmacogenomics. We have 2 current pharmacogenomic targets in cancer. DPYD, which is a mutation which is very important if you're going to be given 5-fluorouracil, which is a commonly used drug, particularly in GI cancer treatment, also some breast cancer treatments. So we're routinely testing patients for this because we know that if you're DPYD deficient in your phenotype, then your ability to metabolize 5-FU and capecitabine is significantly impaired, and the risk of side effects and particularly potentially fatal side effects is much higher. So this is routine screening, and similarly these targets for usually pediatric patients going to have purine analogues such as 6-mercaptopurine, commonly used in the maintenance treatment of childhood ALL, is also being rolled out, and of course on the other side, some other genomic, pharmacogenomic targets also being tested. as part of this whole endeavour, we need to make sure that we are moving into the digital era with the way in which we do this. The ability to access the data and to track what's happening in order columns and so forth is incredibly important. So firstly, the genomic test directory is being moved into digital form. At the moment, if you want to look up what genomic test is appropriate for a particular patient, you have to scan through an Excel spreadsheet. Which you can download from the Genomics medicine site. What we want to do is have a proper digital test directory. We're also in the process of digitizing order management. Again, this is a heavily manual process at the moment, and it makes it very difficult indeed to track where a sample is in the system. And because the turnaround times remain an issue for tissue-based diagnostics, at least, really important to be able to do that. Ultimately, we want to move towards a unified genomic record so that we can link this to people's healthcare records and really understand the outcomes of treatment and the outcomes of our novel therapies in the people in whom we identify this. And of course, the development of common standards for testing and putting this on a secure footing. So there is, I hope you can see, an enormous amount of work going on both in somatic diagnostics and in germline analysis within the NHS. And as we try to move into the new future of genomic medicine, lots of different trends. The automation and the industrialization in our laboratory services is going on apace. This is no longer something where you have a sequencing machine and somebody pipetting DNA. This is something where you need liquid handling robots and large turnover in your laboratory to deal with it. We're progressively expanding the testing offer as the new advances come through, and we're building into the commissioning decisions for the adoption of innovation, all these things as they come through. The investment in data and digital infrastructure is going to be critical to our ability both to maximize the value to the individual patient and also, crucially, to maximize the value to the healthcare system as a whole of the knowledge that we're generating in doing this. And at the same time, of course, we have to make sure that our colleagues are aware of all the changes that are coming through and of the new things which are developing in order that they can advise their patients appropriately. So I hope you can see that there is an enormous amount going on in the NHS to make sure we're responding to some of these challenges. I think the future is very exciting from the point of view of both what we're capable of doing now, and I hope I've shown you from a couple of examples what we're capable of doing as we flex the healthcare system to adapt to some of these new developments. Thank you very much.
Thank you. Time for one quick question. Yes, right, gentleman at the back, please.
Thanks, Peter, Dan O'Connor, ABPI. Really nice to see a multitude of work. Really interested in the Lynch work because you know aspirin is also around, which is also kind of a potential alternative therapeutic intervention that can be linked now. Where do you see how sort of genomic work can really support the prevention agenda? It's really high up on government agenda, it's increasingly high up there on industry, and huge opportunity for patients and public health if we get prevention sort of right. But that diagnostic odyssey and that early diagnostic paths for the patient and then mapping that with probably surrogate endpoints over long periods of time is really going to be sort of crucial. So do you think the NHS can help in that way? Thank you.
Yeah, thanks, Dan. I think inevitably the number of inheritable predisposition syndromes that are amenable to prevention approaches in cancer are quite small. We have Lynch, BRCA and Lee-Fraumeni syndrome, and everything else is pretty rare. Absolutely intervening with aspirin in Lynch is clearly mainstream. We've also been doing, I didn't allude to it, quite a lot of work on identifying the families most at risk of BRCA1 and 2 mutations. We've started with the Jewish population where we know the prevalence is around 10 times higher than the general population, but we have just started doing retrospective identification of people with triple-negative breast cancer, young onset breast cancer, mucinous ovarian cancer, as also being at significant risk of BRCA abnormalities in the germline. And again, relatively few of them have been previously tested. So we're going to go back and catch up and identify those kindreds. And of course, there you have the option potentially of intensified screening for those populations and prophylactic oophorectomy at least to reduce the mortality risk. Similarly with Li-Fraumeni, as we identify more of those kindreds, we're moving towards routine MRI, whole body MRI surveillance for the tumours that they generate. In the broader area of genomic predisposition and prevention, I think things like polygenic risk scores are still at quite an early stage. We've seen some publications highlighting that it is now possible to identify the top decile of risk for example, prostate cancer, and given the controversies about whether or not we should be testing people's PSA and screening for prostate cancer, identifying a population at higher background risk on the basis of their genetic background feels like an intelligent way to be pursuing that kind of question as well. So I think we will increasingly see understanding of genetic background risk as one of the elements in our prevention strategy. Of course, there are huge numbers of lifestyle and demographic elements as well, which are much more dominant in terms of the numbers. But I think as we get a better grip on the genomics, then I think we will have more to be able to do.
Thank you.
Thank you, Peter, and thank you again to all of our session speakers for an excellent set of talks, and I'll hand back to Munir for some final comments.
So thank you very much, Peter, and the other talks we've heard today. So I just want to close the conference and say thank you to a few people. Number one, thank you to all the speakers and the chairs for the sessions. I apologize for the many emails I sent to you to persuade you to give a talk here, but I'm glad you accepted because your talks have been fantastic today. So thank you very much. Number two, to the BPS team that have been at the front desk. The volunteers that have been helping us with the microphones, and the AV team as well. So obviously, without them, this wouldn't have worked so well. And thirdly, and most importantly, to you as well. Thank you for attending today and staying till the end. And so please have safe journeys home, and hope to see you again next year. So thank you very much, everybody.